ميزانين طهران رياض. ولا القالة دينار يمكن بعدين لما يحضر.
Assalamu alaikum wa rahmatullahi wa barakatuh. Good morning, everyone, to the second day of Saph 2022. And would like to welcome you all in this very basically scientific gathering. This is the 15th annual conference of the Saph. So you really all did part of that history. So, and today the first talk is also very interesting. It's about the basic science in the field of pulmonary hypertension. But really before I introduce the speaker, I would like to make basically announcement that at 1 o'clock today, you have a prostacyclin workshop. So it's basically about talking about prostacyclin and showing the prostacyclin basically delivery system and really other logistics. It will be at 1 p.m. at the mezzanine room at Dahran, Riyadh. That's, I think, at the mezzanine level. And in the afternoon, around at 3 p.m., there will be another workshop for the 6-minute walk test, which is as well, we do recommend attending. So without further ado, it's my great pleasure to introduce the first speaker. The first speaker is Mayel Zaidi. She's a research assistant professor, and she had a specific basically interest in the vascular biology and nanomedicine. She works at the moment at King Abdulaziz City for Science and Technology in Riyadh, Saudi Arabia. And she will have a talk today about the role of CLIC protein in the regulation of mitochondrial function in pulmonary hypertension. So please, the floor is yours.
بسم الله الرحمن الرحيم والصلاة والسلام على أشرف الأنبياء والمرسلين سيدنا محمد على آله وأصحابه أجمعين. Good morning, everyone. It's my pleasure to be here today and present my work. Thank you for Dr. Abdullah for his invitation. Thank you for your coming. Actually, I'll give a brief talk about my PhD project, role of click proteins in the regulation of mitochondrial function in pH. pH is a severe lung disorder, as we all know that, characterized by pulmonary vascular resistance caused by vascular remodeling. Endothelial dysfunction initiates this vascular remodeling but in pH, but the underlying molecular mechanisms are poorly understood. Mitochondrial dysfunction is a key contributory in this, the disease pathogenesis. Here is a schematic diagram showing the alterations of mitochondria in endothelial cells. We are focusing in this project just on the mitochondrial dysfunction, which is characterized by increasing the fragmentation and also the switch from glycolysis, from the oxidative phosphorylation to glycolysis, and also decrease in bioenergetic. Click proteins are a class of largely unselective chloride channels. There are 6 paralogs of click proteins that have been identified up to date. They localize to various intracellular organelles, and also they play a role in membrane trafficking, cytoskeleton function, and others. Our group have identified that click-1 and click-4 are elevated in lungs, plasma, and endothelial colony-forming cells, ECFCs in patients with PH, and also click-1 and 4 are structural components of mitochondrial membrane. Click-5 has not been identified in PH, but we studied CLIC5 because it's been localized to mitochondria in cardiomyocytes, and we included CLIC5 in some experiments. So the aim of this project was to study the role of CLIC proteins on energy metabolism, distribution, and function of mitochondria in primary human pulmonary endothelial cells, HPACs, and evaluate potential impact of these changes in the pathogenesis of pulmonary hypertension. I started my experiments by overexpressing CLIC proteins in HPACs using adenoviral gene transfer. This overexpression was confirmed by Western blotting and also by immunofluorescence. As you can see from the Western blotting that the CLIC was overexpressed by 3 to 4-fold increase that what was seen in vitro. Confocal images also confirmed the overexpression. CLIC5 has two isoforms, long and short. The short isoform is the predominant one, and we included that one in our experiments. Then I stained mitochondria to look at the mitochondrial fragmentation counts using NEP2 image software. Here are confocal images showing that mitochondria were fragmented in CLIC1 and CLIC4 overexpressing cells comparing to control cells. And the fragmentation count was highly increased in CLIC1 and CLIC4, but the mitochondrial coverage area was decreased in CLIC5 only. The same analysis was applied also in CLIC silencing cells, and we can see that the effect was reversed by silencing CLIC1 and CLIC4 in HPACs, and the mitochondrial fragmentation count was reduced. Here is just a video showing the movement of mitochondria along the cytoskeleton. And the cytoskeleton plays an important role in mitochondrial dynamics. That's why we included the microtubule organization and actin in our study. And also because CLIC1 and CLIC4 interact with some cytoskeletal and cytoskeleton-associated proteins such as dynein and tubulin. And we wanted to see if they have any direct effect on microtubule organization. Here is representative images of confocal showing that the CLIC1 and CLIC4 increased microtubule polymerization compared to the control cells, and the same there in the polymerization graph that they were increased, while in the circularity was decreased because CLIC5 induced very elongated microtubule organization here. Here are also some time-lapse video recording for 5 minutes. Tubulin is green labeled while mitochondria is red labeled. We wanted to see if there is any effect on mitochondrial alignment with microtubules. And can you play that one, please? The second one. Oops. Second one, please. Yeah, here we can see that mitochondria are going through fusion process, and there is no effect on mitochondrial alignment with microtubules. So we've seen just the changes in microtubule organization, but not in tubulin or acetylated tubulin expression. Then we used TEM, the transmission electron microscopy, to see the mitochondrial structure and also mitochondrial localization in CLIC overexpressing HPACs. And we can see that CLIC1 is localized to mitochondria, inner membrane and outer membrane, the same as for CLIC4. In the literature, it's been said that CLIC1 is not localized to mitochondria, while we prove that click 1 is in mitochondria. Here are some also TEM images showing the cristae, which is the structure of mitochondria, and we can see there is a reduction in the cristae number in click 1 and click 4 comparing to the control cells. As you can see from the images here, so this is the control cells. We can see the cristae are a lot, and you can see them, they are clear comparing to the other cells. So the mitochondria within cells go under divide and join process called mitochondrial dynamics. This mitochondrial dynamics is controlled by several proteins such as mitofusin 1, mitofusin 2, and OPA1. This is for fusion process, while in fission process, the DRP1 and FIS1 control the process. Here, I isolated mitochondria from H-packs overexpressing cells, and then we can see there is a predominant reduction in mitofusin 2, mitofusin 1, and OPA1 in click overexpressing H-packs. This will bring us to overexpress mitofusin 2 to see if we can reverse the effect of these protein. Also, I measured mitochondrial membrane potential using TMRM, which is dye that accumulates with active mitochondria. And I measured that in live cells using confocal. This is for cells overexpressing click 1 and click 4. Here, cells are silencing, so we can see there is a big effect on cells that has been silenced using shRNA, as you can see also from the graphs. Mitochondrial ROS also was measured to assess mitochondrial function using MitoSox dye, which is a red dye that accumulates in mitochondria within the superoxide, not any other ROS production. And we use the flow cytometry to measure that one, and we can see there is an increase in click 1 and click 4 overexpressing H-packs comparing to the silencing one and also the control. So the silenced cells were back to the normal cells. And also I applied the seahorse analysis, which is the technique that measures energy metabolism by monitoring pH and oxygen concentration in live cells. Also, they use some inhibitors to inhibit the ATP production. Within cells, energy mechanism we can form through glycolysis and respiration. And we wanted to see if there is any shift in this process caused by click 1 and click 4. As we said at the beginning, this is the characterization of the disease. So here, the analysis of the seahorse technique, there is massive reduction in click 1 and click 4 overexpressing cells. Comparing to the silencing cells, they are back to the normal level. Also in glycolysis, as I said, there is a shift towards glycolysis in order to give the energy demand to the cells. Here are some images of overexpressing mitofusin 2. As I said at the beginning, that mitofusin 2 were predominantly reduced in cells overexpressing click 1 and click 4. And here I overexpressed mitofusin 2 in H-packs to see if the effect was reversed. And this what we can see, that after overexpressing mitofusin 2, mitochondria were not fragmented and the mitochondrial network is back to normal. Also, I assessed mitochondrial membrane potential, and we had the reverse effect comparing to the overexpressing cells. Also, mitochondrial ROS production was measured in cells click over expressing and silencing cells, and we can see in click over expressing there was a reduction in click 1 and also not in click 4. Seahorse was also applied to measure the glycolysis and the oxygen consumption rate in these cells. And we can see that the cells back to the ATP production was highly elevated in those cells comparing to the control cells, or the cells that were overexpressing click proteins. Glycolysis was reduced to normal cells or the control cells, while, as I said, with overexpressing glycolysis was elevated. Here is the study using the ECFCs, which are the endothelial isolated from patients. We confirmed, just a reminder, that CLIC1 and CLIC4 are elevated in PH patients, and this is a confirmation. This work has been done by our postdoc. They measured the CLIC1 and CLIC4 in plasma, as I said, and also in some of the tissue. This is the immunohistochemistry. Mitochondrial fragmentation was also assessed in these cells to see if we can see the same effect that we were trying to mimic using CLIC1 and CLIC4, since they are from patient cells. So CLIC1 and CLIC4 are already elevated in these cells, and we wanted to measure the effect after overexpressing mitofusin 2. So you can see from the confocal images that the healthy control with mitofusin 2, they were elongated, and this is just healthy control with no overexpressing. And these are the pulmonary arterial hypertension cells, endothelial cells. Mitochondria were fragmented, and after overexpressing mitofusin 2 on these cells, the mitochondria were back to normal. Membrane potential was also assessed in these cells, and we can see a massive reduction also in HPACs, not HPACs, in the PAH cells, overexpressing mitofusin 2 comparing to the control PAH cells. Also, we assessed the glycolysis, and glycolysis was reduced, and ATP production was elevated. And this is the final graph that we confirmed that this study is the first that showed that CLIC1 and CLIC4 regulates mitochondrial endothelial function in pulmonary hypertension, and we could use that one for, as a therapeutic target in the future. I'd like to thank my PhD group at Imperial College, and also I'd like to share this with you, the achievement that I had during my PhD, the first place award for image immunofluorescence from APCAM across Europe. And these are the images that I contributed with. And thank you for your listening.
Thank you. Thank you very much, Dr. Zaidi, for this wonderful review of the topic. Is there any question from the audience? Yes, please. If you can use the microphone at the back there. Yes, go ahead. There's the mic at the back there.
Assalamu alaikum wa rahmatullah. Thank you very much, Dr. Mai. I really impressed by this research and the level and the use of electron microscopy, and it is really great effort.
Thank you.
I have only one comment about the title itself and the research. Of course, from what you said, it may be refined to the subgroup of idiopathic pulmonary hypertension. But the title of the lecture itself, for pulmonary hypertension, it is 5 clinical groups. And from pulmonary arterial of hypertension, we have a congenital heart disease. So this great effort need only to be refined, that the research was a specific subgroup, which we all understand that it is mainly the idiopathic group.
Yeah.
Because what you did is really, I cannot find word to say how it is great. Thank you very much.
Thank you so much.
Okay, so can I ask a question?
Yeah, definitely.
Now, did you have kind of basically correlation between the click expression and the severity of the pulmonary hypertension? I mean, did you have some kind of clinical correlation between, would those people, they have more severe disease, for example, look for hemodynamics or outcome?
Actually, there were randomly just chose, so there is no severity in this study.
Okay, thank you. All right, so if there is no other question, we will move to the basically next speaker. Dr. Zaidi can join us here, please. And the microphone now is with Dr. Abdullah.
Thank you, Dr. Zaidi, and thank you, Majdi, for the introduction. And it's my pleasure now and honor to introduce the second speaker, who is a colleague of mine, Dr. Faiga Ahmed. Dr. Faiga is a senior clinical scientist who works at King Faisal Specialist Hospital. Faiza works also particularly in the Genomic Center at King Faisal Specialist Hospital. She got her PhD in human genetics from University College of London. Faiga is going to talk about an insight into the genetic basis of PAH in a population of Saudi Arabia. Go ahead, please. Thank you so much.
Thank you, Dr. Dalan. Good morning, everybody. It is a real pleasure to be here today. I don't really get out of the lab much, so Dr. Dalan, he invited me, and I couldn't say no, of course. So I'm going to shift from the cellular level into the genomic level for you this morning. So the title of my talk is An Insight into the Genetic Basis of PAH in the Saudi Population. So as a geneticist, I will start with population genetics of Saudi Arabia. Our current population is around 36 million. We have a tribal nature where structurally the tribe is defined by a common descent through which a large number of individuals are united. In some rural areas of the country, tribes may have a very high consanguinity rate, consanguinity meaning descended from the same ancestor, and therefore may be considered genetic isolates. It has one of the highest consanguinity rates among all the Arab countries. The overall rate of consanguinity is between 52 and 57%, and it can reach up to over 80% in certain rural areas of the country. First cousin marriages are very common here in Saudi, and it ranges between 29 and 40%. And because of all of the above, autosomal recessive genetic disorders, especially novel ones, are frequently observed in the clinic setting for mostly all clinical specialties throughout the hospital. And many founder mutations causing different autosomal recessive disorders have therefore been identified. So a little bit about the genetics of PAH. As you all know better than me, it's a rare and severe disease affecting small pulmonary arteries caused by abnormal proliferation of their smooth muscle cells and endothelial cells. This results in increased pulmonary vascular resistance and right ventricular failure. The pathogenesis of idiopathic PH, or IPH, is linked to a number of susceptibility genes, including the bone morphogenic protein receptor type 2, the BMPR2 gene, and this codes for a type 2 receptor member of the transforming growth factor beta family. Mutations in BMPR2 are seen in over 80% of cases with inherited PAH, and in the idiopathic cases, around 25% of these mutations are de novo or new mutations. Only about 20% of the population with BMPR2 mutations go on to develop the disease, which suggests an incomplete penetrance of the gene and a second hit mechanism based on major gene and additional modifier gene has been suggested. The clinical presentation can be variable within the same family, and the incidence is significantly higher in females. Okay, now on to our study. So this was a IRB approved project at King Faisal Specialist Hospital and Research Center. Dr. Abdullah Dalan is the principal investigator and the core of this whole study to start with. The title of the study was Genetic Basis of Pulmonary Arterial Hypertension. It's a prospective study from a highly inbred population. This was carried out over a period of 2 years, and the results were published in Pulmonary Circulation in July of last year. Okay, so a little bit of background first. So about 30 genes are implicated in the development of PAH, and 21 of these genes have shown specific pathogenic mutations. They are mostly autosomal dominantly inherited and exhibit reduced penetrance, meaning again that some individuals who carry a mutation don't always manifest the disease. In IPH cases, the mutation could be inherited from an unaffected parent or occur de novo, and with all genetic diseases, where parental samples are available, determining if a mutation occurred de novo can be helpful in establishing whether it's pathogenic or not. The incomplete penetrance for PAH genes suggests that additional genetic, epigenetic, and/or environmental factors contribute to disease risk or progression. Apart from a study that was done in Lebanon a few years back, I think they had 21 patients, the prevalence of the genetic abnormalities in patients from this region, the Middle East, including Saudi Arabia, hasn't been reported before our study. And therefore, the main objectives of this study were to identify the genetic variations and their frequency in risk genes in Saudi PAH patients, and to go on to determine any possible genotype-phenotype correlation. Okay, clinical methods. This was a prospective, non-randomized study, including patients enrolled in the pulmonary hypertension disease or the Saudi IPH registry. Patients fulfilling the inclusion IPH and pulmonary veno-occlusive disease or PVOD were included in the study and followed up according to routine clinical practice at the section of pulmonary medicine at our hospital in Riyadh. In total, 48 Saudi patients were enrolled that fit the criteria with full informed consent. The inclusion criteria were adults over the age of 18 years with a diagnosis of PAH defined as per the American College of Chest Physicians and the European Respiratory Society guidelines as Group 1 pulmonary hypertension PH, while excluding all known causes, including drugs, connective tissue diseases, portal hypertension, HIV infection, and congenital heart diseases. Patients with PH with associated conditions such as pulmonary venous hypertension were excluded from the study. Okay, so this is a table showing the clinical characteristics of the study cohort. So again, 48 eligible patients were enrolled in the study. From these 48, 46 were diagnosed with IPAH and 2 patients with PVOD. From our cohort, 5 patients were documented to have a familial history of PAH. And most patients were above the age of 20 years old and with a median age of around 30 years. The overall ratio of females to males was 43:5. Okay, my part. Molecular genetics. Okay, so genomic DNA was extracted from peripheral blood from all of our 48 patients, and we carried out whole exome sequencing, or WES, by the ion proton system in-house. We generated an aligned data sets, which were analyzed and filtered using bioinformatics software using our in-house Saudi Human Genome Program, SHGP pipeline. The reads were aligned across the HG19 human genome reference sequence, and all of the variants were annotated using a combination of public and in-house databases. The analysis pipeline that we use is designed to filter out non-relevant variants based on quality, functional characteristics, and frequency in the database, including our in-house Saudi Human Genome Project database, which is very vital to this population. And then further analysis focused on the identification of plausible pathogenic missense, nonsense, frameshift, and splice site variants in 26 genes related to PAH. Okay, this table shows the 26 genes that we chose to screen our patients from the WES. The 26 genes were chosen out of 30 because they were all linked to an OMIM number, which is online Mendelian inheritance of man. And the inheritance varied from autosomal dominant and autosomal recessive inheritance. Okay, the first one is a very small snapshot of actually what WES analysis looks like after filtration if we're doing it manually. I think it starts with like 30,000 rows, and from then we filter using our pipeline and our in-house database. And then we get roughly about maybe 200 to 300 rows that have variants that you would consider, interesting. But because we already know the genes we're going for, the 26 genes, so this is actual patient data. So we will specifically look for these 26 genes and filter them out. And in this specific patient, in this specific patient, you can see that this is quite light. This is one of the genes that we were searching for, and indeed, this patient turned out to be homozygous for I believe it's a nonsense mutation in the exon of this gene. Okay, so the overall results for the molecular genetics. So from 48 included cases in the cohort, 16 of our patients identified a molecular variant in one of the 26 PAH risk-associated genes. Mutations were identified in 30% of the IPH patients and both of the PVOD patients. The two PVOD patients were diagnosed at a very early age, and they were subsequently identified with mutations in EIF2AK4, and they were homozygous for this gene. This table shows the 16 different variants. So you can see that EIF2AK4 were homozygous, so 1, 2, 3, and the rest were heterozygous. It's what we were probably expecting. And in turn, I would like to say that 14 of the 16 variants had not been reported previously. And so this provided a great insight into the genetic basis in our population. This is just a simple pie chart showing the variance and the distribution. 32 were unexplained, and the highest number were in the BMPR2 gene, and then probably EIF2AK4 was more common. A little bit more about the BMPR2 findings. So we identified 4 heterozygous variants in this gene, 2 missense, a frame shift, and a known splice site mutation. And based on in silico analysis that was in the first table of results, they were predicted to be likely pathogenic by the American College of Medical Genetics and Genomics. These mutations were identified in female patients with family history, so it confirmed that these variants do underlie familial PAH, and with reduced penetrance and overrepresentation of females. And the identification of BMPR2 mutations in 4 IPH confirmed the high associations of these mutations with this disease. We also identified 3 homozygous pathogenic variants in the EIF2AK4 gene, 2 nonsense, and a splice site variant. The diagnosis of PVOD among our patients with homozygous pathogenic variants in this gene is in agreement with previous association between these mutations and autosomal recessive PVOD. And the detection of pathogenic mutations in this gene in particular is extremely beneficial because it establishes a precise and accurate molecular diagnosis of PVOD without requiring a lung biopsy. So in this prospective study, we identified genetic variants and frequency of risk genes in IPH and PVOD patients from Saudi. Our results do highlight that the genetic contribution of this disease in Saudi patients through the identification of variation in a total of 9 genes so far previously associated with PAH. In our study, however, we could not confirm previous findings that evidence very young ages at diagnosis in PAH patients with positive gene mutations, except for those with EIF2AK4 mutations. And regarding disease severity, our results showed a tendency for higher disease severity in patients with positive mutations, mainly in those with mutations in that gene. So, the overall findings of this study do highlight the importance of genetic counseling and genetic analysis and counseling in PAH patients. or their relatives, even the asymptomatic ones, with particular emphasis in PVOD patients with the homozygous mutations, that can greatly benefit from the early diagnosis and specific therapy. Genetic testing in general, especially here, can enable the identification of mutation carriers, leading to early diagnosis among relatives of known patients. And it can also provide pre-pregnancy counseling for couples with familial mutations and premarital counseling in severe cases. But I have to mention here that the West platform that we used is not amenable to large gene deletions, duplications, or copy number variants, so we can't exclude that variations in the genes that we checked for are not there, or have yet to be identified. So to conclude, our study is the first to report the underlying molecular basis of IPAH in patients of Saudi Arabian origin. It really highlighted the important contribution of genetic causes for PAH, and this approach, when applied prospectively, is of great benefit to personalized diagnosis and patients' management, while laying the foundations for future prevention. Further studies are required, and we hope to do that in the future. Firstly, on a larger number of patients, and hopefully this will expand the mutation spectrum in this population. And for those 32 patients where the West was negative, perhaps we have to look for novel genes or do cytoarray or chromosomal studies to exclude or include the possibility of large deletions and duplications. So as a geneticist, and I know I have a whole of pulmonary medicine doctors, I do want to highlight this to you. This is a direct quote from my very good friend and colleague, Professor Fawzan Al-Correa in the Center of Genomic Medicine. And this is from one of his papers. So genetic studies on the Saudi population have contributed significantly to the global effort of identifying the causal mutations of Mendelian diseases, particularly in the area of autosomal recessive diseases. The very characteristics that make this population a significant contributor to the analysis of recessive diseases, i.e. consanguinity and large family size, have also recently been exploited to advance our knowledge to other areas of human genetics research, including dominant disorders, by reclassifying dominant mutations when observed in homozygosity in individuals who lack the reported dominant phenotype. And that's very important with PAH. And to highlight how rich Saudi is on genetic basis, this is just a snapshot of a few papers that coming out of my section at King Faisal. And as you can see, we collaborate with literally every department in the hospital. We have, of course, pulmonary medicine. We have our OBGYN and metabolic diseases, deafness, nephrology. So always keep an open mind for genetic analysis for your patients, especially for patients in this population. Finally, of course, I would like to thank our chair and our president, Dr. Abdullah Al-Dalan. Fantastic, lovely working with him and his group. My scientist, Dr. Khushnud Ramzan, excellent pulmonary medicine doctors in our hospital, Dr. Safraz Salimi, Dr. Ihab, my technician, Layla, Dr. Abir, the coordinators of our hospital. I think they do more work than us sometimes, and Fatma, Hamdiya, Nadine, Rana, my medical geneticist, and from medical genetics, Dr. Muhammad Alawain, of course, the Center for Genomic Medicine at King Faisal, and our core facilities for doing all the bench work, and thank you for your attention. Thank you so much.
Thank you so much, Faiga, for this an outstanding presentation. Thank you so much.
Thank you.
We can have questions now. Dr. Hana, please.
Thank you very much. Really interesting case, and thanks for the group. Really very nice. I just want to compare your finding to our finding in cystic fibrosis. So when you did the test, the ways,
Yes.
We know that we have a pulmonary panel. Was that applied with it, like as a screening tool, for example, when you started?
Sorry, so you use a pulmonary panel?
Yeah,
for
there
patients?
is in the hospital. I'm from King Faisal, Riyadh.
Oh, okay.
And we do the same testing. But because there is pulmonary panel that tests for bone morphogenic protein and pulmonary hypertension that is already present. Like, did you run that test before, or you just went into with the whole genome?
No, so I'm really interested where you do your pulmonary panel, because did you send it out? Because we don't have one at
No, it's in hospital. It's in-house.
Okay, we have, okay. So the pulmonary panel, from what I know, the gene panel, if this is what you're, the pulmonary gene panel?
Yes, yeah.
Okay, so we are actually the ones who run the gene panel, so I'm a bit, to be honest, confused, but in our study, we did whole exome sequencing, so every gene known to man that has been identified, we have the data for. But in that set of data, we looked at 26 specific genes that have been reported in the literature to be associated with PAH, and it's as simple as that. But if we wanted to look at even other genes for other diseases, we could look at it. We have all the data for the whole exome.
But
Yes.
with this way, it means that, like also, I compare it to cystic, you have missed other genes that is related to pulmonary hypertension, actually, because you only look to whatever reported, right?
Yes.
So,
That's the aim of the study.
sorry?
That was the aim of the study, to look for...
Yeah,
yes,
exactly. We were trying to, we went beyond the scope of the pulmonary panel, to be really honest, because we even looked at diseases where it was not straightforward pulmonary hypertension, arterial. We looked at even papers that even had it as even a co-finding in a patient. So our WES will extend over any panel that you can look for. And especially what's also very important in this population that I'd probably like to highlight now is that panels that are amenable to Western populations and in the West don't always pick up mutations in the Saudi population, okay? So
Yeah, exactly, like in cystic.
Right.
When we send our cystic patient, only 30% we identified. But then when we did our panel, we had almost 60% of our population
Right.
were positive, but were not reported before. So
exactly.
the same.
So this is why, research like this is so important, because it will show you the mutations that you need to be looking for and the genes.
But you only now mentioned
Can you, Doctora, because we are running out of time, can you please summarize,
Okay.
make it a little bit short?
Anybody who has a question? No, because we really need to understand. We have a lot of pulmonary hypertension patients.
So I
also
have the
in
best
pediatrics.
solution for you, and it's very simple. The Center of Genomic Medicine now offers clinical WES to every single patient in King Faisal through the CGM. You can order on ISIS, and you can have this for your patient as we speak on a clinical basis, whole exome sequencing.
Yeah, but look at your result. You only identified 30%. So what about the others? They are confirmed pulmonary hypertension.
Yeah, I know, but we were only looking at 26 genes. But when we are looking at the whole data, and now we have programs that will also link the disease to the findings, so you will probably get more hits now if you submit for clinical WES.
Okay, Doctora, just to make it short for you, the aim of this study was actually to look for the incidence of the known genes as a cause for pulmonary hypertension in our cohort. We were not looking for other new novel genetic mutations. We focused only on those 26 genes.
But that would be good if you look at it.
That will be the second phase of the study, inshallah ta'ala. Okay, any more questions?
I have a question.
Yeah, go ahead.
Go ahead.
This one, Luke.
Thank you very much. Mine is a very non-clever question. You may have said this, but the PVOD cases that you identified, were they previously identified as PVOD clinically, or did you change the diagnosis?
I can answer that. Yeah, actually, the three of them were identified clinically as PVOD.
So okay,
Yes.
fine. Because often we find that you get
Yes.
the homozygous mutation, and then they don't have clinical features. So the second part of the question is, given the consanguinity that you have, do you have any idea of the overall prevalence of PVOD in Saudi and whether it's higher than in other populations?
I don't know, can you answer this?
Apart from this study, I haven't actually done any genetic workup on other PVOD patients, but that could be something that we could probably do.
If you look at the number, actually we identify 6% of our 48 patients having PVOD. I think it's probably slightly more than what has been published as a cause of pulmonary hypertension, genetic cause of pulmonary hypertension in the West Society. Not only this, if you look at also to the total cohort of 48 patients, we identify 33% of these patients, or 33 having positive genes.
Yes.
This is also higher than the West.
Yeah.
So about 50 patients, 30 of them having genetic mutation. That's a lot.
Yes. Yeah, so then potentially you're sitting on a bigger problem, potentially.
Yes.
Yeah. Thank you.
Thank you.
Thank you very much. On a practical basis, you mentioned something about the genetic counseling. Did you start in King Faisal any genetic counsel clinics? And if you did, because it's really a little bit hard, especially in our part of societies, when you ask the wife not to get pregnant, you know what will happen to the marriage, right? Or don't get married to your cousin or something like that. So how do you think that the genetic counseling will be effective? Thank you.
So I'm going to boast a bit. I think that we have one of the most active genetic counseling individuals and programs probably in the kingdom, okay? And the Saudi population are very knowledgeable and they are very willing to undergo counseling, especially premarital and prenatal counseling as well. I am actually, with this, I'm heading the prenatal genetic testing of the hospital. I have for the past 10 years. And the number of ladies that come in for prenatal testing and that have had counseling for terminations is phenomenal. It's about 98%. So yes, genetic counseling is very strong, especially with a lot more awareness now. And the taboo and the culture issue, it's really moved ahead a lot in the past few years.
Thank you, Dr. Faika, for your great work.
Thank you.
I have a comment on the first question. When we do a study like you did, I have 100 genes or 30 genes which are implicated in the mutation associated with pulmonary hypertension. So that's what you did in your study. Why I will do the genes which is sodium channel transport in the respiratory panel? Nobody said that. So what you did, that you have a genes which is implicated in pulmonary hypertension, you was identifying how it's contributing to your population. And this is completely right. If the genes is not involved in the pathogenesis of the pulmonary hypertension, why I will test for?
Exactly.
This is the answer of the first question.
Okay.
I will not do the respiratory panel because there is no previous study which showed that sodium channel gene mutation has an implication in the pulmonary arterial hypertension. So why I will do it? There is nothing scientific which is pointing towards this.
Right.
We have millions of genes. So what is implicated, we test it to see how much it is sharing in our population.
Yes.
But who said that sodium channel gene mutation has a contribution to pulmonary arterial hypertension? Nobody said that. This is the very simple answer. Thank you.
Thank you very much.
Yeah, thank you for your talk. I just have a question regarding the age, the mean age in the Saudi population is much lower than the Western one. And if the genes have been identified are same to the Western one, is there any differences in the genetic backgrounds or, the genes that have been identified in the Saudi population that could be related to age?
With dominant disorders, it's quite hard to tell, and because of the very different penetrance. So I'm not too sure, but maybe doctor, on a clinical basis, they would be able to answer that a lot better than myself.
Okay, thank you.
Thank you so much.
Okay, thank you so much. So please join us here. Now we're going to move to the third talk and the last talk of this first session. It's my pleasure to introduce Professor Mark Umbelt. Professor Umbelt will be with us virtually from Paris. He's professor of medicine from the University of Paris, and he is the head of the basically internal medicine, ICU, and pulmonary hypertension programs. He's basically world authority in the field of pulmonary hypertension with multiple publications, and he is currently the president of the European Respiratory Society. So Professor Mark will be talking to us about the basic MPR2, how to target this pathway in pulmonary arterial hypertension. Mark, the floor is yours.
together with remodeling, you have some evidence of organized inflammatory reaction. So the take-home message from this cartoon is that when you display pulmonary arterial hypertension, you already have significant remodeling of your small pulmonary arteries, and we need to understand how it works in order to try to counterbalance the mechanisms leading to that. Classically, and we are very fortunate in the field of pulmonary arterial hypertension, traditionally, we have three major pathways of endothelial cell dysfunction. We may target with endothelin receptor antagonist, PDE5 inhibitors, guanylate cyclase stimulator, prostacyclin derivatives, or prostacyclin receptor agonists. And I'm not going to discuss that today. What I would like to tell you is that all these dysfunctional endothelial pathways are due to our consequence of causal mechanisms And the causal mechanisms of PAH need to be better understood. We understand very well the consequences. We know how to interfere with some dysfunctional pathways, but causal mechanisms are very, very important to better understand, better describe. And the beautiful presentation before my talk clearly show us that we understand better and better what lies in the genetics and in the basic biology of the cells of the pulmonary arterial wall. And this could be certainly leading to future treatment targets. And we just heard that BMPR2 is a key genetic causal condition leading to an increased risk of PAH in the community. There are many other genes, of course, but BMPR2 is certainly a gene of major importance. And to make a long story short, you have to consider that BMPR2 is in fact inducing and transducing signals which limit proliferation of the cells in the pulmonary artery wall. So BMPR2 is a kind of anti-proliferative pathway. And when you have a germline mutation in the BMPR2 pathway, either BMPR2 itself or any member of the pathway, you will have a diminished anti-proliferation. So the end result will be increased proliferation and thus remodeling. And thus we have patients who carry germline mutations, which in turn will promote proliferation of the small pulmonary arteries I just showed you. On top of that, the functional BMPR2 usually protects against endothelial cell apoptosis. And when you have more dysfunctional BMPR2, you will have more survival of the wall cells, and this will also lead to increased remodeling. So at the end of the day, we have to understand carefully the BMPR2 pathway, and I'm not going to describe that in great detail, but in year 2000, it was a major year. We realized, the whole community realized, and we just saw that it is the case worldwide, that BMPR2 mutations can be detected in the large majority of patients with a familial history of PH, but also in around, let's say, 20–25% of patients with apparent idiopathic PH. And what is really important is that PH patients carrying a BMPR2 mutation, they are younger at diagnosis, they have a worse prognosis when compared to non-carriers. So at the bottom of my slide, you can see that in a normal individual, you have full function of BMPR2, but in patients carrying heterozygous mutations in BMPR2, you have a 50% decrease of this pathway, and this is exposing you to an increased risk of developing PH. On the left-hand side, you can see the BMPR2, which is a receptor of the TGF-beta superfamily. Below the receptors, you have signal transduction, and then the effect at the cell level. When you look at the literature, and we just heard that, not only BMPR2 can be dysfunctional in familial or heritable PH, but you have also many other genes which are disturbed and dysfunctional in PH patients. But as you can see here, many are in the BMPR2 pathway. Of course, BMPR2 itself, type 2 receptors of the TGF-beta superfamily, but also type 1 receptor like ALK1, type 3 or accessory receptors like endoglin, and of course the transduction signal like SMAD1, SMAD8, etc. So not only BMPR2, but the whole pathway can be affected in heritable PAH, and in fact, even in idiopathic disease, you can find disturbances. So our understanding of these causal mechanism is that it predisposes you to develop PH. So when you have abnormal BMPR2 genetic background, you will have a reduced availability of this anti-proliferative pathway. But what will be important, these are the second or multiple hits on top of this dysfunction. Gender is a problem. When you are a female, you have more than twice the risk of developing PAH in the setting of a BMPR2 heterozygous mutation. Additional genetic factors might be involved, and other issues such as immunity, environment, and oxidative stress can be involved. So interestingly, we have been able to generate rat models of BMPR2 mutation, and Aurélie Hautefort and Frédéric Perrault in my lab, as well as Mark Nichols in a collaboration with Stanford University, they both were able to show that rats with BMPR2 mutation might develop some kind of increased pulmonary pressure in the pulmonary circulation. And what was interesting is that it was affecting only a subgroup of rats, indicating that you might need second hits. And one of the second hits demonstrated by Tian in Stanford University in a collaboration with my group is that when you have inflammatory triggers, you can induce more pulmonary hypertension phenotype. So now we understand that BMPR2 is dysfunctional, and this is the pathway going from the ligands binding to BMPR2 together with a type 1 receptor, transducing the signal through the SMAD pathways and going to the nucleus to bring the end result of the pathway. And you see that you have several numbers here, 1, 2, 3, 4, 5, 6, 7. And these are all strategies which could be used to restore the BMPR2 signaling pathway in the setting of a disturbed BMPR2 signaling. So I'm just going to read through it, and I will give you an example in the next 7 slides. So first, you could increase BMPR2 protein cell surface expression thanks to endothelial gene therapy. That's the most simple thing, but it's difficult to achieve. Second, you can increase BMPR2 level using small molecules that increase translational read-through of BMPR2 mutations encoding premature termination codons, and we will see that with ataluren. Third, chemical chaperones that increase trafficking of BMPR2 misfolded mutants that are held up within the endoplasmic reticulum can be used, at least in theory, to increase BMPR2 availability when there is a misfolded mutant held up or trapped in the endoplasmic reticulum. Then you can restore BMPR2 signaling by ligands that target the BMPR2 receptor complex on endothelial cells. Fifth, you can enhance downstream SMAD phosphorylation with agents that will target the SMAD signaling, such as FK506. Then you can inhibit BMPR2 turnover by the lysosome, inducing cell surface increased expression of BMPR2 protein. And last but not least, as I did present last year at your meeting, you can attempt to rebalance the TGF-beta superfamily pathway with a recombinant fusion protein containing the extracellular domain of the human activin receptor 2A, acting as a ligand trap and thus rebalancing what is dysfunctional in the cells of the pulmonary vessel wall. So very briefly, because I'm conscious of time, I will just give you a few examples. So in our patients with mutations and even without mutations, you have a decreased functioning of the BMPR2 pathways. You are at the mid of your usual function of BMPR2 pathway. And you wish to increase this dysfunctional pathway to 100%. So you can target the endothelial cell, offer BMPR2 cell surface expression increase with endothelial gene therapy. Our colleagues in Australia, in the UK, in the States have showed convincingly in animal models that this is achievable, but we are far from human availability. Another possibility, and it's summarized here, when you have BMPR2 mutations encoding premature stop codon leading to immature BMPR2 protein, you can use small molecules such as ataluren, which will increase translational read-through of BMPR2 mutation encoding premature termination codon. So rather than having an immature BMPR2 protein, you will have a mature BMPR2 protein. So this is very promising. This is very early. This is non-specific. And this has been proposed by Michaela Aldred, Nick Morrell, and colleagues in BMPR2 and SMAD9 mutations. There is a theoretical background, but a word of caution is that in cystic fibrosis, and I heard your discussion about CF, ataluren did not succeed to restore cystic fibrosis CFTR stop codon. So it is promising, but it has not been tested in humans with BMPR2 mutations. Another interesting approach is to try to improve the trafficking of mutant BMPR2 which are trapped in the endoplasmic reticulum. And chemical chaperone like 4-PBA summarized here, they can increase the trafficking of BMPR2 misfolded mutants that are held up within the endoplasmic reticulum. Once again, Nick Morrell, as well as Asian colleagues, have been able to show convincing preclinical data, but we are far from a human solution yet, and this is a non-specific approach. The fourth possibility is to increase not the BMPR2 itself, but the BMPR2 signaling by increasing ligands that target the BMPR2 ALK1 receptor complex on endothelial cells. So rather than trying to increase BMPR2 itself, you increase the ligands which are going to bind to this complex. And this is again from Nick Morrell laboratory. You have the possibility to offer a BMP9 solution to experimental models of PAH, and maybe one day to PAH patients with BMPR2 dysfunctional pathway. So the idea here is to offer increased BMP9 in patients with PAH. Of course, this is complex. This is a very redundant pathway, and there are some discordant results showing that in some animal models, disturbed BMP9 can protect against PH. So of course, there are more arguments in favor of increasing BMP9 availability in PH, but we are still working on that. And I would like to emphasize the quality of the work of the Cambridge group on that topic. I'm going to progress now with agents that enhance downstream smart phosphorylation. You can see here that in patients with BMPR2 mutation, you have dysfunctional BMPR2 pathway, and you may, with FK506, restore normal signaling in the setting of patients with BMPR2 dysfunction. This has been tested by the Stanford group, and you can see very interesting preclinical data here, and also some interesting proof of concept data published in the ERJ by Roham Zamanian and Edash Picocotter in 2017. Inhibition of BMPR2 turnover by the lysosome can also enhance cell surface expression of BMPR2 protein. And this has been published back in 2013. And here you can have an enhanced cell surface expression thanks to agents we know very well, like chloroquine, which have been very publicized recently. Last but not least, we published last year in the New England Journal of Medicine the first convincing human data targeting disturbed TGF-beta superfamily pathway. And here the idea is a bit different. Rather than increasing BMPR2 action, rather than increasing BMP9 action through BMPR2, rather than increasing the transduction proteins, etc., we have proposed to block not the anti-proliferative pathway, which is already dysfunctional, but the pro-proliferative pathway, which is translating through activin receptor 2A and 2B. And here we have a ligand trap, which will block the ligands which are promoting proliferation. And last year at your Saph meeting, I did present for one of the first time the PULSAR study results, a phase 2 double-blind placebo-controlled study comparing the efficacy and safety of sotatercept versus placebo when added to standard of care for the treatment of PH, with a 20% reduction in PVR with 0.3 milligram per kilogram sotatercept and 34% reduction in PVR with 0.7 milligram per kilogram sotatercept, with also increased walk distance, decreased NT-proBNP, improved functional class, on top of single, double, or triple traditional pH therapy. So this is, of course, a groundbreaking result. Based on these phase 2 results, we now enroll patients in phase 3 trials. Stellar has been nearly completed in terms of recruitment, and we should have results within a year. Hyperion is recruiting, and Zenith is recruiting. These are new phase 3 trials testing the efficacy of sotatercept on top of pH therapy. So my conclusion and take-home message is that BMPR2 and its signaling pathway are promising therapeutic targets in PAH. I have summarized on this slide all the intervention I did show you today. The most advanced drug solution is certainly sotatercept, and I look forward to your questions, and I'm ready to interfere with you remotely. Thank you very much for your attention.
So, Mark, with the PULSAR trial, it's quite impressive end results, and you would expect to replicate the same result in phase 3 clinical trials? And are you doing it differently than the phase 2 clinical trial?
Thank you very much, Abdullah, for your kind comment. So clearly, PULSAR was a phase 2, so it was aimed at showing the right first safety and efficacy and identify the right dose for future phase 3. So the primary endpoint of the phase 2 was pulmonary vascular resistance, and the prominent secondary endpoint was 6-minute walk distance. They both were positive, and we could achieve that the right dose range between 0.3 and 0.7 milligram per kilogram. Drug, so you give it every 3 weeks. It's like a biologic therapy in asthma. You know, it's a syringe, you give it sub-Q every 3 weeks. Now for the phase 3, we need to have approvable endpoints, And the agencies only approve drugs which improve how patients feel, function, or survive. So the STELLAR study has a primary endpoint of 6-minute walk distance, so how patients function. And the Hyperion and Zenith trials study time to clinical worsening, if you wish, and they focus on 2 different populations. One population which is early in the course of the disease, within a year of introduction of combination therapy in PH patients, and the other one, more advanced patients where you may hit survival endpoint. So all these studies are very well done, and we hope to have informative results first for STELLAR in 2023, and then for the whole program in the next 4 years.
Patients have to be on double combination therapy, or even monotherapy have been included?
Yeah, it's a good point. So in PULSAR, people should be treated because we considered that you need to be treated to be offered such a therapy because it's investigational and we don't know what is the efficacy. So in STELLAR, 56% of patients in, PULSAR, 56% in the phase 2 trial, 56% of patients were on triple combination therapy. 37% were on triple combination therapy, including a parenteral prostanoid. But there were also patients on double combination therapy and a small minority of patients on monotherapy, only 10%. But if you look at STELLAR, Hyperion, and Zenith, the least we wish to have is double combination therapy. And this will be, I think, very interesting data.
Studying the BBMPR2 in other groups of pulmonary hypertension, specifically, for example, in group 3, where we can help our patients that using this augmenting, genetic augmenting therapy and not developing pulmonary hypertension, for example, or just patients like HIV, sickle cell disease or so? Thank you.
Yeah, thank you very much for a great question. So, of course, currently we focus mostly on group 1 PAH. So in group 1 PAH, you can have HIV, you can have scleroderma, you can have congenital heart disease. So that's a significant group of patients we see in the clinic on a daily basis. But now many groups have identified that in patients with severe pulmonary hypertension and comorbid lung disease or even heart disease, you have a disturbed BMPR2 pathway. And the company developing sotatercept, which was Acceleron, but now it is Merck, because they have been bought by Merck, they have a program on group 2 disease, which is called Cadence. And also they are considering group 3. So you are right. I mean, this dysfunctional pathway is not unique to group 1 PAH. And there are concepts which indicate that maybe it would be wise to test it also in other subgroups. But currently, the focus is mostly on group 1 PAH.
Thank you, Dr. Majdi Idris, for co-chairing. Thanks, everybody, for attending this outstanding morning session. I think we are about 10 minutes late, so we should be back by 10:45. 10:45, exactly. So we have time later on for after the next session for Jum'ah prayer, inshallah. Thank you so much.
Assalamu alaikum. Assalamu alaikum. So, welcome everyone for the second session. It is my pleasure to co-chair this session along with Dr. Nasser Abu Zaidi. He's a pulmonologist from Oman. And we have a heavy schedule ahead of us. So we'll start with Dr. Sitbon, who's going to talk to us today about risk stratification, which goal has maximum impact. And Dr. Sitbon does not need any introduction. He's a well-known figure in the world of pulmonary hypertension from France, and many publications that speaks for themselves. So Dr. Sitbon, please go ahead.
Thank you. Thank you very much for your kind introduction, and I would like to thank again Abdullah and the organizer for their kind invitation to this meeting. Last year it was just virtual, but it's a real pleasure to be here today again. So now we will discuss about the risk reduction in pulmonary arterial hypertension, and we will discuss mainly about the goal of therapies. That's my conflict of interest. So the first question is, what is the ideal treatment goal in PAH? And I think we have to discuss if it is for the doctor or if it is for the patient. I think for the doctor, we have many very strong endpoints like improved survival, reduced hospitalization, etc., improve RV function. But for the patients, it's exactly the opposite. They want to live better. They would like to have a normal daily life activities. They would like to avoid hospitalization, etc. And I like this slide. This is a slide courtesy of Jean-Luc, of the clinical outcome endpoint in heart failure trial, but it can also apply to the phase 3 PAH trial or in clinical practice. If you look at the most scientifically rigorous endpoint like mortality, cardiovascular mortality, and cardiovascular hospitalization, you have this on the top, and just at the end, you have the symptoms, you have the worsening of heart failure without hospitalization, etc. But if you look at what is relevant for the patients is really, really different. And for the patient, the most important for them is symptoms, exercise tolerance, quality of life, work, family, hospitalization, to avoid hospitalization, to limit the visit to the clinic, etc. And finally, mortality is probably at the end. So during the past 20 years, we did a lot of progress in terms of improvement in survival, and this was mainly due to the increased use of combination therapy. And today, when we have the last data from the French registry, the 1-year survival is about 90%, 3-year survival is 73%. We can do better, of course. I think to define the treatment goals, we first have to know the prognostic indicators. And for the prognostic indicators, we have the clinical signs like symptoms, signs of right heart failure, functional class, etc. We have exercise capacity assessment. We have the assessment of the right ventricular function with right heart cath, MRIs, echo. And finally, we also have biomarkers. And with all those variables, we constructed some equations, some scores to predict outcomes. And these are mainly derived from PH registry data. We don't have any trial to do that. So the first was the NIH equation, registry equation in the 80s, and the last one was the REVEAL and the ERS risk stratification table. What do we want to do with this risk equation? We would like first to predict outcomes. We would like to predict survival. We would like to predict the event-free survival, clinical worsening. Number two, which is the most important thing for me, is to define the treatment goals and finally to guide management. Let's start with the REVEAL. You know that the REVEAL the Reveal score, Reveal 2.0, and now the Reveal Lite and Reveal 2.0 Lite. They are derived from the Reveal registry, which is a US registry with a development cohort of more than 2,700 patients and a validation cohort as well. And they did a Cox multivariable model, and from the coefficients from this model, they create a calculator that can predict both survival and clinical worsening. And as you can see here, this is the data published by Ray Benza. He showed that to a score of between 0 and 6 is associated with a low risk. And we can maybe consider a score between 0 and 6 as a potential treatment goal. Every other scores are not acceptable, and to have a score of 7, 8, 9, and more is not, of course, acceptable. And we have an excellent granularity, as you can see on this survival curves, according to the score. However, you see that on the left-hand part of the slide, it's quite complicated to calculate the risk, and you need a lot of variables to calculate the Reveal score. So let's move to the risk stratification table from the guidelines, and you have this famous traffic light table with the green, yellow, and red, and the ideal, of course, is to be at low risk. And it is a very important statement in the guidelines to achieve and maintain a low-risk profile, and it is recommended as an adequate treatment response for patients with PAH with a very strong class of recommendation, 1, but a level of evidence of C, because we don't have any trial to confirm this. And so the ideal is to fulfill all or part of the criteria in the green column. This is very important, and this plays a prominent role in the newly proposed treatment algorithm from the World Symposium of pH. And as you can see, the risk is, from the beginning, we have to assess the risk to decide what kind of treatment we have to give to our patients. But more importantly, is to reassess the risk during follow-up. And the only acceptable option is to be at low risk. And this is for the most important treatment goal we have to achieve. How to define a low-risk profile? And I would like to show you what we did in the French registry. In the French registry, we decided to look at the number of low-risk variables present at baseline, or more importantly, achieved at follow-up. And we chose 4 low-risk criteria. Functional class 1, 2, 6-minute walk more than 440, right arterial pressure less than 8, cardiac index more than 2.5. In an additional analysis, we also add the criteria on BNP or NT-proBNP. And why we chose those 4 variables? Because we had all the data available at baseline and at first follow-up in more than 1,000 patients. And we found that in univariable analysis that at baseline we found that all those variables are associated with outcome, and also at first re-evaluation, but more importantly in a multivariable analysis, at baseline we only had the 6-minute walk distance, but at follow-up, functional class, walk distance, right arterial pressure, and cardiac index are prognostic indicators, and they were independent. That is why at baseline we have a quite good discrimination in between the different curves. This is the survival according to the number of low-risk criteria present at baseline between 0, 1, 2, 3, or 4 criteria. But this was more granular at follow-up, and we have a much better discrimination at follow-up. And for patients achieving 3 or 4 low-risk criteria, the survival was quite good, but of course, 4 criteria is better than 3, that is better than 2, 1, and of course, 0. It doesn't mean that to have 0, it is high risk, because many people misinterpret this. This method was valuable only for the determination of the treatment goals and to look at the low-risk patient, not to look at the intermediate or high risk. And in many papers comparing our method to other, there is a misinterpretation of that. In additional analysis, we put 6-minute walk distance. Not 6-minute distance, NT-proBNP criteria or BNP criteria, sorry. And when we add this criteria, the 2 hemodynamic criteria went out. They were no longer independently associated with outcomes. And with just functional class, 6-minute walk distance, and the BNP or NT-proBNP dosage, we were able to determine what is probably the best associated with outcome. And as you can see, for patients fulfilling these 3 criteria, it's very, very simple. That's outpatient clinic. Functional class 1, 2, walk distance more than 440, and a normal BNP or NT-proBNP. You don't need something else. You don't need, if you fulfill these 3 criteria, you don't need echo, you don't need MRI, you don't need right heart catheterization to know that the patient is at goal. I think it's very important, and this has been also validated in the Compera registry published by Marius Hooper. Recently, we had the result of the EFFORT prospective study. EFFORT prospective study was a study, it was a prospective study, and we look at the low-risk criteria achieved not only at the first follow-up, but regularly during the 3-year follow-up. And we found very, very similar results with functional class 1, 2, a walk distance not 440, but 360 meters. The cardiac index was 2.4, exactly the same, and BNP was a bit higher than in the guidelines. But it was very interesting because when a patient achieved at least these 3 treatment goals, not only at first follow-up, but at any time during follow-up, this decreased the instantaneous risk of death or lung transplantation. So now how to improve restratification? First of all, we have to maybe to better discriminate the intermediate risk because too many patients are at intermediate risk. And that was what it was being proposed by the Compera investigators, and they change a bit the threshold for the BNP, NT-proBNP for the class, for the higher risk patients, but for they consider to split the intermediate risk group in 2 groups. One, according to the 6-minute walk distance, and another criteria according to the BNP or NT-proBNP level. And they found that what they call intermediate low risk was similar to low risk at baseline, but we have a good discrimination between intermediate high and high risk. But at follow-up, we have an excellent discrimination between patient at low risk, the same definition that we had previously, patient at intermediate low, intermediate high, or high risk. And we validate this in the French registry, and the 2 papers have been published on the same issue of the European Respiratory Journal. Let's move to the assessment of right ventricular function. Do we need to assess RV function to improve restratification? We showed that when we consider additional hemodynamic variables such as the stroke volume index, we can discriminate prognosis in patients achieving 3 or 4 low-risk treatment goals. And this is very interesting. And we found a threshold of 38 milliliter per square meter for the stroke volume index for patients achieving a higher level of 38. They have a much better survival than for patients who did not. But as you can see here, those patients were quite good because they achieve 3 or 4 low-risk criteria. So we can discriminate again, and we can select a subset of patients with very, very good long-term outcomes. What about echo? This is the main question I have at each talk. What about echo? We know that many echo variables that are associated with outcomes, TAPSE, tricuspid regurgitation severity, the size of the inferior vena cava, but also the ratio of TAPSE over SPAP. So we know that we can discriminate in between according to these different echo variables. However, which is more important is to look at what happened in patients who are already considered at low risk. Can we discriminate more? And that is the main question, and we don't have any answer for that. It's exactly the same for MRI. For MRI, we know that the right ventricular end systolic index and right ventricular ejection fraction can discriminate patients at low risk or intermediate or high risk, especially for the right ventricular ejection fraction, and also in particular at follow-up. And this could be interesting. The problem is the accessibility of MRI, because it's quite difficult to do MRI in all patients. For example, in my center, we see, I would say, 70 patients a week. It's impossible to do 70 MRI a week. It's impossible. So that's why we need a more simple methods. Biomarker could be very interesting, and we know that the ideal prognostic biomarkers is to look at not only the right ventricle, because, for example, NT-proBNP, we just look at the right ventricle, which is a consequence of the PH, but maybe to look at what happened in the pulmonary arteries, inflammation, endothelial dysfunction, remodeling. Do we have biomarkers for that? And we also have biomarkers for consequence on other organs like kidney or liver. So the biomarker should be a circulatory biomarker. It should predict clinical worsening. It could give an objective evaluation. It is easy to quantify, and it could be cheap. There are some studies on cytokines, for example. In cytokines, it seems that some cytokines may be useful to look at the survival. I don't want to go into too much details. We also have an interesting work from the Imperial College in London by Chris Rhodes, showing that using proteomics, they can also look at the survival, and according to the number of proteins they found in the blood of patients, they can determine the risk and the survival. Here we have between 0 and 3 here, 4 to 6 here, 7 and 9 here. This is very interesting work. And finally, quality of life questionnaire. We have to focus on the patient, and quality of life questionnaire, we have some generic life, SF36, or the CAM4, which is more dedicated for pulmonary hypertension. However, none of this measure has been used to support a labeling claim. It is exactly the same for PEH, SYMPACT, or the emphasis 10. However, the emphasis 10 is for me very interesting. It has been developed in the UK, and the emphasis 10 questionnaire is just 10 questions, but it correlates with functional class, with exercise capacity, with hemodynamics, and it is an independent prognostic marker. I think emphasis 10 should be tested in future risk stratification studies. So, but maybe the machine will decide for us, and this is what has been published by the group of Ray Benza, showing with the Bayesian network derived risk prediction model. It is called FORA, using many data from a patient. The machine can determine what is the risk for an individual. So, to conclude, so for pH risk stratification, we have a lot of information: clinical, exercise capacity, RV function, biomarkers, and this is very important to look at that for the choice of initial therapy. However, there are some variables of interest, like 6-minute walk distance, functional class, right heart cath data, anti-BNP or anti-pro-BNP levels, and you can simplify. And to simplify, functional class, walk test, biomarkers are very important because it's everyday practice and you can do it easily in our patient. And finally, future treatment goals perhaps they may include novel prognostic biomarkers and also quality of life, in other words, patients' perspective. So thank you for your attention.
Thank you, Prof. Sitpon. You can join us here. Thank you for sticking to the time, please. We'll keep the question at the end of the session. Now it's my great pleasure to introduce the next speaker. The next speaker is Prof. Jean-Luc Vechery. He is a professor of clinical cardiology, director of pulmonary vascular disease and heart failure clinic in Hospital Erasmus, Bruxelles, Belgium. He's going to talk about severe combined pre-capillary, post-capillary pH, a distinct phenotype, treatment update. The floor is yours.
Thank you very much. Salam alaikum. Thank you again I want to echo what Olivier just said about being so glad to be here with you today. It's been so long, and I hope that this pandemic will go away and that we can continue and keep on the friendship we have since quite a long time. So my task today is to discuss the entity that is probably the most complicated for us, which is the situation of pulmonary hypertension that complicates left heart disease. And I will give you a couple of updates on the presentation, but also on the treatment that we have. So please take some attention to check my disclosures with many companies who have a compound for the treatment of pulmonary hypertension. So the overview, and it's of course very complicated to cover everything in one talk, so I decided to focus mainly on the situation of heart failure with preserved ejection fraction. That is one of the leading causes of pulmonary hypertension. But of course, I will mention also other specific situations, probably because this is the most challenging situation where we have to make the differential diagnosis with idiopathic pH, and sometimes it's a bit complicated. So 2 parts. First part is going to be dedicated to mechanism, clinical phenotypes, and how much there might be an overlap with pH, especially idiopathic pH. And then I will show you what we know in terms of treatment of pulmonary hypertension complicating the left heart disease. So if we start to look at the mechanism of heart failure with preserved ejection fraction, there's a very important shift in paradigm towards something that would be more mechanistical or more pathobiological instead of more traditionally hemodynamic. And as you can see here, heart failure with preserved ejection fraction results in is the consequence of a cascade of events, starting with precipitators, comorbidities that are very well known, disturbed physical and chemical environment, involving mainly endothelial cell activation and dysfunction, which leads then to the disease affecting not only the cardiomyocytes, but also outside of the heart, the rest of the body, and leading to renal damage, etc., etc. All these mechanisms, interestingly, are quite independent from the level of ejection fraction, and all these mechanisms can also be seen in patients with PAH. In fact, endothelial cell dysfunction lays the bed of pulmonary hypertension. The consequence of these pathobiological changes are clinical expression by various phenotypes of heart failure with preserved ejection fraction. And there is an evolving concept that moves away from the traditional one, which is hemodynamic, which suggests that there's a transition from diastolic to systolic dysfunction towards what we know now. The current understanding is microvascular inflammation, direct cardiomyocyte injury, again, endothelial cell dysfunction, which is very different from the hemodynamic thinking process so far. And it's very likely that in the future, we will probably not consider left ventricular ejection fraction as a way to really describe what patients are suffering from. And from our perspective in the pulmonary hypertension world, I think it's going to make our life much easier, especially because we move on to the concept of phenotyping, where ejection fraction might only be one part of the jigsaw. Sanjeev Shah and other colleagues proposed this roadmap, the multi-organ roadmap, describing 3 distinct clinical phenotypes. And again, this might change in the future in heart failure with preserved ejection fraction. Type 1 would be the exercise-induced increase in left atrial pressure. Type 2 would be the volume overload with high blood pressure. And type 3 is the one that is interesting to us, pulmonary hypertension and right ventricular failure. It's very important to understand that in the heart failure world now, pulmonary hypertension is recognized as being a specific entity that requires a lot of attention. Type 1, typically younger patients, moderate diastolic dysfunction, near normal NT-proBNP, so we tend to see those patients quite a few times. One of the most common is the type 2, volume overload, high blood pressure, typically obese diabetic patients, high prevalence of obstructive sleep apnea, and those have probably the worst left ventricular relaxation. And the type 3 looks at older patients with significant chronic kidney disease, pulmonary hypertension, and RV dysfunction. So as you can see here already, we have those phenotypes that are important to understand when we deal with heart failure with preserved ejection fraction, and especially the type 3 one is important for us because, again, it's PH and RV failure. Now, if you go deeper in the understanding of the histology of the pulmonary circulation in those patients who have PH associated with HFpEF, I really like this important study by Pfizer and co-workers where they retrieved the collected samples from autopsy or from transplant or cardiac surgery with the idea to define where was the site of obstruction in the pulmonary vessels, starting from the arteriolae to the venous, the venulae. So really browsing the whole pulmonary vascular bed and trying to find out what was the site of obstruction. For that purpose, they compared control patients at the top, patients with pulmonary hypertension due to HFpEF, and patients with PVOD at the bottom. And you have from your left to the right the central pulmonary artery, the venulae, and the small arterioles at the right hand side. What you can see is that patients with PH due to HFpEF have arterial damage. They have medial hypertrophy, they have venular involvement, but the small vessels, as you can see here, are also affected by a disease process. And the bottom are patients with PVOD who have indeed more remodeling of the pulmonary arteries, but also have remodeling from small vessels. And in fact, when you compare this to the hemodynamic presentations, patients with PH due to HFpEF have a lot in common with patients with PVOD in terms of histological changes, and that might explain why some of our patients with HFpEF do develop significant pulmonary hypertension, severe pulmonary hypertension, or CPCPH as it is currently defined, and it's the small intravascular intimal bed that is really affected by the disease process. There's also some hints that we were talking about genetics. The discussion, the heated discussion this morning was very interesting, and indeed there are some genetic predisposition, very likely. This is an animal model work done by Fabrice Santini and David Montagny in their animal lab, where they were able to demonstrate that KCNK3 is dysfunctional in animal model of left ventricular pressure overload. So there are those abnormalities that we find in PAH also that are present in patients with PH due to HFpEF. Now, if we move a little bit away from the histology and look at the patient itself, this is a slide summarizing or showing you the heterogeneity of the HFpEF syndrome, which obviously has more comorbidities when pulmonary hypertension developed than patients with PH, and you can see all ventricular dysfunction, and I would say the traditional cardiology thinking of abnormalities that are found in patients with PH due to HFpEF, and in green, all the associated conditions, all the comorbidities for which the burden is very high in the situation of PH due to HFpEF, which is clearly a contrast with IPH. Of course, you can have iron deficiency in patients with idiopathic PH, but it's unlikely that you may have atrial fibrillation, for example. Obesity may be present. Sleep apnea is very common in PH due to HFpEF. It's much less common in patients with IPH. So even if there's a pathobiological rationale or common grounds between the two conditions, they are different in their clinical expression. And this is shown here in this very nice slide in the supplement of the European Heart Journal. Irene Lang and Massimiliano Palazzini were the authors of this paper, where you can see the pyramid of the burden of comorbidities and in the context of pulmonary hypertension. And clearly the burden is much higher in group 2 PH as opposed to group 1, although there might be some overlap. So again, having a look at comorbidities can already give us a hint about the severity of the disease when pulmonary hypertension develops in HFpEF, and that might serve also as a way to make the distinction from a differential diagnostic point of view. Now let's move on to treatment. Of course, what you've seen, you may think, and it's right to think, that because you have common grounds between PAH and PH due to HFpEF or PH due to left heart disease, the thinking might be that we should treat or we may treat pulmonary hypertension, and as you know, the situation we have today is not really going in that direction. One of the very important difference between PH-HFpEF or PH in left heart disease in general and PAH is that the streamline or the treatment algorithm starts with the treatment of the underlying condition. This is absolutely critical. Even before assessing pulmonary hypertension in our patients with left heart disease, we need to be sure that they receive the most appropriate treatment. And I'm not talking about the role of pulmonary hypertension in prognosis, because of course it plays a role, whether it's HFrEF, reduced ejection fraction, preserved ejection fraction, or valvular heart disease. And we have standard treatments for all these conditions. The next speaker will talk about the groundbreaking empagliflozin treatment for HFpEF, which is the first treatment we have for this condition, but for the others, we have very well-established treatments. Then the second step would be to try to identify other causes leading to pulmonary hypertension, and as you know, in patients with heart disease, sleep apnea syndrome is common. It's unlikely to give you a lot of pulmonary hypertension, but you may find pulmonary embolism. You may find COPD, and you may even find pulmonary fibrosis. So looking at these conditions that may aggravate or contribute to the development of pulmonary hypertension is extremely important. And then we have additional therapies and intervention. We call them advanced therapies. For HFrEF, for example, of course we have transplant, but we have also the implantation of left ventricular assist device, which is a very nice way to treat pulmonary hypertension in patients with HFrEF. There are very, very few patients with HFrEF that stay with significant pulmonary hypertension that would preclude heart transplantation now that we're implanting left ventricular assist devices. HFpEF is a bit more complicated. We have interventional cardiologists know very well that we have this interatrial device to probably more specific to the type 1, the exercise-induced pulmonary hypertension or increase in pressure. I will show you a very brief slide suggesting that the interatrial device may work from a hemodynamic point of view, but doesn't really work to decrease the rate of events. And finally, patients with valvular heart disease can have also some specific surgical treatment, and you have, of course, this overlap between HFrEF and valvular heart disease, where you can use MitraClip to reduce the severity of mitral insufficiency in patients with heart failure. So these are very advanced therapies. Now, if we go back to HFpEF, this is the last treatment. These are the last treatment recommendations for HFpEF according to the European Society of Cardiology. And as you can see, the highest level of recommendation with a low level of evidence supports the use of diuretics. Diuretics for treatment of HFpEF. That's the most established treatment and the most recommended treatment. Of course, we have now empagliflozin. The publication was too late after the guidelines were released to be implemented, but it will likely be implemented. I'm not going to talk a lot about that. What is interesting is that each time we have tried to use the PAH pathways to treat heart failure with preserved ejection fraction, without pulmonary hypertension or with pulmonary hypertension, we do not know that from the trials. Well, actually, we have failed. And you can see here cytoskeleton, an endothelin receptor antagonist, sildenafil, and vericiguat in 2 trials that did not demonstrate an improvement in the primary endpoint, whichever was the primary endpoint for the treatment of heart failure. There's a lot of meta-analysis that were done that do not support the use of these compounds to treat heart failure with preserved ejection fraction. So even if you have a good rationale, a good pathophysiological rationale, the studies must be done, because sometimes you have very unpleasant surprises that are shown here on this slide. Now, if we move back or move forward to the pulmonary circulation, and we ask ourselves, do our current treatment for PAH work in the context of pulmonary hypertension, specifically due to HFpEF, but also in valvular heart diseases? You can see here a list of trials with only 2 randomized controlled trials, small ones, monocentric, being positive on the primary endpoint, that is an improvement in PVR. We have neutral trials, but more worrisome, we have now 3 randomized controlled trials, well conducted, that demonstrate that the use of sildenafil in patients with valvular heart disease, corrected valvular heart disease, may be harmful, that treatment with bosentan and macitentan in the context of patients with heart failure is associated with an increased risk of side effects. So based on this, it is very difficult to provide a positive recommendation on the use of pH therapies in the context of CPC-PH. Meta-analysis or reviews are also suggesting the same, even for a class of drug like PDE5 inhibitors that we would believe are pretty much harmful. And as cardiologists, we know how to use them for years. We have patients with erectile dysfunction that are asking for those treatments, and we know that they are quite safe in the context of left heart diseases. However, the reviews and meta-analysis do not support the use, even if you put all the studies together, and it's very important to see that the authors themselves caution rather against the use and acknowledge the fact that we are lacking properly conducted randomized controlled trials. Even if you look in the community off-label use, and this is coming from the veteran registry in the US, if you start using PDE5 inhibitors, likely over-the-counter, in patients who didn't have necessarily a right heart cath, I think it was only 2/3 of the patient had no right heart cath before being treated. So those patients received sildenafil based on an echocardiography, whether they had group 2 or group 3 pulmonary hypertension. You see that the outcome doesn't seem to be that positive in this context. So even the off-label use doesn't seem to be improving patients so well. Now, what's the plan for tomorrow? Because what I've given you is today. What is going to happen tomorrow? Well, tomorrow tomorrow is already today, and I'm going to show you a couple of studies that have been completed or are underway. Those 2 studies, Saph and Serenade, were closed before completion. Saph was oral treprostinil in patients with HFpEF, and Serenade was macitentan in the same condition. One had change of 6-minute walking distance versus baseline after 24 weeks for Saph, and Serenade had % change from baseline in NT-proBNP NT-proBNP at week 24. It's surprising that this study was conducted when the MELODY trial looking at PH due to HFpEF was a trial suggesting a treatment intolerance associated with the use of the ERA. South Pah was stopped by the sponsor after 84 inclusion when 310 were predicted, slow enrollment, but also there was a high rate of drop-off in both placebo and treated group, which is drawing our attention to the fact that patients who are receiving prostacyclins in this context may do not fare very well. And Serenade was also stopped after sponsor decision after 143 inclusions, so roughly halfway from the trial. Some of these results, at least for South Pah, are on clinicaltrial.gov, but they are not interpretable since the study has been stopped way before the complete inclusion. So it tells you that it's not that easy to conduct clinical trials in this context. There are two studies that are completed and awaiting publication. One is Soprano with macitentan as the study drug in patients who had persistent pulmonary hypertension after left ventricular assist device implantation. The primary endpoint was a change in PVR at week 12 versus baseline. And the other study was the dynamic study using oral risiguat at 1.5 milligrams TID, 26 weeks. The primary endpoint was a bit curious, but it's the change in cardiac output, very likely based on the left trial in heart failure with reduced ejection fraction, suggesting that there was an increase in cardiac output. Only the Soprano study, to the best of my knowledge, has been presented. It was presented at the ISHLT annual meeting in 2021. Again, this study was stopped early after 57 inclusion out of the 78 that were predicted because of slow inclusion rate. There was indeed a decrease of 0.74 Wood units in pulmonary vascular resistance with a p-value, but there was no effect on the secondary endpoint, and this study awaits publication as well as the dynamic trial. There are three ongoing. One is Passion, oral tadalafil, sponsored and championed by our friends Stefan Rosenkrantz and Marius Hooper. This is a very important one. This is probably the best design, one of the best design study by investigators and not company, and we will see what comes up with this trial. The primary endpoint is time to first event. The HELP trial with intermittent administration with levosimendan. This is a pilot study. Of course, it's intravenous administration. The primary endpoint is a change from baseline in wedge pressure with bicycle exercise. And the last but not the least probably is the Cadence trial with sotatercept, where patients are going to be randomized to placebo, sotatercept 0.3 and sotatercept 0.7 milligrams subcutaneously, 180 patients. Really PH HFpEF in this case, and the primary endpoint is change from baseline in pulmonary vascular resistance. We will see if we can do better with this last one as we did before. But again, we are waiting with a lot of interest and excitement the results of the Passion trial. Now, if we go back to this phenotyping roadmap that was proposed by Sanjeev Shah, and if we look at the three different phenotypes, what we can see is, unfortunately, in red are the trials that didn't show an improvement in the primary endpoint. So type 1, we have nothing. The reduced lap heart failure, which is the intra-atrial device, was published very recently and didn't show an improvement in any of the clinical outcomes that were decided for this trial. Panache with an adenosine 1 agonist and oral nitrates or inhaled nitrate didn't work either. Type 2, we'll hear about Emperor Preserved, very important study, but the two others, Paragon and Vitality, were negative or neutral. And type 3, South Pah and Serenade were halted. We're waiting for HELP, Cadence, and of course, I forgot to mention Passion on this slide. Now, of course, designing randomized controlled trials for left heart disease is challenging. There are key learnings and three important ones. The first one is to define the candidate criterion, and very likely having an ejection fraction above 50% is something that is important. We need to find out those patients who have the most right ventricular ventricular involvement. That's the type 3 phenotype. We need to have an accuracy of diagnosis for consistent phenotyping. We need to have clinical heart failure diagnosis. We need to invest in hemodynamics. There's no way we're going to do trials in pulmonary hypertension without a right heart cath. We can address that with cardiopulmonary exercise testing. And then finally, we need to improve the RCT design by choosing an appropriate endpoint. For phase 2, it should be safety tolerance. We need efficacy signals, can be given by changes in PVR or CPET variables. And then only if the phase 2 is positive, we can move on to phase 3 to have real clinical events trials, because this is the way we do it in the field of heart failure. There's no reason why it would be different in patients with PH due to HFpEF or PH due to valvular heart disease. Now, take-home messages. Clearly, in contrast with PH, the treatment of CPC-PH or severe pulmonary hypertension should focus on the underlying condition. This is very important and, again, very different from what you see in PH. The randomized controlled trials that are conducted so far do not support the use of PH-approved therapy or the use of targeted PH pathways in the context of heart failure. Probably that the future and deep phenotyping will help us to better understand this complex syndrome, but at this stage, the main message is that patients with severe CPC-PH should not receive PH-approved therapies. Thank you very much.
Thank you very much, Prof. Vacheri. Thank you. As usual, it's a very comprehensive lecture. You can join us, Prof. Now it's my great pleasure to introduce my co-chair, who's Dr. Abdullah Al-Khudair. He is a clinical interventional cardiologist and pulmonary hypertension, and he is the director of adult cardiology program in King Fahd Medical City. He's going to talk about very interesting and hot topics with cardiologists, that is SGLT2 inhibitors as a new treatment for HFpEF. Dr. Abdullah.
Thank you very much. Assalamu alaikum. Thanks, everyone, and thanks for the organizing committee for inviting me. Thanks for Dr. Vacheri for laying the background for my talk. So I'll start to just answer the question is, what is HFpEF? And this has been answered in the prior talk, but basically it's a syndrome where there is symptoms and signs of heart failure. By essence, the LV function is preserved, and we can talk about that. What is the preserved ejection fraction? Is it EF more than 40% or EF more than 50? Or that's a matter of debate, because there's now a term called HF or HF mid-range reduced ejection fraction. Usually there are echo features of diastolic dysfunction. The BNPs are usually elevated, and if we do hemodynamics or invasive assessment, the filling pressures are elevated as well. So as we saw from the previous talk, HFpEF is very common. The traditional teaching is that it accounts for about 50% of all heart failure population. As we saw in the previous talk, it is associated with increased morbidity and mortality. The pathophysiology is quite complex. Some people say that in contrast to HFrEF, where the problem is in the intrinsic myocardium, in HFpEF, the problem probably starts outside the myocardium with hypertension, aging, renal failure, and all of these lead to myocardial hypertrophy and fibrosis, decreased diastolic compliance and relaxation, then subclinical systolic dysfunction, and also renal dysfunction with all of its devastating side effects. We saw that very nicely in the previous talk. HFpEF is heterogeneous condition. There is the advanced age HFpEF. There is the HFpEF with mostly PH. There's the HFpEF with volume overload and obesity. And of course, diabetes, OSA, CKD always play a factor in all of these different phenotypes. All of these issues, they will activate the pro-inflammatory pathways leading to fibrosis and increased collagen deposition. Interestingly, if you compare HFpEF to HFrEF, the left atrium and the hemodynamic causing pulmonary hypertension is quite different. And the left atrium remodels differently between HFpEF and HFrEF. This may be a target when we test therapies for either conditions. Dr. Bashiri nicely put out this table here, when to suspect HFpEF, what is the pretest probability, and we can see different variables here that you have to take into account: age, risk factors, presence or absence of atrial fibrillation, ECG findings, echo findings, and finally cardiac MRI findings. And you can come up with a pretest probability to decide whether or not your patient has the condition of interest. Pulmonary hypertension and HFpEF is quite common. Some studies said it's up to 80% of their population had some degree of pulmonary hypertension. However, I have to say this is a very difficult to estimate because of there's no uniform use of hemodynamics in HFpEF studies. The cutoff of pulmonary hypertension is different between different studies. As we said earlier, HFpEF is a very heterogeneous group of patients, and of course there is the issue of referral bias. What we know, either in group 2 or group 3, the presence of pulmonary hypertension to these cardiac condition makes the prognosis worse. That's a fact, and I think this has been shown over and over. Now, because this is a devastating condition with lots of comorbidities, people have tried different agents, especially the ones that have shown benefits in patients with heart failure and reduced ejection fraction. So we can see here to the top right, irbesartan was tried in the I-preserve, candesartan was tried in the CHARM preserved in the bottom left, perindopril was tried, vericiguat was tried. Unfortunately, in all of these trials in patients with HFpEF, the primary endpoint was not met. I'm not going to spend time to talk about these, but I've elected to talk about the TOPCAT and the Paragon heart failure trials before going into the SGLT2 inhibitor. So the TOPCAT is a very controversial study, assessed spironolactone in patients with heart failure and preserved ejection fraction. And their definition was EF of more than 45%. The issue with this trial is it's a multinational trial and involved mostly North America and also Eastern Europe populations. And as you can see at the bottom here, one of the inclusion criteria was that either you have high BNP levels or you have a history of hospitalization in the previous 12 months. And unfortunately, these hospitalizations were not adjudicated, and that created a lot of some problems with this trial. So basically what it showed is that the primary endpoint, which is a composite of death, cardiac arrest, aborted cardiac arrest, or hospitalization, was not met. However, if we look at the secondary outcomes, hospitalization was also not significant across the line of unity. The same was for mortality from any cause, myocardial infarction, and stroke. So overall, it was a negative trial. However, when we look into the details and when they did a sub-analysis based on geographical variation, they noticed this finding. So patients who were enrolled from the Americas, North or Latin Americas, they had benefit of spironolactone, while those who were enrolled from Eastern Europe, Russia and Republic of Georgia, they did not have this benefit. And the explanation, or the possible explanation for this finding, was that most likely the thresholds for admitting heart failure patients was different between the two continents, and hence the difference in outcomes. So overall, this study was considered to be a negative. However, if you really wanted it to be positive, then you have to specifically mention that it was positive in heart failure admission reduction in patients from the Americas, North and South Americas. Going next to the other important study in this field, which is the Paragon, which built into the impressive findings of the Paradigm heart failure trial, which tested sacubitril neprilysin inhibitors in patients with HFpEF. So the Paradigm heart failure trial tested this agent in patients with HFrEF and showed impressive results, reduction in primary endpoint. So the natural progression was to try this into HFpEF population. So we can see the inclusion criteria. age more than 50, signs and symptoms of heart failure, EF more than 45%, elevated BNPs, evidence of structural heart disease, so similar to other trials in this field. So they excluded alternative diagnoses or probable alternative diagnoses, such as significant pulmonary disease, and they said including primary pulmonary hypertension, although I think we've moved away from using this term. Nonetheless, the primary endpoint which is total hospitalization and death, was not met. It crossed the line of unity. If we look at hospitalization alone for heart failure, maybe it almost touched the one there. So you can argue whether or not this is a positive finding, but the primary endpoint was not met in this trial. Same thing here if we look at the changes in quality of life, summary score in the Kansas City Cardiomyopathy Quality of Life Questionnaire, it was not significant. Again, this is the same thing, showing the overall finding. Composite endpoint was not positive. Total hospitalization, maybe. Death from cardiovascular causes, clearly not positive. If we look at subgroup analysis, we can come up with several observations. One is patients who were younger or who were older, they showed some benefits with sacubitril valsartan. Females also showed the same thing. And quite interestingly, if we stratify based on EF, those patients who had lower EF, they showed some benefit as opposed to those who have higher degree of ejection fraction. All of these are hypothesis-generating observations. Now, moving to the main topic or main talk of this main point of this talk, which is the sodium glucose transporter 2 inhibitors. So, what are those? Basically, those are derivatives from the original compound, which is fluorescein. This has been extracted and known for over 150 years. So, if you Google it, you find that actually it's been there for quite a while. And people have known its hypoglycemic benefits. It's basically an extract of apple tree roots. And how did this compound found its way to the cardiology literature? So basically, I would say it's probably by chance. In 2008, the FDA mandated that any diabetic trial should have cardiovascular safety as one of its outcomes. So because we know that some of the diabetic agents have shown increased cardiovascular risk, and that was the reason for this mandate. This was in 2008. So when industry started to test this agent, they had to show cardiovascular benefits. And this was one of the first studies, the EMPA-REG outcome. And basically it says that in patients with type 2 diabetes, and those patients were high risk for cardiovascular events who received empagliflozin, actually they had lower rate of the primary composite CV outcomes. And of course, when you see this signal, you go and work on it. So this was tested in the HFrEF population in 2 major landmark studies, the DAPA-HF trial and also the EMPEROR-reduced trial. So this one was testing dapagliflozin, and this one was testing empagliflozin. And in both trials, impressive results. So patients with HFrEF, the risk of worsening of heart failure or death from cardiovascular causes was lower. Same thing in the empagliflozin trials. And this leads us to the EMPEROR-preserved trial, which tested empagliflozin, this time, however, in patients with preserved ejection fraction. So it is a randomized, double-blind, parallel group, placebo-controlled, event-driven trial. It included patients with NYHA class 2 to class 4 heart failure. EF has to be more than 40%, and patients had to have high BNPs. And if we look at table 1 here, it's quite a sick group of patients. So median age was 71. Most of the patients, 80% of the patients were NYHA class 2. And if we look at the EF assessment, a third of the patient had EF between 40 and 50, a third of the patient had EF of 50 and 60, and a third of them had EF more than 60%. And this is a nice distribution because I think it has some implication on how we interpret all of these trials. So what do we find? For the first time in HFpEF population, we have a primary endpoint positive. What is the primary endpoint? It is a composite of hospitalization of heart failure and cardiovascular death. Of course, this is driven mainly by hospitalization. There was, when looked at separately, death was not significant between the two groups. There was also an improvement in, or slowness in the degree or the decline in renal function with empagliflozin. However, the change in the quality of life measures was not significantly different between the two groups. But you can argue that hospitalization by itself is an important quality of life parameter. So, subgroup analysis. Look at this interesting finding here on the top. So, most of the benefit was in those patients who had EF less than 50%. If we look at EF more than that, we see that benefit is still there, but it's attenuated. So again, this begs the question of the different phenotypes of HFpEF and whether or not it's practical just to lump them all together and say they're all HFpEF, let's just give them one treatment and see what happens. I think they are quite heterogeneous, and we have to do more in terms of specifying which HFpEF population are we interested in. So in summary, the Emperor Preserved showed a 21% relative risk reduction in the primary endpoint. The secondary endpoint was also positive, whether that was heart failure hospitalization or the slope of decline in GFR. Safety overall was a safe medicine. The serious adverse events occurred similarly between the two arms. There were slightly more uncomplicated genital and UTIs and hypertension in a patient with empagliflozin, and very reassuring, the rates of DKA was very low. So if we put this study and compare it with other studies that tested different medications in HFpEF population, we can see that the primary endpoint was met for the first time in contrast to all of these other trials where the primary endpoint was not significant. If we compare the empagliflozin and the HFpEF and the HFrEF population, we can see quite similar outcomes there. So the rates of cardiovascular death or hospitalization was similar. Same thing for the time to first heart failure or cardiovascular death, and same thing for the total hospitalization. So quite similar effect across different degrees of ejection fraction. Now, this study looked at a 12-week of dapagliflozin and its effect on patient-reported outcomes in patients with HFpEF. And what they saw was impressive. So there's an impressive improvement in the quality of score or quality of life questionnaire. And to the right, you can see also an impressive improvement in 6-minute walk test, about 20-meter difference. I looked at some of the PAH trials and see the difference in 20 meters, and this is considered to be a significant difference improvement. In the top cat, the spironolactone resulted in only 1.5-point improvement in this questionnaire. The Paragon, it only 1-point improvement, and the Emperor HF resulted in 1.3, as opposed to 5-point improvement in SGLT2 inhibitors. This has been attempted again to show the same findings with empagliflozin. So remember, this one was dapagliflozin that showed improvement in patient-reported outcome. But when we tested it in empagliflozin in the Imperial Preserved trial, the two arms did not have any significant difference in terms of 6-minute walk test or the quality of life questionnaires. So why is the discrepancy between these different agents? So probably one is related to the different baseline characteristics of the study participants, because different trials have different patient population, different degrees of sickness in those patients, and probably that's why we saw different outcomes when it comes to patient-reported outcomes. It is unlikely to be related to the difference between EMPA versus DAPA, as this is probably a class effect rather than a specific effect to the medication itself. We're awaiting the DELIVER trial, which is, again, it's going to be the sister trial of Emperor Preserved, however, this time with dapagliflozin. It's a large trial up to 6,000 patients, and patients with preserved and mildly reduced ejection fraction. We cannot talk about EMPA or SGLT2 inhibitors in PH conference without mentioning this study, which basically showed that empagliflozin resulted in improved PI pressure, mostly diastolic PI pressure, in those patients who had prior CardioMEMS pressure sensor. So the CardioMEMS is this device in the top right here, has some indication in patients with HFpEF, difficult to manage. And basically you can get an idea about the PA pressures, and you adjust your treatments accordingly. So they took those patients who have this device already, and they randomized them into either EMPA versus placebo, and they found that those patients who had EMPA, they have improvement in the diastolic PA pressure, which was the primary endpoint. The mean PA pressure, the systolic PA pressure also followed the same trend. With improvement in those hemodynamics in patients with empagliflozin. So now the question is, what is the proposed mechanism of all of these benefits of SGLT2 inhibitors? And I think I tried to search for one single answer, but I don't think we have any. We have multiple proposed mechanisms. Is it the glucose or the hypoglycemia benefit? Is it the oxidative stress reduction benefit? Is it the weight loss reduction or weight loss benefit? Is it reduction in inflammation? So is it all of these different benefits? And I think that's probably the right answer. It's multiple different mechanisms. We don't exactly know which one is the predominant, but theoretically there's multiple different ways to think why SGLT2 inhibitors can be beneficial in those population. So in summary, HFpEF is a very heterogeneous entity with a high degree of comorbid conditions and difficult to control symptoms. In patients with HFpEF, for the first time, SGLT2 inhibitors has a clear benefit in reducing heart failure-related hospitalization with possible improvement in other patient-related outcomes such as 6-minute walk test, quality of life, as well as possible hemodynamic improvements. This benefit has been consistently independent of the diabetes status, so in patients with or without diabetes. So it is no longer an endocrinology drug. It is now probably more of a internal medicine / cardiology drug. A final finding that I saw across all of these HFpEF trials, the presence or absence or the severity of pulmonary hypertension as a subgroup analysis was never tested in any of these trials. And I'm probably would like to ask one of our experts here of why that is, because this could have given us a hint where to go if we stratified patients according to the presence or absence of hypertension, pulmonary hypertension. And with that, I'd like to conclude. Thank you so much.
Thank you very much, Dr. Abdullah. Thank you. You can join us. Just now, the session is open for discussion.
Assalamu alaikum. Thank you very much. Two questions for about HFpEF. Is there a role for beta blockers in the absence of AFib?
Sorry, I didn't mention that because it will take too long to just go over all the medications that have been tried in HFpEF. But in the absence of atrial fibrillation, angina, or reduced ejection fraction or any other arrhythmias, I don't think there's a specific role for beta blockers, unless you're invoking it as a secondary line agent for resistant hypertension, then maybe.
Another question for the SGL2 inhibitors. Why wasn't there an arm of augmented diuresis added to the trials? I mean, could it be the diuretic effect, especially that they actually worked even in non-diabetic patients? So maybe it's all the diuretic effect. Shouldn't there be an arm of augmented diuresis? Because these patients were, I think, optimized and then randomized to placebo versus the SGL2 inhibitor. But theoretically, many of them might not have been really optimized. didn't have the CardioMEMS, for example, measurements. So to tell if it's really diuretic effect or not, shouldn't we have an arm of augmented diuresis?
I'm not sure I understand the question correctly, but you're suggesting that should it be better to have an augmented diuresis before randomization? Is that the question?
No, I'm saying the SGL2 inhibitors might work only by having extra diuresis. And the patients who were randomized in the studies were optimized and then randomized. Theoretically, it's only the diuretic effect, especially that these medications work in non-diabetics. So maybe it's not the blood glucose effect.
Yeah, I mean, we've tried diuretics before and didn't work. Spironolactone works like it's a diuretic by essence. So and it didn't work. And it's hard to imagine that all of these benefits is just related to volume stab. We have Lasix for many years, and yet our patients don't do well. So again, I'm not entirely sure what is the exact mechanism of how these medications work, but it's probably it has to be more than just the diuretic effect. I'm not sure if Dr. Bashir, you have any other thing to say.
No, I totally agree with you. We don't know how this drug works. We need to admit that. Sometimes it's upsetting, but this is a fact. I do not think it's diuretic effect, because if it would have been the case, then why on the long term would you have such an effect? Diuresis, I mean, a diuretic would have an effect on symptoms, but unlikely on patient's outcome. It's very important to see that the main effect driving the positivity of Emperor Preserved was the reduction of hospitalization, which was quite significant. And there's another observation that is also common to heart failure with reduced ejection fraction, is that you have a decline in renal function. You have a decline in GFR during the first weeks of treatment. So there are many unknowns, but it's very unlikely that we can explain all the benefit just by the diuretic effects. As you mentioned, those patients were very neat and they were probably balanced in terms of fluid, so it should be something else. But we need to admit we don't know what this is.
Go ahead, Abdullah.
Thank you so much, all of you, for an outstanding talks. We truly enjoyed it. But my question to Olivier, redefining the intermediate risk group has been long waited. A patient who walks for 200 meters is not like the patient who walks for 350 meters, for instance. Same thing applies for lower levels of pro-BNP compared to high level pro-BNP, yet not reaching the high risk group. So the question to you, of course, this makes a lot of sense now. But would you think this has some implication now on the treatment or changing the guidelines to be more aggressive in patients who have high intermediate risk group, especially with IV prostacyclines?
Thank you, Abdullah. Very important question about the why we decide to have a better discrimination for intermediate risk is, of course, because the treatment decision could be different for intermediate high and intermediate low. So we test this approach for the initial therapy in our cohort we published in the Blood Journal, and on the role of triple combination as compared to double or mono. And when we look at the intermediate high, in the intermediate high risk, we have a much better survival in patient initiated with triple, and for the patient initiated with double is better than mono, which is not the case in the overall population. And in the intermediate low, maybe double combination therapy is not better than monotherapy initially. So probably we will have some recommendation different for patient at intermediate high or intermediate low, but this is not, maybe not for the initial therapy, but for the follow-up. And if we consider sequential combination therapy, it's probably not the same if the patient is at intermediate high or intermediate low risk. And this is very important. For example, if we consider the place of Selexipag, for example, as the third drug, if you start initial double combination therapy with an endothelin receptor antagonist and a PDE5 inhibitor, today it's difficult to know if you have to give Selexipag or to transition to riociguat or to give sub-Q or IV prostacyclin. And this will help us to make the right decision but it's difficult to say intermediate high is IV prostacyclin, intermediate low is Selexipag. It's difficult to say like that, and we have to keep in mind that we are a physician, we are physicians, the patient is the patient, and we have to consider the relationship in between and all the perspective of the patient, the perspective of the physician.
Go ahead, please. Thank you to all the speakers. My question is regarding the obesity phenotype and pulmonary hypertension. Do you think it is the same issue about HFpEF and the response to SGLT2 inhibitors without like real diabetics or without the frank heart failure? I think there has been some issue about obesity per se and these drugs. Can we have some impressions about that?
Thank you very much. I will leave Abdullah answering on this part, but you're right. Obesity is one of the problems we have with those patients with HFpEF, with or without pulmonary hypertension. That said, if you look at the BMI of all the patients included in the trial, they are not that significantly high. So there might be a component that is associated with obesity, but the interaction analysis does not suggest that the BMI plays a role in the benefit in terms of a reduction of events, at least in the emperor preserved. I don't know if you agree with that. Yeah.
Thank you very much. Regarding the right ventricular function assessment, do we use 2D to assess ejection fraction, 3D to assess ejection fraction, global longitudinal strain for the right ventricular function, or using the shift of interventricular septum to the left?
So you want to answer, Olivier, the question about RV function? So I'm starting with I'm putting my cardiologist hat on. All these tools are very important to assess function. And you're right, 2D, 3D strain are very interesting to assess function. Now, the issue we have with 3D and perhaps strain is that it's highly vendor dependent. So we need to be aware of the differences between vendors in terms of interpretation. And also the question is, what would be the added value of RV dysfunction compared to more simple variables that are available worldwide? So that would be the main question. Cardiologists would use surely echocardiography, pulmonologists would use MR. Now there's a crosstalk between the two disciplines looking at imaging. So it is a very important tool, and we know that treatments can be, that RV function can be modified depending on the tool you're using or independent of the tool you're using with medical intervention. That said, how much does that tell us about the future of a patient? I do not think we're there yet. Olivier, I don't know if you want to add something. The pulmonologist's point of view.
No, I know that there are many tools and many variables to assess when you want to look at the RV function. The problem is, do we really need that for the patients who is at goal? I think we use it. We use echo, we use MRIs, not too much, but echo and right heart cath, but especially in patients who do not achieve the goals. If the patient achieves the goals, the clinical goals, that's fine. I think we don't need something else. But for the vast majority of them, they do not achieve those goals. And we need additional tests. And among those tests, we have, of course, the evaluation of RV function. But as a pulmonologist, when we have a result on an echo, we do not trust in that, and we confirm with right heart cath just to make the right treatment decision. That's our experience.
Just before a fight breaks out between the two of you, maybe I can intervene with a question. Jean-Luc, in the cath lab, we give fluid to bring out, unmask a HFpEF phenotype, but also we do the opposite. Well, we do, and I think you do as well. We administer isosorbide dinitrate boluses, and we can sometimes see the most amazing improvements in hemodynamics. I had a case last week of a patient, she actually had torrential mitral regurgitation, and her wedge went from 26 down to 11 with a couple of boluses of dinitrate. And her mean PA pressure went from, I can't remember, something like 40 down to 19. Now, when we've tried with other examples like this of then using these drugs chronically, it just hasn't translated into clinical benefit. And I don't understand why. Do you have any thoughts on that?
Thanks, Luke. I think we need another hour to tease that out because it's very complicated. Nobody knows why it is the case, but we all share the same experience. It's amazing to see how with whichever the nitrate you use, we use nitroprusside, and within minutes you see the pressures going down. You will see that for patients who have more the isolated post-capillary pulmonary hypertension phenotype, and the effect on systolic pressure, in fact, is even more amazing than the effect on the diastolic pressure. Perhaps that for nitrates, the reason is the tolerance. That is a very well-known phenomenon, and we see that with hydralazine as well. Also, perhaps because amongst the phenotypes, neither of us dare talking about it, but you have that sensitivity to vasodilators that is very diverse in the population of HFpEF. It's interesting, but I do not think we're there yet. But beyond the fact that there is indeed a pharmacological tolerance to these compounds that is very well known in the treatment of angina, for example. That would be my answer. And it's curious, in fact, that cyclic GMP stimulators or PDE5 inhibitors do not work better than what we've seen either in trials or even on clinical trials. So clearly, in IPCPH cases, PDE5 inhibitors do not work. They may be effective, although it needs to be demonstrated, in patients who have the most severe pulmonary vascular remodeling, and that population is very difficult to identify.
Just finally, if I may, you said exclude patients with AF. That's going to exclude a lot of patients, but why? Maybe I didn't pick up on why.
Well, the reason is that there are a couple of reasons for that. And again, it takes an additional hour to discuss this, and we can discuss that over lunch. But the reason is that those patients have, because of atrial fibrillation, more events that are completely independent from the underlying, they are not dependent from the underlying condition, but completely independent from pulmonary hypertension. That's the first. The second is that it has an impact. If you want to do a cath study, you know very well that when you're in a cath lab and when you have a patient in AF, it's a pain in the neck to have a proper wedge pressure analysis, and even if you do LVEDP, you may get fooled due to the presence of atrial fibrillation. So cath interpretation is difficult. And then lastly, we had a lot of discussions in the various meetings and steering committees, and we thought it would, because it's highly prevalent in this population, I think in Emperor Preserved it was 40% of patients who had AF, something like that. In Melody, we had 75% of patients with AF. And I think it contributed to the fact that the study was not positive. So the idea would rather be to accept a certain amount of patients with atrial fibrillation, but strengthen the criteria for pulmonary vascular disease in those studies. That, it's based on thinking, not really on hard science.
Thank you. Really brilliant session. Thanks very much.
Thank you.
All right, if there's no more questions, I think we should close session. I would like to thank Professor Pah and Professor. I'm my co-chair, Dr. Abdullah, and thank you for you all. And now it's time for prayer time and lunch.
Quick
and
announcement here.
Okay, Dr. Go ahead.
Okay. Now we will have about 20 minutes to relax and do refresh. Lunch will be just behind this door, behind this wall. It will open at 12:45, okay? And then we'll resume back here at 1:30. At that time, also there will be, in the same time, prostacyclin workshop upstairs. So we'll see you at lunch at 12:45. Thank you.
Have you thought they were?
Well, I think we'll get started. I'm Salih Dammas, and my co-chair is Dr. Hanan Fan, and we'll start the afternoon session. The first presentation will be by Professor Nazarini Galay, who's very well known, a world authority in pulmonary hypertension. He's a full professor of cardiology in Bologna, Italy. And the title of his presentation will be the initial triple oral combination therapy in PAH. Very sorry to keep you waiting, Professor. Unfortunately, it's prayer time and people are a little bit late. Thank you very much for being here today.
Thank you. You hear me? Okay, I share my presentation. You see my slides?
Yes, we do.
Thank you. Thanks for the invitation, and let's to start just not to lose time. And this is the distribution of the randomized control studies performed in pulmonary arterial hypertension with different treatment strategies. And this led to the approval of 12 drugs interacting with 3 pathways: endothelin pathway, nitric oxide pathway, and prostacyclin pathway. And those drugs have been utilized with different strategies: monotherapy, sequential combination, initial combination, and risk-oriented treatment strategy. Sequential combination has been validated by robust studies like the Seraphim study. This is the reduction of the event achieved by the combination of PDE5 inhibitors and macitentan. Also, triple combination therapy, sequential, has been tested in the largest study in PAH, the Griffon study, and the triple oral sequential combination also reduced by 37% the event as compared to the continuation of sequential double. So we have this demonstration that the sequential oral, triple oral is effective. In addition, we can show efficacy also in patients mildly symptomatic. And the triple combination reduces the events. What does it mean? It means that only appropriate functional class may be not sufficient to analyze, to evaluate the effect of the drugs. We need a combination of parameters. This is our experience on sequential combination therapy with selexipag on top of PDE5 inhibitors and endothelin receptor antagonists. And as you can see, the addition of the third drug reduce pulmonary arterial pressure, increase the cardiac index, and reduce pulmonary vascular resistance. If we analyze the compounds which interacts with the prostacyclin pathway, epoprostenol, treprostinil, and oral selexipag, you see that they produce the same trend of hemodynamic effect. If we can concentrate on PVR, you see that the reduction is in the same direction, but there is a different power in the efficacy. The more powerful is intravenous compound, intermediate is subcutaneous, and less powerful is selexipag. Initial combination therapy. This was proven to be effective in the ambition study. The initiation of the double combination therapy with PDE5 inhibitor and endocrine receptor antagonist provide a risk reduction of 50% as compared to patients with monotherapy. This is another important signal. And whatever is the type of combination, the efficacy, the effect, hemodynamic effect, is similar. This is the combination of macitentan and tadalafil. Finally, the risk-oriented treatment strategy, which has been proposed in the current guidelines. I remember you that the new guidelines will be published at the end of August, 1st of September this year. And in the current guidelines, you see how the profile of risk of patients is a guide for the different treatments. For example, initial triple combination, including intravenous prostacyclin analog in high-risk patient is recommended, while double oral combination therapy with PDE5 inhibitors and endocrine receptor antagonist is recommended in patients at low or intermediate risk. Then we need to re-stratify the patients after starting or after upgrading therapy, because our objective is low risk profile. And this is also another important concept. We should not be satisfied only reaching the specific combination of therapy. We need to demonstrate that the patient has reached the goal, which is low risk profile. What then about the initial triple combination, which is the topic of my lecture? We have data in which patients at high risk and treated with a triple combination of epoprostenol, bosentan, and sildenafil improve dramatically the hemodynamics as early as 4 months. And the survival of these severe patients also it's very satisfactory if we consider the severity of the patient. So the concept of initial triple is proven. And these are data from the French network showing experience with different triple initial combination, but many of them have utilized the intravenous or subcutaneous form of prostacyclin analogs. And one of the results of this analysis, this registry analysis, is that the more drugs are utilized by the patient, the better is the hemodynamic effect. This is another study combining triple combination therapy utilizing subcutaneous treprostinil. So what about initial triple oral? We have at this point the results of the Triton study assessing the efficacy of initial macitentan, tadalafil, and selexipag as compared to the double combination therapy of macitentan and tadalafil. The primary endpoint was the reduction of pulmonary vascular resistance, and this data were assessed at week 26. And these are the results. There is no difference between the two groups. In fact, both groups achieved a very important reduction of pulmonary vascular resistance and also a very important increase of 6-minute walking distance. But no difference were observed. An additional exploratory analysis was performed on outcome. And this is the combination of initial triple oral therapy as compared to initial double therapy. And as you can see, event-free survival at 2.5 years was 85% in initial triple oral combination as compared to 74% in initial double oral therapy. And if we analyze survival, time to all-cause death, survival of 2.5 years with initial triple combination was 98% as compared to 91%. So these are strong signals, even if they are not definitive data, because the statistical power of this study to show significant difference. But the message is very powerful, and my opinion is that this important difference need to be explored more with additional studies. In the future, we will have different possibilities of triple combination. We have this study is a phase 2 study with sotatercept, a novel first-in-class fusion protein which influences the BMPR2 pathway. Is a drug which is administered subcutaneously in an intermittent way. And the initial phase 2 study showed that there is a dose-related improvement, reduction in pulmonary vascular resistance, and the patients included in this study, a part of them were already on triple combination therapy. So some of these patients were on a combination of 4 drugs. Finally, there has been a suggestion about a different strategy to avoid combination, but to shift the drugs of the same class, delaying in some way the combination. These have been studies open, not blinded, and some of the parameters utilized, like 6-minute walking distance, WHO functional class, were possibly subjectively influenced. The only parameter which is objective and not and there is no possibility of influence is NT-proBNP. And the NT-proBNP was not different in the studies, while 6-minute walking test improved and functional class improved, but we know how those parameters may be influenced by the knowledge of which drug the patient has been taken. So key messages: current PAH therapy is not able to cure the disease. However, it improves outcome if used appropriately. PAH therapy is not a simple prescription of drugs, but a continuous process including a constant reassessment of the effects based on restratification. Use of double and triple combination therapy is supported by robust evidence and is recommended in almost all patients. Initial combination therapy with an endocrine receptor antagonist and PDE5 inhibitor is the treatment of choice in treatment-naive patients. Initial triple oral combination therapy is promising, but require further randomized control trials. Treatment escalation is required if therapy does not result in the low-risk status. Shifting therapy between oral drugs from the same pathway has not been tested properly and might delay combination therapy. Thank you.
Thank you very much, Professor Gallet, and we'll open the floor for questions, please. If you can kindly introduce yourself.
Assalamu alaikum. Dr. Khaldun Lahmoud من الكويت. I'm a heart failure cardiologist, and this reminds me of our journey in heart failure therapy when we started with the triple therapy, then went to the quadruple therapy. We started with the sequential therapy, and then we moved into the more horizontal 4 pillars of heart failure treatment. This kind of reminds me of what's happening here. So with our interest in pulmonary hypertension and taking up this past experience of heart failure, the work on initiation of triple therapy or quadruple, if the new drug that was presented is going to be there, should all doctors be guided towards direct initiation? Is this the future in pulmonary hypertension compared to what we are doing in heart failure, and taking the lesson from that to shorten the period that we took from sequential until we reached to the all together medication? So that's my question.
Yes, I'm also a cardiologist. I know very well our attitude in patients with heart failure. We go directly to 3-4 drugs without any doubt. Even because the study in heart failure were more easy than the study we can perform in rare disease like PAH. So the possibility of triple combination therapy as at the beginning, initial, without taking into consideration not class 4 patients, because for class 4 patients, which are about 5% of our patients, is obvious to give all the drugs very rapidly. But the patients treatment naive in functional class 2, to give them 3 drugs, 3 oral drugs, we need the data. We need the data for several reasons. And this is why it was performed the Triton. Unfortunately, the Triton were not sized for outcome. And so I'm quite confident that if we organize a randomized control study fully sized, we may demonstrate improvement of the triple oral combination therapy. But this has to be done. We cannot do as we did in the past with heart failure patients. In this field, data are very important.
Naz, thank you so much for your elegant talk and for participating in Saph 2022. 2 quick questions for the sake of time. Now, I don't know what went wrong with Triton. Do you think it's the duration that probably too short to assess the change in the primary endpoint, maybe even the secondary endpoint? And how come the PVR was not affected so much, as well as the 6-minute walk test, and yet 2 years later, there was a change in the survival and time to clinical worsening? Do you have an explanation for this?
I have some hypotheses. The hemodynamic effect was quite large for both groups at 6 months. I think it's very difficult to have more than what has been achieved on PVR and 6-minute walking distance. But the discrepancy between outcome and hemodynamic or functional effect is already known. You remember the study with imatinib. In the study of imatinib, we had the opposite. A very important hemodynamic and functional effect, but no effect on the outcome. Apparently, now with this triple, with the Triton, we are observing trend of improvement outcome improvement of hemodynamics and exercise capacity, but the difference between the two groups on outcome requires more time to be achieved. But this is an hypothesis. We have seen also in cardiology in the past, when we tested the oral inotropic drugs, we improved the left ventricular ejection fraction, but we increased also side effect and mortality. So the discrepancy between outcome and the functional parameters is possible because the pathway for improving outcome may be different by the pathway for improving hemodynamics and exercise capacity.
The second question quickly, Saleh. Now, you know, the change in the hemodynamics is very profound when you start prostanoids. You presented the Olivier data and some other data. So maybe it's a time now to, instead of doing Triton with 3 oral combination therapy, to test the hypothesis of starting triple combination therapy, but one of them should be prostanoids. You know, if you have this significant change in hemodynamics, this may be translated in the long run for survival as well.
Yes, these are good hypotheses, but we need the data to support them. I'm very confident that the prostacyclin analogs are very powerful, and in fact, the intravenous and subcutaneous forms are utilized in the more advanced patients. But it's difficult from the acceptance of the patient not to start all patients with triple, including subcutaneous or intravenous, because these are more invasive drugs and will not be accepted by the patients. This is why, in my opinion, the possibility of early triple combination therapy is useful only with these oral compounds.
Just one question. Why the neprilysin is used in heart failure and it causes reduction in the pro-BNP and remodeling of the left ventricle? Can we extrapolate that for the right side of the heart? And the other question is why drugs in the right side is not useful for the left, and the drug on the left heart is not useful for the right heart?
You are perfectly right. And this is true. This is absolutely true. And I have no explanation. Probably the pathways who control pulmonary vascular resistance and systemic vascular resistance are different. And this may explain why, and also the myocytes of the left ventricle may be different by the myocytes of the right ventricle. This may be a reason why beta blockers, endothelin receptor antagonists, ACE inhibitors, and so on are effective in the left heart failure, while they are not effective in right heart failure, and vice versa. I do not have an explanation, but this is absolutely clear. The data we have in the past 20 years says that the two circulations and the two ventricles seems different from the response to drugs.
All right, I think for the sake of time, thank you very much, Professor Gallet. That was a very informative and educational presentation. Thank you for your time and wish you a very good day.
Thanks for the invitation.
I am delighted now to present the second presenter, Dr. Shaya Shaya, our colleague from Riyadh. He is a consultant in pulmonary and pulmonary hypertension at King Fahd Military Hospital in Riyadh. He did his clinical fellowship in pulmonary hypertension at University of British Columbia, Vancouver. He is currently the director of pulmonary hypertension clinical program and the director of adult respiratory medicine fellowship program and co-director of internal medicine residency program in his center. Welcome, Dr. Shaya, and the floor is yours. He will be presenting early IV prostanoids in pulmonary arterial hypertension impact on outcome.
Assalamu alaikum wa rahmatullahi wa barakatuh. Bismillah ar-Rahman ar-Rahim. So today I'm going to talk about the early IV prostacyclin in pulmonary arterial hypertension, and is this going to impact outcomes or not? So I have nothing to, my talk will have no conflict of interest. We have to understand at the beginning that IV prostacyclins has multiple mechanism of action in multiple cells and smooth muscles, endothelial cells, leukocytes. This can be represented by vasodilatation effect, antiproliferative effect, also a platelet aggregation effect. Also, this is decrease the inflammatory action, and so it is a potent pulmonary vasodilator therapy compared to the others. Prostacyclin therapy has multiple forms, like in epoprostanol, treprostinil, iliprost, and selexipag in different form and different routes. We have to understand that, and my talk is almost continuous of the Prof. Gallier talk. We have to know that the treatment of pulmonary arterial hypertension is evolving over the time. So we have to understand the differences and the time manner of using therapy in pulmonary hypertension. So since 1990s till 2015, there is multiple drugs come over and over for treating and managing pulmonary arterial hypertension, and different outcome at the same time has been evolved. In the beginning, they are talking about the 6-minute walk test, the hemodynamic, then they are talking about the end result of time to progress of the disease, time to death, and so on. And also there's monotherapy, sequential therapy, the combination therapy, either upfront combination or upfront triple therapy. So there is multiple things need to be considered when we are talking about epoprostanol today. So we are going to take it like time by time, so from 1994 to 2002. And this is the aerial approved studies in IV prostanoids and its outcome. So the initial study, they are looking into the epoprostanol versus conventional therapy. So the conventional therapy is anticoagulation, oxygen, digoxin, calcium channel blockers, and also diuretics. So there is 3 randomized non-blind studies talking about WHO function class 3 and 4 in pulmonary hypertension using epoprostanol. And also there is another study looking to the scleroderma and pulmonary hypertension. The other important thing that we have to understand, the epoprostanol is the only treatment shown to reduce mortality in pulmonary arterial hypertension in one single study. So all these studies, I'm going to talk about it by one by one. So in 1990, there is a study talking about using an epoprostanol in the beginning. So there is sustained improvement in hemodynamic, symptomatic improvement. The data that's shown that in that study doesn't show the survival benefit because there is premature formal analysis. And also the drug is well tolerated, and they conclude that this treatment can be used as a bridge of transplant before transplantation in pulmonary arterial hypertension who is severely ill. However, 4 years after, the medication has been used more. So, and looking to the survival in this group of patients, so 18 patients has been included, function class 3 and 4, and 1 to 3 year survival was 86, 72, and 63%. Compare the historical data in an IH registry of 77, 51, and 40%. 2 years after, now we are starting to use these medications as a part of the as a part of pulmonary arterial hypertension targeted therapy. So we use epoprostenol to conventional therapy that I have mentioned before compared to the conventional therapy itself. And there is improvement in terms of survival after 12 weeks of using EPO, and also as well as there is improvement in hemodynamics. 2 years after, the epoprostenol has been used in primary pulmonary arterial pulmonary hypertension, as called at that time, by McLaughlin. 38 patients has been included. Long-term treatment is associated with significant reduction of hemodynamics, specifically in this study, pulmonary vascular resistance. Another study after 2 years shows another perspective of pulmonary arterial hypertension. Instead of idiopathic pulmonary hypertension, we are looking to the scleroderma and another targeted patients. So there is improvement in 6-minute walk test, as well as there is improvement in hemodynamics. So in summary, it is a breakthrough at that time till 2002 that there is a treatment can improve outcome compared to all other previous medications. So what about the survival? We need to get to have another look to the survival and treatment in pulmonary arterial hypertension, specifically at that time using epoprostenol. So NIH registry at 1991, they have 32 medical centers, 194 patients included in this study. The estimate as median survival of patient is 2.8, and the survival rate was 68, 48, and 34% after 1, 2, and 5 years in those patients without a specific targeted therapy of pulmonary arterial hypertension. So we need to compare all these things to epoprostenol. So in 1997, there is a study showing the improvement in long-term effect and survival in pulmonary arterial hypertension with using continuous epoprostenol infusion. So 69 patients has been included, function class 3 and 4 as well. This observation shows 80 and 76 and 49%, which is significantly difference between the historical data and NIH. Also in 2002, there is another study looking for the long-term use of epoprostenol in function class 3 and 4 as well. And the improvement in the numbers you can see in the survival from 85, 70, 63, and 55 in 1, 2, 3, and 5 year survival, as well as in this study showing there is improvement in hemodynamics. What is interesting in this article is looking to function class 3 and 4. The one who has function class 3 has better survival compared to the function class 4, as well as if the patient baseline 6-minute walk test more than 380, they have better survival than patient who has less than 380 meters. Whether there is a change by like 112 meters between the group, there's no difference in terms of survival. So the important thing is the baseline 6-minute walk test, not to change the 6-minute walk test. So what about the survival in pulmonary hypertension and the impact of epoprostenol therapy? This study by McLaughlin 2002, 162 patients has been included over 36 months from 1991 to 2001, function class 3 and 4 as well. And what is interesting in this study, the improvement and sustained improvement over this 32 months by in terms of cardiac index, cardiac index, mean pulmonary artery pressure, as well as right atrial pressure, and pulmonary vascular resistance. We can see in the period 1 and 2 and 8, over 32 months, there's sustained improvement and continuous improvement in their hemodynamics. When we compare, and this is when they compare the survival of NIH to this study and patients, there is significant differences in terms of 87, 60, 76, and 62 survival at 1 and 2 and 3 years compared to the historical data. And as well shown there, the functional capacity of 3 is different from the 4. So after 2000 to 2002 till 2015, the campus a bit is changed and switched to different target agents. And as well as there is improvement and evolving more therapy at that time. So the monotherapy, the sequential therapy, the ideas of upfront combination therapy is started. So as I mentioned before 2002, there is no therapy except ibuprostanol, and after 2002, the bosentan start to be there, and then other medications come after. So this is Netherlands study looking to differences between bosentan and ibuprostanol as a first-line therapy. They compare together, and you can see in 2002 there is starting of bosentan when it was approved at that time. So in this study, there is improvement in both arms, either bosentan and ibuprostanol, on both arms, increasing in 6-minute walk test, and bosentan after 1 year, and also after 3 years. As well as in ibuprostanol, there is improvement in 6-minute walk test, and after 1 year and 3 years. What is interesting here, there is more increase in ibuprostanol group compared to bosentan group. However, when we put this 6-minute walk test differences between the bosentan and also ibuprostanol, it was not reflected to survival benefit in this study. So patient without disease progression in bosentan is 72 at 1 year, 57 at 2 years, and 45 at 3 years. And it's almost similar to the ibuprostanol group. What about the survival? The survival in the bosentan group is 93, 89, and 89% at 1 and 2 and 3 years. It's almost similar to ibuprostanol group. So the differences in 6-minute walk test wasn't reflected and wasn't translated to survival in this study. Meta-analysis looking for all the studies that has been published till 2009 by Prof. Gallier and other group. It was looking to all the study from 1990 to 2008. All these medications has been approved, compared most of them to placebo, and also looking for idiopathic pulmonary arterial hypertension. The main target that time is 6-minute walk test at the primary endpoint. Some of these studies looking to to hospitalization and survival benefit or not. What is interesting here, when they take all the medication as subclass groups, either prostacyclins, then endothelin receptor antagonists, as well as the phosphodiesterase inhibitors, the subgroup analysis according to the 6-minute walk test and the class of treatment and the class and the class of drugs, there is doesn't shows any statistical significant heterogeneity in terms of mortality when we compare all these together. So there is survival improvement in this meta-analysis. There is reduction in hospitalization, a favorable result on exercise capacity, functional class, and hemodynamics. But we have to think about a treatment strategy. We need to improve the mortality because the mortality is still high. So another systemic review in 2010 looking for the same almost ideas. We need to look for different way of treating patients. So ibuprostanol is a potent therapy, but we need to change our way of treating the patients. So the exercise capacity has been looked into all these studies, either ibuprostanol, endothelin receptors, and phosphodiesterase inhibitors. All these treatments show the small and significant improvement in exercise capacity, as well as in pulmonary vascular resistance. However, the ibuprostanol groups or treatment and studies shows more improvement in mean pulmonary artery pressure compared to all other forms of pulmonary targeted therapy. In 6-minute walk test, you can see from 0 to 100. There is different study looking to improvement in 6-minute walk test. However, all these studies doesn't show any mortality benefit. And this also, the reduction in mortality was observed in trials, only in the trials that including functional class 4 who severely ill pulmonary arterial hypertension. The studies that doesn't include this group of patients doesn't show any mortality benefit. So treatment is improved survival, mortality is still high, maybe some differences in class 4, heterogeneity to determine the poor prognostic factors and survival. Although the differences in 6-minute walk test and hemodynamic maybe it's higher in ibuprostanol groups, wasn't reflected clearly in the survival benefit compared to others. So we have to switch our gear to different outcome and different way of strategy to treat pulmonary arterial hypertension, to monotherapy, to sequential therapy, to combination therapy and triple therapy, as mentioned previously, the previous slide by Prof. Gali. So this combination therapy between the bosentan and ibuprostanol to prove the benefit of bosentan in 2004. They have a placebo group, 10 patients, including ibuprostanol in this group. The other group is 18 patients with bosentan and ibuprostanol. There is improvement in total pulmonary resistance, and also there is improvement in functional capacity, especially more in the combined group. However, the other study, a Bayesian study looking to sildenafil to ibuprostanol, or sildenafil alone, or ibuprostanol alone. There is improvement and significant differences between the combination therapy sildenafil and ibuprostanol compared to both of them alone. And also this has been also reflected in the combined group or combined medications to the death, hospitalization, and also lung transplantation. So let's talk about different view. We are going to start oral therapy, and if there is deterioration, we are going to add ibuprostanol. Is it going to make any difference? So this is done by Jacob, looking to the patient who deteriorated in oral bosentan to be switched or add ibuprostanol. So after 4 months of initiation of, they include 16 patients. After 4 months of treatment, there is deterioration in patient condition. So ibuprostanol has been added. What is interesting and important there is improvement by 83 meters in the group who adding ibuprostanol, as well as there is continuous improvement up to 20 months after. So this is a significant, important point. We have to mention it here. So also this has been reflected in hemodynamic when they apply 6 months cardiac MRI in terms of all these parameters. There's significant changes in those 16 patients. So what about the upfront therapy with presence of IV prostanoids? This is a French registry and from 2006 to 2010, published in 2014, in IV ibuprostanol. They include 2,009 patients, 43 patients naive of previous any treatment. They are including patient who has started in monotherapy ibuprostanol, upfront combination ibuprostanol with any other oral agents, plus all others pulmonary hypertension targeted therapy. So they have ibuprostanol monotherapy compared to combination therapy, 26 patients, to all other forms of pulmonary arterial hypertension. It is clearly from this study looking to the combination therapy is better than monotherapy of ibuprostanol. So combined ibu plus oral is better than ibuprostanol alone. And this is also better than the other forms of pulmonary arterial hypertension. And this we are talking about functional class and 6-minute walk test. And this also has been reflected, the same for hemodynamics. When you are talking about survival, the group who has started an ibuprostanol, they have better survival compared to the other group with no ibuprostanol and their treatment. When they split the combination therapy and compare it with non-pulmonary non-patient naive or non-ibuprostanol groups, there's difference between the two groups by 90, 85, and 81% compared to 76, 58, and 53% after 3 years of treatment. So ibuprostanol has a difference, make a difference in this group of patients. So what about a triple therapy? So we are talking about combination therapy. What about triple therapy of including ibuprostanol, bosentan, and sildenafil? So this has been published in 2014, 19 patients, a pilot study by Prof. Septon. So what's mentioned here is 6-minute walk test, mean pulmonary artery pressure, cardiac index, and pulmonary vascular resistance is continued to be improved and sustained improvement in triple therapy after around 32 plus minus 19 months of treatment. What is more interesting in this article is talking about the triple combination therapy. All patients has been included in this study are alive after 41 months. The survival is 100% after 1 and 2 and 3 years. The transplant-free survival as well as 94% over 3 years. So we can see there is evolving an improvement and progression of treatment of adding epoprostenol to the therapy. So we can see the previous study talking about monotherapy, combination therapy, and triple therapy. The outcome is a bit based on the time manner that you are talking about. And if you are talking about 6-minute walk test, pulmonary vascular resistance, or hemodynamics, or if you are talking about the survival in the later studies. So let us take a different angle of seeing epoprostenol itself. So let's say we are using epoprostenol. If there is can we see any difference in terms of outcome, in terms of rapid using of the dose or titrating the dose higher than the usual, or formulation of the therapy? So this study is Japanese study looking to this specific point. They have survivor of 32 patients compared to the 14 patients with non-survivor. So 16 of them, they have rapid increase epoprostenol, and the other group, they have slow increase in epoprostenol. The blue lines is representing the survivor. The red one is representing the non-survivor. The main groups, the blue one are the group who who survived, and the blue one, the red one is non-survived. And we can understand from this graph that the higher dosing, they are survived according to their suggestion. Also, this has been reflected when they have rapid increase of dosing of epoprostenol compared to the slow increase. So the rapid increase in epoprostenol shows more reduction in pulmonary vascular and pulmonary arterial pressure compared to the slow reduction or slow increase in epoprostenol dose. And based on all these things, they noticed the survival is different. So after 9.5 years, survival is 100% in patients who has rapid increase in prostacyclin dose. The rapid increase meaning after 3 months, they reach around 20 nanogram per kilogram per minute, and after 1 year, they reach more than 45 nanogram per kilogram per minute. Thank you. What about the hospitalization and the outcome when we are changing the formulation of epoprostenol? So let me make it simple. Let's say we have the old epoprostenol, the thermosensitive, and the other one thermostable, which can be stored for longer time, and this may affect the patient outcome or not. So by switching the patient to more stable forms of epoprostenol, it makes a difference by around 1 to 12% in terms of survival. It's mentioned already by Prof. Galeh. I think answered Dr. Abdel Dalan question. Adding invasive therapy in pulmonary arterial hypertension is not an easy job. Patient with pulmonary hypertension is not just to add therapy. We have to look for multiple things going on around it, because that may affect the outcomes. There is a complexity in using epoprostenol, so we have to take a decision carefully and involve your patient when you are going to use it. The stability of the drug, training of the patient, storage and half-lives, side effects, titration of the dose, line-related complication, and also the cost and health system supplies and drugs. All these it cause major impact on the outcome in your patient. So in summary, epoprostenol over 2 decades is still continuous and evolving. And epoprostenol shows improvement in 6-minute walk test, exercise, and hemodynamics. Improvement is sustained and could be continued as well. Survival is improved. However, there's a questionable regarding some groups. Monotherapy, sequential therapy, combination therapy, all has been tried, but we cannot conclude that epoprostenol should be always used in pulmonary arterial hypertension for multiple other reasons. So It is the first therapy has been shown in randomized controlled trial to improve survival, and it is the opening the door for all other medication to be evolved and started, and a new era in scientific and clinical study of pulmonary arterial hypertension. The result of this study over 20 years is an ibuprostanol continue to be important in therapeutic management of pulmonary arterial hypertension. And finally, the efficacy and tolerability of the ibuprostanol has been considered. More convenient formulation of this therapy looks to set and ensure the continuous use of this ibuprostanol and this group of treatment. And thank you.
Thank you, Dr. Shaya. Ibuprostanol is the first drug to be approved for the treatment of pulmonary arterial hypertension. Thank you, Doctor. It's been a comprehensive coverage for a 20-year clinical experience. If you don't mind, please have a seat with us. As you're attending, we'll have the questions by the end of the sessions. We'll proceed to the third.
Thank you very much, Dr. Shaya. And now we'll go to the next presentation. And it gives me great pleasure to introduce my dear friend, Dr. Hussein Ardi Ghofrani, who is a professor of pulmonary medicine. He's the head of pulmonary vascular research in the Pulmonary Vascular Research Institute in Gießen, Germany. He's a world figure in the field of pulmonary hypertension. And his presentation over the next 20 minutes, he will be telling us about the role of immunotherapy as well as antifibrotic as an emerging possible medications for pulmonary hypertension. Ardi?
Yes, Saleh, thanks for the very kind introduction. I am trying to share my screen and I'm unfortunately not able to do so. But first of all, I'd like to very much thank everyone, the chairs of this very interesting session, the organizers of Saph, and also all colleagues around the world of the PVRI community for allowing me to give my presentation here. I've been listening, very interested to the previous talks that were clinically focused, very important and very knowledgeable. My talk, provided I will be able to share my screen, which is still not possible, unfortunately, will be focusing on something that is rather preclinical and forward-looking. It is about the upcoming potential new therapies, both immunotherapies as well as antifibrotic therapies. Asking the technical staff how I can resolve this technical issue. Yeah, there seems to be something ongoing. Yeah. This is very unfortunate. I'm trying to send Hassan my presentation while this is ongoing.
We're trying RD, so we'll give Hassan a couple of minutes if they can fix the problem.
Yeah, okay.
Just 1 minute. If it works, otherwise, deep apology, we might have to take the next presentation until this is fixed.
Sure.
So we'll just wait for a minute.
Yeah, I'll send Hassan a Dropbox link for my presentation, and then maybe you can open it and forward the slides. No, for me, it's still not possible. Although, yeah, wait a second. Hello?
Record.
So the next talk is also virtual, so if the speaker is ready, we can ask RD to fix the problem and go to the next presentation. Yeah, yes, we do, Artie, so please go ahead. Yes, we do hear you very well. Go ahead, please.
Vascular disease. And for that, I think we should have the development, which was also partly mentioned, in mind, which is based on the hypothesis that, in fact, there are similarities between the phenomena that are ongoing in the vessels of patients with pulmonary hypertension and in tumor growth. With tumor, we associate an unlimited growth, immortal cells, loss of apoptosis, high proliferation, and even metastasis. And some of these aspects, maybe with the exception of metastasis, also apply to cells that we find in the vessels of patients and also the surrounding tissue. We see sustained proliferative signals, we see the resting cell death, and there are many, many similarities also on the genomic level that are shared between this, so to say, pseudomalignant disease and cancer. I think one of the first attempts to address this was work coming from our group with the investigation of one of the very early tyrosine kinase inhibitors, imatinib, which at that time was approved for the treatment for chronic myelogenous leukemia, which in preclinical models of pulmonary hypertension was able to reverse the disease back to normal. And also in a very early case, it was shown that a patient who was already on triple combination therapy, and remember this case was published in 2005, so this was what was the standard at expert centers at that time, already triple combination therapy, received imatinib on top and reduced pulmonary vascular resistance dramatically, alongside with improvements in the exercise capacity and functional class. And the study ultimately, both the phase 2 and the phase 3 study, both were positive. So the study wasn't negative. It wasn't a negative trial. It was a positive trial. Even with regards to outcome, it was clear that the classical time to clinical worsening was not in favor of imatinib, but most of the dropouts and the events that were recorded were associated to imatinib-associated side effects and not worsening of pulmonary hypertension. Nevertheless, the development of oral imatinib at that time was stopped from the company, and it was halted, but it was still thought-provoking and inspiring for future developments to come. I can now not forward my slides anymore. There we are. So how about other classical anti-fibrotic treatments? So coming now to the core of my presentation, and we were intrigued by early developments in the field of idiopathic pulmonary fibrosis, a disease which is characterized by progressive so to say, fibrotic changes in the pulmonary parenchyma, but also accompanied by changes in the pulmonary vasculature. And one of the first tyrosine kinase inhibitors that was considered for the treatment of IPF was nintedanib. Nintedanib was initially also approved as an anti-tumor agent and has a triple kinase inhibitory characteristics, inhibiting both VEGF, FGF, and PDGF. So we were able to show in a specimen from patients with pulmonary hypertension that many of the tyrosine kinases that are tackled by nintedanib are in fact also operative in patients with idiopathic pulmonary arterial hypertension. And therefore, we tested the effects of nintedanib in experimental pulmonary hypertension in both models of Sugen hypoxia combined with placebo or with nintedanib. These data were also corroborated in a monocrotaline model, and it could show, well, one would say at least moderate effects of nintedanib on pulmonary hypertension and also cardiac output and remodeling in these experimental animals. Nothing too impressive, but there was a signal of efficacy. However, we also treated some patients on compassionate treatment basis with nintedanib, and what we were able to show was that in some of these patients, 3 overall that were treated, there was even a slight deterioration in their clinical condition, all together not very convincing what we were able to observe clinically. And we also monitored some biomarkers in parallel from the circulating blood with no major or noticeable effect. That's why we put, at least for us, the development of nintedanib for pulmonary vascular disease aside. Another antifibrotic agent, hitherto not considered for treatment of pulmonary vascular disease, pirfenidone, also approved for IPF. And pirfenidone is what they say a kind of dirty drug. No one really knows how it works and why it works, but it worked in IPF. And therefore, we exposed again our preclinical models with incremental dosages of pirfenidone, being able to show that it has, in fact, anti-proliferative capacity, not only in fibroblasts, but also in pulmonary smooth muscle cells, and reduces proliferation in a dose-dependent manner. So there seems to be some notion that it could work in future. This has to be tested also in pulmonary vascular disease. And I believe that before we jump into group 1 pulmonary arterial hypertension, we should have a closer look at patients with pulmonary fibrosis that have accompanying pulmonary hypertension who receive pirfenidone with or without nintedanib, and see whether or not and pre-existing pulmonary hypertension alongside with improvements or at least stabilizations of the fibrotic component may also benefit from treatment with these anti-fibrotic agents. So another immunomodulatory and anti-fibrotic approach obviously could be, and this is more and more addressed also clinically, with agents that interfere with the BMPR2 signaling. We know that the change in BMPR2 genetics in some of our patients result in the development of hereditary pulmonary arterial hypertension. We have just learned that upcoming novel therapies such as sotatercept, which interfere with the activin receptor type B, may indirectly also interfere with the BMPR2 signaling. And there are attempts to substitute the ligand to the BMPR2 receptor by substituting genetically engineered BMP9 and 10, work that is very significantly done at Nick Morell's site in Cambridge. So BMPR2, in fact, could be an immunomodulatory and also anti-proliferative approach for the treatment of pulmonary vascular disease by rebalancing the ratio between proliferation and anti-proliferation in this disease area. Now to the second topic of my talk, immunotherapies for pulmonary vascular disease. And I have to say, I wasn't even considering immunotherapies for the treatment of PVD before I was subjected with a task for this presentation. And we have not so far done work in our own basic labs with this approach, but we should keep in mind that there are already reports out there that although few, but maybe with an incremental noise around it, that some of the checkpoint inhibitors, PD-1 checkpoint inhibitors that are very successfully implemented in the treatment of non-small cell lung cancer may in fact even induce pulmonary hypertension. Here's a very well described case report from Tim Lam's group, which I recommend you to read, who could show that a patient who, because of lung cancer, had multiple cancer therapies, including novel TKIs and classical chemotherapy, with the introduction of the PD-1 checkpoint inhibitor, developed severe pulmonary hypertension. And yes, I can imagine which question you have. They have excluded pulmonary embolism as a potential confounder and did not find anything else than the conjunction with the checkpoint inhibitor therapy. Having said that, there are some reports about immunotherapies, and there is a very interesting paper published in the JACC journal in 2019, where vaccination against endothelin receptor type A was used as a potential therapy for experimental pulmonary hypertension, at least. And in this study, in a monocrotaline model of pulmonary hypertension, an active control of bosentan was used, and compared to bosentan, the vaccine was equally effective in reducing pulmonary hypertension. And as another positive control, a monoclonal antibody against endothelin receptor A was used, and it was as efficacious. Now one could speculate what's the use in developing vaccines with their own side effect profile or very expensive monoclonal antibodies if we have very elegant small molecules that we can use. We have many approved endothelin receptor antagonists, but in fact, this opens speculation even for a kind of preventative therapy, should it be efficacious, and at least opens door for a very interesting novel development, which we should keep in mind now, after having seen that if you have identified the eligible target structure, you may even use vaccination. And with the development of very smart novel vaccination techniques like the mRNA vaccines, I wouldn't be too surprised if we maybe in the next 5 to 10 years also tackle pulmonary vascular disease with vaccination. There is another group of agents that are potentially interesting. They are also from the box of anti-tumor therapies, the so-called CDK inhibitors, and palbociclib and dinaciclib are approved therapies for the treatment of breast cancer, for instance. So there is clinical experience using those agents. They are comparably well tolerable, and our working group has spent a lot of thoughts into testing these agents because we see signals that are going along the CDK signaling pathway, that there is preclinical evidence that these drugs may work, and we suggest to continue the work on the clinical field. Another immunomodulatory pathway, the FOXO pathway, which is an intracellular signaling pathway that directly addresses signaling into the cell nucleus and induces or suppresses the reading of our DNA with a subsequent production of proteins, is the FOXO members of signaling molecules. And Soni Pulamsetti from our group found that FOXO is strongly upregulated in both human as well as experimental pulmonary hypertension. It was very remarkable to see in idiopathic PAH patients how strong this signaling pathway is upregulated, particularly the phosphorylated form of FOXO. And that's why by genetic manipulation in experimental models, the FOXO signaling pathway was promoted, showing that it could impressively reduce experimental pH. And also there's a small molecule, again, one that we could use as a repurposing of drugs, because paclitaxel is now very well known in the oncology field and used as an oncological agent, as well as it is used for prevention of restenosis in cardiac disorders and peripheral arterial occlusional disease. So paclitaxel is at hand, and we are starting an investigator-initiated trial with inhaled paclitaxel based on funding coming from the German government very soon. It is in fact utilized already in preclinical models with inhalation of paclitaxel in the Sugen hypoxia model, showing that cardiac function and also pulmonary hemodynamics could be beneficially influenced. So in a very kind of snapshot, I wanted to just show you that vascular cell growth plays a major role in pulmonary vascular disease. We know from imatinib that it is efficacious. However, the NDA has been withdrawn due to unforeseen side effects, but there is potentially novel developments ahead that we can see. We know that seralutinib is currently under development for the treatment of pulmonary hypertension. Seralutinib is a novel tyrosine kinase inhibitor. And we also know that inhaled imatinib and also a novel formulation of oral imatinib are currently under investigation. We also look forward to potential development of novel antifibrotic agents and the investigation whether or not pirfenidone and nintedanib may offer some potential in pulmonary vascular disorders. And also the immunotherapies, namely the vaccinations, could offer in future some merits for pulmonary vascular disorders. I'd not like to close without acknowledging collaborative work from my colleagues from the Imperial College in London, from the Max Planck Institute for Heart and Lung Research in Bad Nauheim, with my colleagues from Gießen, and also thank our sponsors, which are mainly sponsors from the German government and the German Research Foundation. So I'll stop here, I'll stop sharing my screen, and I'm happy to answer any.
Thank you very much, Arti. Excellent talk. Obviously, further research is needed to find out about the potential use of these agents. And who knows, we might be sitting here in a few years talking about these as first-line medications for PAH. We'll have a couple of questions for Dr. Ghofrani, please.
It's Luke Howard here. My first question is, why the hell aren't you here? You don't have to answer that one.
It opens the country for travelers. So our Bundeskanzler is more hesitant, and that's why we are still very restricted in our travel abilities. And also locally, my hospital still serves a lot of COVID patients, and that's why internally we have a travel ban that kept me from coming. Otherwise, I would have loved to come, as you know.
As you know, in UK, it's party time. So my question to you is around pirfenidone. And I'm just wondering if perhaps pirfenidone may be of more relevance to the right ventricle. I presented a year in review yesterday and very briefly touched on the potential for pirfenidone to reduce extracellular volume in the left ventricle. As you know, we still really struggle to find treatments that benefit the myocardium directly. Do you think that may be an area to explore of particular interest for this drug?
Yeah, I should have listened to your year in review because that's very intriguing. In fact, we used pirfenidone also in the pulmonary arterial banding model. And in fact, it reduced extracellular matrix and also gave way to cardiomyocytes in favor of myofibroblasts. So there is some noise about potential benefits of pirfenidone on the myocardium. I was so far not successful in convincing our colleagues from the IPF field, Ganesh Raghu and others who were instrumental in the IPF studies, to look deeper into their data. One way forward could be to look specifically for the subgroup of patients in the IPF trials that had a signal for pulmonary hypertension. This exercise has been undertaken in previous trials, but for pirfenidone and also for the nintedanib study, unfortunately, I'm not aware at least of this exercise, because there one could see in real life whether or not it had a direct impact on also the pulmonary vascular component of the IPF disease.
Hopefully I'll see you in San Francisco.
Definitely. San Francisco is booked.
Great.
All right.
It's later the year, so we need to get rid of the COVID restrictions until then.
Soon, inshallah. So we'll have two more questions. Can you introduce yourself, please?
Afrani, my name is Fatima Haji. I'm a rheumatologist from Bahrain. Thank you for a very fascinating lecture, actually. My question comes from the rheumatology background. So I was looking at one of your slides. There is the etanercept was kept, and it's an anti-TNF, in one of the slides that we're showing about the sotatercept. Do you think anti-TNF has any role in the management of PAH, and is that the reason why better treatment of rheumatoid arthritis is the cause why they do not have the incidence and prevalence of PAH is not that high as the others? And what do you think about the anti-CD20 and tocilizumab, as they're all immunotherapy are used in systemic sclerosis? Any role with PAH in that? Thank you.
I can say yes to both of your questions. I think they play a role, but we need to understand better which subpopulation of the patients might benefit, and also we need to solidify the science behind it in specific models. Some of the anti-TNF approaches have been tested in preclinical models of pulmonary hypertension. I'm trying to intensify the collaboration between our PH community and the rheumatology community. I have very good friends in EULA and EUSTA, and also some that are interested in preclinical science. And we need to understand each other better, because we have an interesting overlap of agents that might be helpful on both sides of the aisle. So yes, I think that rheumatoid arthritis drugs, particularly the new biologics, may also be interesting for patients with pulmonary vascular disease.
Hi, I'm Dr. Khaldoun Al-Hamoud. I'm a heart failure cardiologist. Thanks for the elegant lecture. My question is both driven by my ignorance and curiosity regarding the fascinating comparison between how it behaves like cancer. Out of eager curiosity, has any pulmonary hypertension patient who developed cancer, treated for cancer, showed any improvement by cancer therapy simultaneously? Thanks.
Not to my knowledge. I wouldn't be surprised if there were cases which I'm not aware of. We have worked the other way around and are currently looking at a lady at our institution who suffers from breast cancer and receives palbociclib and has pulmonary hypertension. She had pulmonary hypertension even before her breast cancer, and she was quite stable with her classical pH medications, experienced, unfortunately, a breast cancer, and then got palbociclib therapy as a maintenance therapy for her breast cancer. So now we are monitoring her very closely. The therapy is not on board long enough, so we can really judge whether or not it has a potentially positive effect, but we will monitor her closely, and I'm really looking forward to other cases or case collections so we can combine our experience and derive some notions. There have been various reports about use of different tyrosine kinase inhibitors for their primary indication, and there were also investigations whether or not the occurrence of pulmonary hypertension was more or less in those primary tumor indications. One negative example, obviously, is the example of dasatinib, also a tyrosine kinase inhibitor, which has a higher incidence of inducing pulmonary hypertension. So we should be very, very specific when considering agents and really look at them one by one, because we cannot generalize for the group of agents that we are investigating.
Thank you very much, Artie. That was an excellent talk, and thank you for answering the questions. Enjoy your upcoming weekend. Thank you.
Thank you very much. I'll stay on board and listen if I'm allowed, but it was a pleasure. And thanks for sharing so elegantly and friendly, Saleh. I wish I would have been there.
Take care. Hope to see you next time.
Our next presenter will be Professor Anton van Noordegraaf from Netherlands. He is a full professor and chair of the Division of Pulmonary Sciences at the Amsterdam University Medical Center, a tertiary referral center for pulmonary arterial hypertension in the Netherlands. He is a professor who published over 300 articles in peer-reviewed journals. His research is focused on pulmonary arterial hypertension, right ventricular failure, pulmonary hemodynamics. He is nucleus member of the working group Pulmonary Circulation of the European Society of Cardiology and Pulmonary Vascular Diseases of the European Society of Respiratory Medicine. Welcome, Dr. Anton, and he will be presenting RV remodeling, target for treatment and potential risk stratification parameter.
Thank you, Dr. Aldemas, and also thank you for Dr. Aldola for having me here. To answer the question of Luc, why I'm not in Jeddah, well, I tested COVID positive just 24 hours before my plane was leaving, so that's why I'm here. But I have no symptoms, and don't be afraid, I will not spread the virus through the virtual kennel. I the presentation will be on the right ventricle remodeling, target for treatment, and potential risk stratification. I will. Sorry, there's somebody else speaking, which is also good. I will not talk about all the potential treatment targets we do have on the right ventricle, because that will be a topic on its own. I will not talk about all the new medications which are upcoming and which have also a direct effect on the right ventricle, as already Professor Giovanni pointed out, because that also would be a direct topic on its own. But I do think the right ventricle and the effects of the drugs we are studying now are very interesting, especially also the discussion on periferdonia. I will talk about the effects of the drugs we use for the pulmonary vasculature and how they affect the right ventricle in a direct way, in an indirect way. So first of all, how important is the right ventricle risk assessment? Then can we create reverse remodeling of the right ventricle? And can we use the right ventricle as a treatment goal? The first question is very easy to answer, because the disease is about the right ventricle. How does the right ventricle cope with the load? If the right ventricle copes with the load, sorry for the automatic movements, then it's fine for the patient. When the right ventricle is unable to cope for the load, even if the load is low, then we do know that our patient is failing. And this is seen in all the risk stratifications. We have the parameters of the forward failure, so the cardiac output, stroke volume, mixed venous saturation, and we have the backward failure, right atrial pressure, pericardial fluid, or we have the forward failure parameters like exercise capacity and maximum oxygen consumption, or the backward failure, which is venous congestion and fluid retention. This slide is timed for some reason or the other, so that's why it's moving forward and backward, but that's okay. And NT-proBNP, of course, covers everything. We do know that the MRI adds, the right ventricular parameters adds to the clinical parameters. So if we add the MRI parameters to the clinical symptoms, like the 6-minute walking distance, NT-proBNP, we do know that the sensitivity and the specificity of the outcomes is better predicted. This is a study from Andy Schiff from Sheffield, again showing that even direct imaging parameters adds to our risk assessments. Then the question is, of course, can we non-invasively assess parameters of the right ventricle by means of MRI imaging or echo imaging? We studied this in a study recently published in Chest, and what we did is we looked for the predictive value of the MRI in the follow-up of patients with idiopathic PH after 1 year and compared it with the invasive measurements we did at the same time. So idiopathic PH, measuring CMR and right heart catheterization at the same time, follow-up after 1 year, and we split it up with survivors and non-survivors, and then we compared it to what happened at the follow-up time to predict the value of the right ventricle in the risk assessment. And what it turned out in univariate Cox regression analysis, and these are the follow-up parameters, no surprise, in the right heart catheterization, the right atrial pressure and the cardiac index was so important. And of course, this is very well known already from D'Alonso data from the previous era. And for the MRI, it was stroke volume index, right ventricular ejection fraction, right ventricular end systolic volume, and right ventricular end diastolic volume, which of course makes a lot of sense based on all the literature.
Professor
Yes?
Anton, please, can you make sure if your the slides are working, because for us it's fixed on the first slide. Sorry, we have some technical issues. So, yeah, right. Now it's good. Thank you. Yeah, it's okay. But can you put it on the full screen, please?
Yeah, but if I do it on the full screen, then it seems not to follow. Okay, but I will try to do it on the full screen. You tell me when it's not moving.
Okay.
So again, this slide shows what we already know. It shows that the hemodynamic parameters which predict prognosis are invasively right atrial pressure and cardiac index, and non-invasively stroke volume, ejection fraction, right ventricular end systolic volume, and right ventricular end diastolic volume. Is it moving?
It is fixed.
Why? Then I had to make it a little bit. Is this okay?
No, it is just zoomed, but the slides are not changing. Yes.
Okay, I will do it in that way. Okay, then we looked to the predictive value of the different models. So we looked to the invasive model, we looked to the hemodynamic model, and we looked just to functional imaging model. How best does it predict risk? And for that, we use the I-key index, which just predicts the best model of fit. And what we learned from this is, first of all, that the parameters which are predictive are the parameters we already know from all the risk scores we do have. But the other important thing we learned from this is that just a risk score based on imaging and functional assessment, so including the age, sex, 6-minute walking distance, and right ventricular ejection fraction, was just as good as taking into account the invasive measurements. And for that, direct assessment of the right ventricle by means of imaging, in this case it was MRI, so we don't have this data for echocardiography, provided with a similar risk assessment as the invasive measurements. Now I'm moving to the next topic: can medical treatment create reverse remodeling of the right ventricle? And again, right ventricle failure is characterized by enlargement of the right ventricle and a decrease in stroke volume. We do know the sequence of failing. So we do have the normal heart, then we have the hypertrophy and not so much dilatation. But then at a certain moment in time, the right ventricle adaptation is not enough, and then the right ventricle is continuing to dilate, right atrium size is increasing, and also the stroke volume is dropping down. So the two hallmarks of the follow-up of the patients with PAH reflecting this is a decrease in stroke volume and an increase in right ventricle and systolic and diastolic volume. And so simple it is. And when we reverse this system, for instance, after a lung transplantation or after pulmonary endarterectomy, we decreases the size of the right ventricle to its normal size. We decreases right ventricular hypertrophy, but also we decreases the size of the right atrium. So there is really a reverse remodeling. And this slide, and I will enlarge it again, is from the transplantation service from Groningen, and they just looked at severe PH patients after lung transplantation. And what you see is if you reduce the pulmonary vascular resistance to normal values, then the right ventricle recovers completely to really normal values. So the size normalizes, the stroke volume normalizes, and even the ejection fraction normalizes to the normal values. So we do know that the right ventricle can reverse to almost to the complete normal situation. Now the question is, can we do the similar things with the treatments? And this brings a little bit together the first two presentations. Can we be so aggressive that we normalize or can almost normalize pulmonary vascular resistance? Well, we do have data on this, and this data from Marielle van der Veerdonk from 2011. And she looked at the change in pulmonary vascular resistance and the changes in the right ventricular ejection fraction. And what you can see from this slide is if you reduce the pulmonary vascular resistance for at least 45%, and remember that number because it is repeated in every study, then the right ventricle is recovered and the right ventricular ejection fraction is improving. Before that number, so if you have a less reduction in pulmonary vascular resistance, the right ventricle can either recover or not, as you can see on the blue dots, which are on both sides of the horizontal axis. So this number was also found by other groups, and this was a group from Sans in Dieppe who did a modeling study on the right ventricle and found that if you have a 45% reduction in pulmonary vascular resistance, your probability of that there is reverse remodeling of the right ventricle is almost 100%. So you had to have quite a reduction in pulmonary vascular resistance before you can be sure about that. As you can see that. Why is this? Why is this so much? Do you need so much reduction in pulmonary vascular resistance before the right ventricle follow? And the reason is very simple. First of all, the right ventricle recovers from its stroke volume, so cardiac output goes up. And after that, and the pulmonary vascular resistance drops more down, then the right ventricle will recover as well, because then the pulmonary artery pressure also will be reduced. Now I will summarize a little bit the first two presentations and bring it back to the right ventricle. So from 1 to 2 treatments, and from 2 to 3, from 3 to 4, as Adi already alluded to, because this might be the future. Why does it matter for the right ventricle? Again, a study from Marielle van der Veerdonk. We compared in a retrospective way, and I will enlarge the slide. In a retrospective way, we compared the monotherapy area and the combination treatment area. And this was done by MRI again. So we looked at the right ventricular ejection fraction, the right ventricle and diastolic volumes. The open bars are the monotherapy, and the gray bars are the combination treatment arms. And what you can see here from this is that the combination treatment had much more effect on the right ventricular ejection fraction than the monotherapy. And monotherapy could be either sildenafil, or it could be either bosentan, or it could be even prostacyclin. So combination treatment had more effect on the right ventricular ejection fraction, and this is also pointed out in the pressure volume graphs from this slide, from this study. And I will again enlarge it. So this is the normal situation. Here we have the right ventricle. The normal right ventricle is a normal stroke volume. Then we have the situation before the therapy. Then the pressure volume loop is moved to the right side. And after the combination treatment, you can see that the pressure volume loops is a bit going more to the normal situation with a drop in the right ventricle systolic pressure, which is an enlargement of stroke volume, and a better situation as in comparison to the situation to monotherapy. So even in the pressure volume loop, you can see the improvements. Coming back to the study from Olivier Sidbon from the French series, he showed dramatic improvements if he applied upfront triple agent therapy to a very selected group of patients, young patients, female patients, with mainly idiopathic PH and hereditary PH, with dramatic hemodynamics, so high right atrial pressure and so on, and a very low mixed venous saturation. He looked to it, and then he looked to the improvements. And what you can see from this slide, and I'm just only looking to the pulmonary vascular resistance, is that there is a dramatic drop in pulmonary vascular resistance if you initially apply intravenous prostacyclin together with endothelin receptor antagonist and PDE5 inhibitors. And again, it's a retrospective analysis for a selected group of patients, but this at least might explain why we do find these graphs, and again, I will enlarge it, as already Professor Collier pointed out, this interesting finding from the retrospective analysis that survival benefit is mainly found in the patients, and that's the red line, who had initial combination treatment, prostacyclin together with the other ones, and not so much a difference in survival between a mono and a combination treatment. And of course, this all has the restrictions of selection bias and retrospective analysis, but still it is very intriguing, and it fits in the idea that the right ventricle needs a very dramatic improvement in pulmonary vascular resistance before it can recover and can provide a long-term survival. And this is another study from an echo study coming from my friends from Italy, again showing that if you drop the pulmonary vascular resistance by means of the triple agent treatment, so the closed dark dots in this slide, and compare this with the triangles, historical cohort monotherapy, that the echocardiographic parameter, the right ventricular fractional area change, only improved in the triple agent treatment. And again, of no surprise, you can see that the difference is almost the 45% change in the pulmonary vascular resistance. So our treatment aim, in my opinion, in the patient is at least obtain a pulmonary vascular resistance drop in 45%. At least then we can be sure that the right ventricle is benefiting from the treatment. So then the question is, okay, we don't achieve this in all our patients, so can we use the right ventricle monitoring as a direct way to treat the patients? And can we use it as an endpoint, for instance, in studies? For the first time it was used as an endpoint in a study. It was a phase 4 trial. This study is published this month, so it's very recent. It's the REPAIR study. We just looked to the effects of adding a drug, macitentan in this case, to the right ventricular structure function on patients with pulmonary hypertension. So the patients could be either treatment naive or could be on PDE5 inhibitors or other drugs, but not an endothelin receptor antagonist. And we looked just to the non-invasive measurements for right ventricular stroke volume by means of MRI. Can be measured very accurately and probably more better than invasive measurements, stroke volume, and also to the pulmonary vascular resistance, and the secondary endpoints were all the MRI parameters. The study setup was very simple. It was a phase 4 trial, multi-center based, and it was prospective, single arm, so no placebo arm, and we used the MRI in the right heart catheterization at the initiation of the treatment, which could be that the patient already was on treatment, and after 26 weeks. These were the inclusion and exclusion criteria, which is quite standard. And again, I had to remind you that it could be either in treatment-naive patients, patients receiving stable background PDE5 inhibitors, or in treatment-naive patients in which the macitentan was added initially with PDE5 inhibitors, and this was equally spread, so 1/3, 1/3, 1/3 of the patients. The results are pointed out on this slide, and it showed that indeed the right ventricular stroke volume improved, and it improved most in the initial combination treatment, and also that there was a drop in pulmonary vascular resistance, and also that the right ventricle, as you can see on the left slide images, increased its size, and the left ventricle decreased its size under this treatment. Looking to the secondary endpoints, which were of more interesting, you can see that everything moved in the same direction, which is in the direction of improvement. So right ventricle, I'm not sure that you see it, so I will enlarge it now. Right ventricle end diastolic volume decreases, which you expect as right ventricle end systolic volume, because improvement of the right ventricle is a decrease in volume, and improve in right ventricular ejection fraction, and a decrease in right ventricular mass. So it is just a strategy to show you that by means of non-invasive imaging using the right ventricle as an endpoint in a very small study, actually, you already can see the benefit from the drug on the right ventricle in a phase 4 trial setting. And of course, this can be of more importance if you are using drugs which has both benefits on the pulmonary vasculature and the right ventricle. You want to monitor directly what's going on the right ventricle. Then a more direct way can be also use the right ventricle as a treatment goal. So say, well, we wanted to have an improvement in the right ventricular ejection fraction, and let's try to titrate the medication on it. We have only one single center experience study on this, recently published. It's not a large study, and it has all its limitations of a single center study, but at least it shows the feasibility of it. Again, it was 30 patients, treatment-naive patients with idiopathic IPH or hereditary PH, year 3 or 2, no contraindication for CMR, and the clinical worsening was derived as hospitalization or reduction in the 6-minute walking distance of more than 15%. Patients could be at the entrance being a near 2 or near 3. If they near 2, then they could baseline combination therapy when they had an hormonotherapy, when they had a destabilization after 4 months, moving to combination treatment, and then moving on to triple therapy, so adding prostacyclin. And every 4 months, we made the follow-up measurements. And the near 3 patients started at combination treatment and moved to triple agent therapy if they were deteriorating. And actually, in the study, total of 11 patients of the 30 patients deteriorated, so had a deterioration in right ventricular ejection fraction of more than 3%. And all of those patients had an escalation of treatment afterwards. And here you can see the results. You can see that all the right ventricular ejection fractions improved after you escalate the treatment. Of interest is also the effects on the 6-minute walking distance. You might expect that 6-minute walking distance already predicts deterioration, but it did not in our patients. So we had a deterioration in right ventricular ejection fraction, but there was not so much a deterioration in 6-minute walking distance, only non-significant changes. And this is also very clear. First of all, the right ventricle will deteriorate, but try to adapt, so keep its stroke volume up. And clinical worsening is just following after the right ventricle deteriorates. So this indicates that following up the right ventricle gives you a little bit the advantage of being ahead of the deterioration. The same is true for the anti-proBNP. The anti-proBNP followed much better the changes in the right ventricular ejection fraction, and probably the anti-proBNP is the best surrogate measure for the right ventricular ejection fraction to follow up our patients and to escalate treatment directly if NT-proBNP is going up. So coming to conclusions, the right ventricle reflects the risks of the patient and can be assessed non-invasively and can even replace the invasive measurements. The most important measurements for the right ventricle are stroke volume and the volumes, and for the invasive, of course, the right atrial pressure as well. Only if the pulmonary vascular resistance is reduced by more than 45% coming from many studies, we can be sure that the right ventricle is recovered. And therefore, we needed to be more aggressive, probably more aggressive initially, and therefore we also need novel drugs to achieve this.
Prof? Sorry, the slide is fixed again, sorry.
Okay, the most important slide. Okay, I will move it up now. Now you see it?
Yes.
And finally, and I'm very sorry that it's so fixing, so I had to use this trick all the time. But at least, and then the final point is right ventricular function can be used as a treatment goal, and it makes worthwhile to use imaging in the future as an endpoint, secondary or probably primary endpoint, especially if we are now looking to drugs which directly seems to affect as well the right ventricular function. So the case of Perifidone was mentioned in the previous presentation of Professor Giovanni. It might be a good one because you reduces collagen and stiffness. It might be a bad one because you needed some stiffness of the right ventricle. And therefore we really needed, especially if we entering the new era of novel drugs, molecular drugs, attacking molecular pathways, we needed these insights. So by that, I want to thank you for your attention, and I'm open for questions. And again, sorry for the slide presentation problems. I don't know why.
Thank you, Professor Noordegraaf. It is clearly mentioned that combination therapy is more effect on the right ventricle. So I will start with a question until we get further questions. Having decided upon a patient, such patient on combination therapy, and having such data about certain medications with clear RV remodeling effects, does that affect the choice of the drug to be used in the combination therapy?
Thank you for this question. At this moment, we think that most of the remodeling on the right ventricle is just by reducing pulmonary vascular resistance, as is also clear from the repair study. But we also do know that the drugs has direct effect on the right ventricle. And we needed really sophisticated models to sort out which is the effect directly on the right ventricle and which is the effect on the pulmonary vascular structure, reducing its load. So we needed animal work for this as well, and we needed pressure volume loops and elastance measurements in the cath lab to really sort it out. Because the right ventricle is attached to its pulmonary vascular load, so I'm not sure that macitentan effect was a direct effect. I do think it was just the effect of lowering the pulmonary vascular resistance. But we do know animal data that it has also some impact on the right ventricle.
Thank you. We have 2 more questions.
Yeah.
Please, Doctor.
Thank you very much. My questions, in cases of severe left heart failure, when we use LVAD as a bridge for transplant, some cases they recover to normal without even transplant. Can we implement this using RVAD as a recovery for the right ventricle in advanced right ventricular failure? This is one question. The other questions for RV assessment, you used 2D ejection fraction, 3D ejection fraction, longitudinal strain to assess the RV systolic function. And the third question is any RV score to assess the severity of mild, moderate, severe RV dysfunction?
Thank you very much. And first, starting with your question, I spent actually 3 years of my life to answer that question, and it's unresolved. Can we save our patients by means of a right ventricular assist device? And I find it very intriguing, and I spent a lot of work on it. Actually, it also changes the flow in the pulmonary vascular system, and so it makes it a more continuous flow in the pulmonary vascular system as a consequence of that. we do know from single lung transplantation patients that if you silence the flow in the abnormal lung, that it can recover. So if you just reduces flow, the pulmonary vascular system can recover. So that makes a very strong argument to say, well, if you can bypass the lungs, probably we can recover the disease. And I have patients who had received a single lung transplantation. The other lung, the donor lung, was rejected. So they are using their own diseased lung, and the pulmonary vascular resistance is much lower than at the time of the transplantation. So silencing flow is something, but using the RVAD, we not only silence the flow, we also change the flow patterns. And the question is, how does this interact with the endothelium and the pulmonary vascular systems? And from now, at least from the animal studies in the piglet, then the pulmonary vasculature seems not to be so enthusiastic about this, because then it doesn't reverse to remodeling in our measurements. But having said that, I'm still working, trying to find that out, because it would be a great solution to silence the progression of the diseases in the lung and also recover the right ventricle. But another problem to relate to this is if you put in the right ventricular assist device, then you immediately weaken the right ventricle, because the right ventricle is an athlete. And if you just keep him out of his sport, then it will get in an atrophic state, and you can't keep and if you then the load is not reduced and you stop the right ventricular assist device, immediately you will be a problem. So currently it is not feasible to use in the clinic, but there are many interesting questions remaining, especially bypassing the flow through the lungs. Then the other question is about the parameters we can use, the right ventricle strain analysis, the shortening of the free wall. And there are many parameters. Most of the parameters have been studied in single center studies and only not related to so much to the other. And I would say that I'm not recommending one parameter, but recommending the complete picture. And either you use the echo or you use the MRI, it doesn't matter so much. It should make sense. So if your strain is going down, your right ventricle is going dilating more progressively, and you can also have some indirect measurements of stroke volume, then you can be sure that the patient is not doing well. I would not rely on one single parameter. And then the final question, of course, that makes the most sense of it. If that is the consequence, then you might probably better go for right ventricle score, which incorporates many parameters. And I agree again with you that that would be the most perfect way, because it's not only TAPS, it is not only this or that. Probably the MRI has the advantage of measuring accurately the volumes, which are very mandatory in this field, but it's not available. So we had to need an echo score, which it uses different parameters, which are more or less unrelated, and which can be done. And I'm really keen to see this develop, because I do think this is what is needed. But we needed robust data to prove the value of it.
Thank you.
I hope this answers your question.
Hello, Anton. It's David Jenkins here from Cambridge, UK. I very much enjoyed your talk as usual. I wanted to ask about the studies showing a reduction of 45% in PVR needed for RV remodeling. Do you think this applies only to group 1 PAH patients, or is it more generic for patients across the PH classes? The reason I'm interested is for the group 4 patients with CTEPH, the pioneers of pulmonary endarterectomy surgery, Philip Darteville and Stuart Jameson, always said you should aim to reduce the PVR by at least 50% to get the best results for patients. So it was very interesting to see the science behind that in those studies. But do you think they relate to other classes of PH, or is it just group 1?
Yes, I do, and it's quite disappointing that early clinicians already found out what I tried to prove.
Exactly.
So, but in that,
surgeon view of 50% reduction seems to be exactly the same as the science.
It's all looking to the patient, and indeed, and so it really applies to all types of PH and all classes. And the reason why is that the right ventricle is not the purpose of the body. Maintaining the blood pressure is the purpose of the body. So the first thing which goes on is that if you reduce pulmonary vascular resistance, let's say by 20%, all this benefit is immediately used for increasing stroke volume. And as a consequence of that, the product of stroke volume times mean pulmonary artery pressure, which is the work of the right ventricle, remains the same. So that means that the right ventricle not per se benefits from this. And normally, so it can be or cannot be, but normally, but if you reduce it by 45%, then stroke volume is at least normal in all cases, and the stroke work is reduced in all cases. And that is the reason that for that we do know for sure that by 45 or let's say 50%, the right ventricle benefits. And that is also for the surgeon important indeed, because you wanted to have at least reverse remodeling after surgery. So in high pulmonary vascular resistance after surgery is very bad. So yes, I do think it is the case for every type of PH, and there is physiology behind it.
Thank you.
Okay, so we're enough with the questions. We'll just take few or one question for Dr. Shaya. Thank you, Professor Anton. We'll back to our speaker, Dr. Shaya. He's been here. We'll just take one question, if any, for his talk. I would like to remind you it was about the early IV prostanoids in PAH and how it impacts on outcome.
Hi, Assalamu alaikum. Dr. Shaya, my question is to you. I mean, like having epoprostenol on trials, you will have a very strict inclusion criteria. You will have the greatest epoprostenol nurse and a team that can work on it to deal with all the complications of the lines and the logistic stuff behind it. Now, in reality, and then again, my background is rheumatology, with the patients who have systemic sclerosis or other diseases that are already bombarded with immunosuppression. What's your input on putting the IV epoprostenol, the rate of infections and complications in those categories, please?
So first of all, we have to understand that the idea of epoprostenol and pulmonary arterial hypertension is a potent therapy, has significant improvement in all the parameters that we are already talk about, especially in a group 1 pulmonary arterial hypertension. So although there is a benefit and there is a risk for all these things. So whether we are talking about mortality or survival, or we are talking about the infection and the risk of infection in this group of patients, yes, there is a risk of infection, especially in patients who are immunocompromised. There is a risk of infection even if they are not immunocompromised, but the mortality is higher if we are not using these kind of medications. So I would say if you have a patient with severe pulmonary arterial hypertension, especially in scleroderma, those people who has a higher risk compared to all other groups of pulmonary arterial hypertension, this kind of medication is important. And if they have severe disease of PH, the risk of mortality is higher compared to the others. So I would not withdraw this kind of medication in terms of risk of infection. Taking care of the patient, taking care of the lines, be always comprehensive in such cases. Try to minimize the risk of like infections. I think there is studies, I didn't mention it because of the time, about the use of epoprostenol and the complication expected. They said about 0.3% of risk of infection and risk of mortality related to the infection compared to all the patients with epoprostenol.
Just to add on that, do you have any numbers like in your center, if we are comparing those on immunosuppressive versus non-immunosuppressive, the rate of infection in both groups?
I'm not aware about this.
Okay, thanks.
I would like to make an announcement.
Yes.
If you are when you are done.
We are at the end. We're reaching the end of our sessions about this crucial, about the management of pulmonary hypertension. Thank you all for attending. Thank you, Dr. Shaya, and all the presenters. Dr. Hassan, yes, please.
Yes. We are about 20 minutes behind in our schedule, so we will take about 10, 15 minutes maximum for the break. Then I just want to remind everyone that our dinner tonight, our gala dinner, is not in the hotel. It's in a venue outside, and therefore we have buses down by the gate outside. So 7:15, between 7 to 7:15, we will everybody should gather by the lobby. Then we walk down to the main gate. You cannot miss it. 3 huge buses, red buses waiting outside. So please, people who are not here and you have a friend who was here, you remind them to. 7:15, between 7 to 7:15, we'll meet, we will walk down to the buses. Buses will move around 7:30. And the venue is not too far. Here's the buses there. And that's the venue. Thank you.
So we'll have 15 minutes break.
Yeah, 15 minutes maximum.
We'll be back at 4.
Yes.
Thank you.
check.
شايف هو نفر هناك هو زي كذا كلام شوي شوي يجي سيجنال يجي صوت this signal is off no input off wait off thing
Check.
this for this okay this full off no signal if off قصدك انه ليش في سيجنال كلام بسم الله الرحمن الرحيم صوتي شغال صح؟ this off no maybe
Check.
turn off turn it off
Check. Check.
خلاص تمام بس غفران اذا حطوا الاسكوب
نبدأ. Shall we start? طيب بسم الله الرحمن الرحيم الصلاة والسلام على سيد المرسلين سيدنا محمد وعلى آله وصحبه أجمعين. We will conclude day 2 by this very important session. We have four presentations which are very challenging cases in BAH. I will share the session Badr Al-Ghamdi from College of Medicine, King Khalid University, Abha, and my colleague Khalid Najashi from Prince Sultan from Medical City from Riyadh. We will start with Dr. Hussam Satija, but before we start, I think we had a notice from the organizing committee that the presentations will be 20 minutes each instead of 30 minutes. So it will be 20 minutes so that we can finish on time. Dr. Hussam Satija is well known in Saph community. He's been participating well with us across the Saph development. He is an adult pulmonologist from King Fahd Medical City in Riyadh, intensivist, and also specialized in sleep medicine. He has actually excellent series of patients with pulmonary hypertension and had a lot of experience in managing the PH patient. So Dr. Hussam is going to present to us RV ICU management in BIH, and we are glad to see you, Dr. Hussam. So please notice the 20 minutes. Go ahead, please.
Thank Saph for inviting me to be part of this year's Saph, and I apologize for not being able to be with you physically in Jeddah, but I thank the organizers for making this happen virtually. So we're going to go ahead and start non-traditional case presentations. As you know, the patients can be complicated, but I'll show 3 of my patients who were recently managed in the ICU with various reasons and various outcomes. The first patient is a congenital heart disease lady who is known to have severe pulmonary hypertension on triple therapy, and this is her baseline studies from an echo and from an X-ray perspective. Her last PVR was very high, and actually she was referred and accepted to undergo lung transplantation, but she was on the waiting list. She presents one day with hemoptysis to the emergency room. Her X-ray on the left is her presenting X-ray, and immediately we decided to go for embolization. It's not an uncommon complication of congenital heart disease, and she did require mechanical ventilation and intubation to facilitate the process of her procedure. Unfortunately, she was not immediately extubated. With the complexity of her pH and gas exchange, she had to be in the hospital for about a total of 8 days. The course was a little bit complicated by having a little bit of hypotension requiring vasopressor support, a little bit of delirium at one point, but eventually she was successfully weaned and managed. Her echo during the ICU stay was still as bad as before, but it shows more dilatation in the right ventricle stable and a little bit worsening of the function. This is her X-ray towards the end of her ICU course, and after extubation, she managed successfully to go home. The next patient is a little bit more complicated patient. She is again an older, probably in her mid-40s, known to have severe pulmonary hypertension, on triple therapy. She was referred for pulmonary hypertension, but not enlisted yet, and her disease is somewhat severe. She was somewhat stable, but her presenting symptoms were worsening lower extremity edema and ascites. That turned out to be an early COVID at that time. She later on developed fever and pulmonary infiltrates. Was hospitalized, eventually had to be moved to the ICU with worsening of her gas exchange and her X-ray findings to the point where she developed ARDS, despite the fact that she was very well managed in the floor, but she deteriorated and she required ICU care and mechanical ventilation. As the case with other COVID cases, she did have multi-organ failure, including renal failure and dialysis. She sustained a cardiorespiratory arrest. She was successfully managed early on, but within 48 hours, she arrested again and she passed away. The third case is another young, unfortunate young woman. She's in her 20s, the classical group 1 PAH patients who was advanced and progressing despite the multiple lines of therapy. She was on triple therapy, but she refused IV therapy, and she is already referred for transplantation. She's accepted for transplant. She presented with worsening symptoms. Just there's nothing that was leading to this except the progression of her disease. She was having right-sided pleural effusion as well as ascites and lower extremity edema. She was hospitalized because she was discovered in the clinic to have hypotension, and she did eventually require to have mechanical ventilation. These are echo pictures from her stay in the ICU showing the severe dilatation in the right side and encroachment of the right side to the left side, where she did have a significant comorbidity. She was contacted for urgent transplantation. She was 26 years only, but unfortunately we could not secure that for her. Discussions with the family about the next step, considering the ECMO, and since she was not a candidate for transplant or urgent transplant at that time, we decided against doing an ECMO, and we made her DNR, and eventually she passed away. So the idea here is just to show you different aspects of pulmonary hypertension. As we see more patients, as the patients survive a little bit longer, they are expected to require ICU care, whether for a progression of the disease or for some comorbidities that can, put them in the ICU and require specialized care in the ICU. The natural progression of the disease, most people actually, they die with PH, with progressive right ventricular failure or respiratory failure, and some of them, they die with sudden death. Most of the times now, mortalities do occur in the hospitals, majority of which occur in the ICU settings, and it will always be nice to shed light on this somewhat not very well tackled field in medicine. Anything can really make the patient who is semi-stable or somewhat stable in pulmonary hypertension to deteriorate and require ICU stay. The list is longer than this, definitely, but anything can happen to these patients. And the patients, when they have an ICU-related morbidity or pathology, there's a lot of things that happens in the ICU that can negatively influence their underlying disease and contribute to the overall outcome, from the drugs that we use in the ICU, from sedation, from fluids, from mechanical ventilation. These people are more prone to hypercoagulability, and sometimes they develop sepsis, which has a very negative influence on the heart itself, as well as the hemodynamics. So it's a complex interaction between the disease causing the problems leading to the ICU and the ICU diagnoses contributing and complicating the course of the pulmonary hypertension. This is a historical study that looked into 99 cases of patients who were admitted to the ICU, and as more severe, Apache 2 is reflective of severity of illness. The more severe the disease is, the more likely that the patient will not survive. And as you see it clearly in the graph, they try to identify certain parameters that can contribute to the negative outcome. It was noticed that dialysis, vasopressor, and mechanical ventilation need is these three parameters were statistically significant negative outcomes on the patients, which probably a lot of our patients in the ICU require these three interventions. This is another interesting study that's somewhat old, even before the advancement of pulmonary hypertension, looking into people who, multi-center from Europe and North America, looking into the people who required intensive care management, and specifically looking into CPR. There were 132 patients who underwent CPR only 8 patients of whom actually left the hospital without a major significant neurological deficit. So a cardiopulmonary arrest is almost a death sentence for these patients. And when you look into the 8 patients who survived out of the group, you will notice the time interval into their arrest was very short. So, and it happened in a very well controlled environment, either in the cath lab or in the hospital. And that tells you, like, this is not real life. Most of the times they arrest when they were not attended, and the outcome is usually very poor. A more recent study published last year looking into a single center from England, this is Aspire registry. They looked into 242 critical care admissions, and they looked into the longer term survival from the beginning up until 8 years of post incident, and they managed to stratify the patients into medical, surgical, and obstetrics. The majority of the cases were medical, as you can see, and the medical reflects the blue line. Most of them, they have a very bad outcome early and long term relative to the surgical and obstetrics, and the data is very clearly showing that. And if we are interested in the ICU management, you look into the mortality from the ICU. There were 157 cases, 72% had an ICU survival, and only 59% had hospital survival, meaning that about 40% of them, they will die before they go home in that acute hospitalization. Unlike the obstetric and the surgical cases, they are less, and most of them were done actually in an elective fashion, not being having to have an emergency surgeries the majority of the time. The same study looked into the factors that, the characteristics of the patient. Majority were idiopathic in nature, about 2/3 of them. They were functional class 3 at large, and their hemodynamics are somewhat severe with mean PA pressure of 47 and cardiac index of 2.6. They looked into the factors that contributed to the outcome, and they came up with this OPALS score, looking into parameters similar to the Apache, but they looked into the oxygenation, looked into the platelet count, looked into the age of the patient, looked into lactic acid and sodium levels. And the more numbers of abnormalities, as mentioned in this article, the more likelihood of mortality. And if they have the 5 of them, the mortality rate can reach up to 100% of the time when they require admission. So to understand the physiology and the link physiology and the pathology, I would like to go through the basics of the right ventricular function in general. Pulmonary hypertension circulation is under a negative pressure or low pressure system, and this is required in order for the right ventricle to be able to function. Right ventricle is unlike the left ventricle. There is a major differences in the myocyte structure and the ability to tolerate pressure, and it is just a conduit for the blood to reach into the pulmonary circulation so that the gas exchange will take place, and then it will be returned to the left ventricle. So the differences are very well established. What happens during, this is physiologically as well, notice the pressure volume loop for the right ventricle is different than the left ventricle. There is no isometric contractions the minute that the ventricle starts filling, and the minute even before contracting, the ventricle will start emptying because of the low pressure system down the stream, and as well as when the ventricle starts relaxing, immediately the system will start filling again. So this is the normal physiology of the ventricle. When we start dealing with pulmonary hypertension, with the understanding of the pathobiology of the disease, when the pressure overload, there will be fibrosis, capillary loss, growth arrest, cellular and metabolic changes that takes place. This will lead to increase in the PVR eventually, and the right ventricle will have to deal with a higher pressure, which is unaccustomed to, and this of course will lead to changes in the right ventricle. And it's very well described that this increase in the afterload, which is the amount of strain that the right ventricle has to pump blood against, will be depending on what will happen to the ventricle. Either it will be adaptive or maladaptive. The adaptive, there will be constrictive hypertrophy, there will be a good amount of functionality retained, minimal fibrosis, the ejection fraction is somewhat maintained, there is a good coupling between the ventricle and the pulmonary artery, and usually the patient will be clinically stable with minimal side effects, unlike the maladaptive, where the patient will have some right ventricular failure that is clinically present in the cases. And this is the picture of the what happens in the right ventricle. Unlike the thin membranous right ventricle, normally it will become hypertrophied, and the upper top on the right side here, the ventricle is adaptive and it has some constrictive functionality. The muscular increase in the wall will compensate for the increase in the pressure, and the patient will be somewhat stable, at least clinically, unlike the maladaptive right ventricle, where the coupling will not happen. function. And of course, this will be influenced by the volume of the status. And this is very relevant in the ICU, as the patient will have volume issues at large, whether less volume or more volume. Intensivists, emergency room physicians tend to give fluids as the first knee-jerk reflex to anything, and that can have a lot of consequences. And of course, the pressure volume loop will change. In the middle, the green button that will be showing you the adaptive ventricle when it is adaptive. The changes are very obvious. It looks similar to the left ventricle, but there will be maintenance of the elastance of the ventricle to the pulmonary, the ratio between the end systolic ventricular elastance and the pulmonary artery capacitance or elastance as well. And that ratio will maintain adaptability of the system and the ability to the right ventricle to perform its function. And anything that will tip this off, whether it is precipitated by another illness or volume or ischemia, that will lead to maladaptive physiology. And in maladaptive physiology, the ventricle will dilate more, and the ratio between elastance and the end systolic right ventricular elastance and the pulmonary artery elastance will be tipped off, and the patient will start having clinical failure, and it will be obvious. And of course, this will be associated with consequences hemodynamically on the patient in the ICU. If you look here, we are interested in the ability of the system to overcome the PVR. Usually the PVR is somewhat fixed in the pulmonary hypertension, and if anything, it will be increased by the changes that we mentioned and the underlying pathology. It's somewhat difficult to really control it that much unless we are doing treatment to specifically decrease the PVR. The patients are usually on double and triple therapies, but that definitely can be escalated to, I will talk about it a little bit later on. And sometimes we'll have to support the ventricle with inotropic support in order to improve the the contractility of the right ventricle in order to help overcome the consequences of this. One other concept that is very relevant in our ICU patients is the ventricular interdependence. The right ventricle ejection fraction is somewhat dependent on the septum of the left ventricle. The septum contractility will contribute a lot to the function of the right ventricle and its ability to empty its blood and the cardiac output. So any changes in what happens in pulmonary hypertension, this is normally the left ventricle is the dominant one. The right ventricle during diastole will be really pushed, and during systole it somewhat dilates because of the volume. But what happens in the maladaptive state or in the pulmonary hypertension state, that the septum will be moving to the other direction, and that can have two consequences. The right ventricular dilatation will influence the right ventricular contractility and function, and at the same time the encroachment of the septum into the left ventricle will definitely have consequences related to that. The fluid status of the patient is very serious in the ICU, and look at the differences when the patient with pulmonary hypertension have more fluids, meaning that this relationship will be more tipped towards the left ventricle with all its consequences from increasing the end diastolic pressure, increasing the tricuspid regurg, septum shift, left ventricular filling will decrease, and definitely the cardiac output will also decrease. Unlike the time when the patient fluid managed very well, still the consequences are somewhat milder and manageable. One other concept that, the blood supply to the right ventricle is different than the left ventricle. So the more pressure the right ventricle has to deal with, the more ischemia the right ventricle can sustain. And this is an ECG of a patient who is definitely not normal, known to have pulmonary hypertension with some ischemic changes. But after ICU admission, the changes in ischemia were more apparent in the form of the florid right bundle branch, and that was an indicator of right ventricular ischemia in this patient. So if you put the whole thing together between the dilatation and embedment on the left ventricle, there will be decrease in the left ventricular filling output, decrease in the supply to the compromised coronary arteries. There will be right ventricular ischemia. There will be decrease in the right ventricular output, and we will get into the vicious cycle. That's what happens to the patients when they are in the ICU, regardless of their underlying pathology, unless we are really vigilant and trying to avoid this from happening. When the cardiac output becomes less, especially in the ICU, the perfusion of the organs, specifically the gut, becomes compromised, and the patients are more prone to developing sepsis, unfortunately. And of course, sepsis by itself is a bad disease. It's definitely a killer in patients who have preload dependent states when their systemic vasodilatation, and that can lead to their mortality, which we see commonly in patients with pulmonary hypertension. Reminder about the mechanical ventilation. Patients in the ICU are commonly mechanically ventilated. Mechanical ventilation will change the intrapleural pressure from negative into positive. The more positive the intrapleural pressure, the more influence that will have on the preload and the afterload, and consequently on the function of the right ventricle. On the left-hand side, the graph shows that the PVR will be changing according to what level of ventilation we are trying. The best thing is to try to ventilate the patient at their FRC, which is sometimes difficult to determine, but we have to avoid extremes of volumes. So low tidal volumes are definitely required. And of course, in the ICU, this is not an easy to obtain because that might be associated with hypercapnia. And at the same time, we don't want to give too little volume because that will even contribute to the increase in the right ventricular or pulmonary vascular resistance. Reminder, in the ventilator, we always say 90-88% saturation is good. For PH patients, we need higher. We do need to give them, be liberal with oxygenation. PEEP has a negative effect on the preload and afterload. Hypercapnia, both of them, they have negative consequences on the cardiac output, and we try to optimize that. It's not an easy task, but it's something that we have to be aware of. Of course, we mentioned the fluid management, and we are always bothered by the fact, do I give fluids, or do I give small amount of fluid, or do I try to avoid the fluids altogether? Hypotension is a serious consequence for these patients, and normally we give fluids, fill the tank first, and then try to vasopressors. In this case, in pulmonary hypertension patients, probably we should more aggressively start inotropic support earlier than the usual, because we are trying to avoid too much volume, and at the same time we are trying to avoid the negative consequences of the hypoperfusion to the rest of the organs. We have multiple agents that we use. The ones in the green are the most likely studied, and we use them a lot. Try to avoid, and this table probably makes it clear, try to avoid the things that can increase the PVR. All of them, probably almost all of them, will increase the cardiac output, yet some of them are tachyarrhythmic in nature, and we are trying to avoid them. Hypotension is another thing that we sometimes face, so we'll have to be careful and weigh the risks and benefit of doing some of them for these patients. Arrhythmias are not very well tolerated. We cannot wait long. Aggressive treatment of arrhythmias is a must in patients with pulmonary hypertension in the ICU. Lots of time we have to give antiarrhythmic agents or even DC shock them out of it, rather than waiting for the effect of some medications to work. And of course, remember, beta blockers are not very welcome. Digitalis for AFib is not something that has a very clinical value.
2 minutes,
what we know
Dr.
to do,
Hassan.
yeah, I'm almost there. Pulmonary vasodilators, we are very well familiar with them. We tend to use them. We tend to upgrade the patients to these therapies. One thing to be aware of, that you have to do it correctly. You have to use the proper machine so that you will get the best out of the pharmacodynamics of the agents, and get the best out of it. Last thing we consider is the ECMO. ECMO services are very available in the Saudi Arabia. Of course, VV ECMO, what we use for COVID, is not for PH patients, and sometimes actually VV ECMO can overload the right ventricle and can precipitate right ventricular failure. So it is the VA ECMO, and again, it's not an easy decision to do. We have to be thinking before we start ECMO, similar to our patients, are we doing transplantation or just a bridge to recovery if we believe the underlying mechanism is really reversible and we can go back into a very stable, good baseline, which is not easy to do. Literature is somewhat, there are some literature on this showing some benefits out of it, and small case series, I won't pay too much attention for it, but it definitely can work. So in summary, this is what we need to be doing. We have to treat the underlying causes for the deterioration, optimize the fluid management, reduce the right ventricular afterload as much as we can, utilize medications properly, optimize the cardiac output, again with dobutamine and other vasopressor or inotropic support agents, and optimize the pressures in the system altogether. Once all these things fail, you have to really be very honest and aggressive in terms of end-of-life care for these patients. One last note, be careful. This came through the COVID time, where patients who have severe ARDS from COVID, an echo, if it's done, it will show similar picture to this. This is a real-life other case with COVID and right ventricular dilatation. Maybe there will be a consultation to the PH service. Be aware that this is something that we do not want to treat. It's a different ball game, and recently they are talking about NO therapy for COVID patients, none of which is really efficacious, and it's just a contribution to the negative outcome of these patients if the right ventricle will not be adaptable and able to manage or to cope with the ARDS that that we have. It's a difficult field. You can build your experience. You need some years of experience and seeing more patients. But every time you come across some difficult, rare case, I think the thing is to be wise in the way you approach them and try to get the best out of it. Thank you.
Thank you, Dr. Satija, for this elegant, and thank you for keeping really 20 minutes time. I think because Dr. Satija is with us online, we will take the questions now. So if you have any questions or comment to Dr. Hussam Satija from Riyadh, please go ahead. We have a question here. Yes, go ahead.
Assalamu alaikum, Dr. Khaldoon من الكويت. I'm a heart failure cardiologist, and what his son said is quite, just clarifies for pulmonary arterial hypertension, we quite find the opposite when it comes to group to the heart failure related. And when we put in LVAD and we suffer from the whole conundrum of RV dysfunction after left ventricular implantation, well, in we see that the diuresis, the reducing pulmonary vascular resistance is a strategy. Inhaled nitrates with milrinone and between these are actually our main tools that we use in this case. And from what I understood, if this patient, because this patient in pulmonary arterial hypertension suffering from an RV is a totally different game. So for my education about this is because when I think about it, I deal with the RV failure secondary to LVAD implantation. We use milrinone, we use inhaled nitrates, and we try to reduce peripheral vascular resistance to help the RV. But from what I understood, it is an opposite when it comes to pulmonary arterial hypertension. Could you clarify this for me, please?
It's somewhat, thank you for the question. It's a different patient population. When you deal with a complication secondary to your intervention, the cause of the right ventricular failure will be a little bit different. In these patients who have pulmonary hypertension, their PVR is high. So any strategy that will reduce the PVR, that will be very much welcome. In your case, probably the ventricle is unable to deal with the load of the volume that has flooded the right-sided circulation and the consequences of that. So a little bit of mechanistically, maybe a little bit different. In PAH deterioration, we do use sometimes milrinone, but be careful because these patients are ill. They are not using them just as a primary strategy. They have other diseases that might be leading to some volume, marginal volume status, or a little bit of hypotension. And milrinone can cause them to have more hypotension. This will lead to more ischemia, and this will even contribute, it will counteract the benefit that you get from the PVR reduction, from the ischemia that takes place in the right ventricle, and the systemic hypotension that takes place. So again, it's somewhat related concepts, yet they are different in the cause. What are you trying to achieve at the end of the line?
Okay, so we'll take a second question there. Yes.
Thank you, Dr. Hussam. One question: patients on IV prostacyclins who's coming for like septic shock, and sometimes there is conflict between the primary team and the ICU team. They want to de-escalate or sometimes stop the IV to avoid the hypotension. What's your comments on that, on approaching patients with the IV prostacyclin?
Make the call. Withdrawal of IV prostacyclin is not simple. We have patients, we reported a couple of cases long time ago that they committed suicide by rapid turn off of their prostacyclin because of the short half-life. So again, you have to weigh the risks and benefits. Maybe decreasing the dose a little bit might help the blood pressure, but definitely the underlying cause has to be managed. We have to really source control the source of the sepsis and infection and be liberal with using inotropic support. These patients, they do need vasopressors and they do need inotropic support. Norepinephrine is good, sometimes it's not enough. So be liberal in giving the systemic agents that increase the systemic vascular resistance, and preferably if they have a good positive inotropic effect, that will be welcome. Even dobutamine, sometimes we cannot use it in real life practice, although it is the recommended one because it can be associated with hypotension. So it has to be staged and you have to be very patient. Every action in the ICU has sometimes negative consequences. It's never clean cut, and it is always the risk-benefit ratio and the the response of the patient to our immediate intervention. And you have to be very open-minded in making adjustments and readjustments after the adjustments.
Okay.
So I think that's the whole essence, but you should be the one who calls for like the prostacyclin therapy, not the ICU.
طيب, we will have two last questions. I think we will start from here. Go ahead, please.
Thank you very much, David Jenkins from Cambridge, UK. I'm a cardiac surgeon and I do pulmonary endarterectomy and transplantation. And I'd like to completely agree with the speaker about the importance of vasoconstriction in the most severe PH and RV failure, because it's keeping the systemic blood pressure up that keeps these patients alive. And that's why the inodilators that we're used to using in left heart failure really don't help in these patients with severe PH and can actually lead to a real deterioration. So it's a really critical point that our colleague made, who's a heart failure cardiologist, that you think an inodilator will work well because it may reduce pulmonary hypertension, but actually in practice they don't work well at all, and the most severe patients with RV failure need vasoconstriction, not inodilators. And that's a key point in the ICU, which I think my colleague there has realized.
Okay, we'll take in comment. Dr. Hussam, you will respond to both. So let's state the last question there, and then we will give you the conclusion. Go ahead, please.
Thank you, Dr. Hussam. I would like to ask about the critical condition of this pulmonary arterial hypertension patient. I'm Dr. Isam Sabri from Pulmonary Hypertension Unit in Kuwait. Now, this patient in ICU has two problems. First, the right ventricular preload is usually diminished in this patient. Increasing the right ventricular preload by IV fluid will have a deleterious defect due to interventricular dependence and increase the D-shaped movement of the interventricular septum. And reducing the pulmonary vascular resistance by our treatment to the pulmonary arterial hypertension will lead also to the decrease in the systemic vascular resistance. And this patient is usually hypotensive. So the management itself has a dilemma. The patient is hypotensive. You cannot increase the left ventricular preload because actually this will be deleterious in the right side. And at the same time, you cannot use potent vasodilator because they will reduce the blood pressure, leading to hypotension and affecting organ perfusion. Thank you very much, sir.
Thank you.
Go ahead, Dr. Bida. You want me to answer? So I'll answer. I think you just summarized the complexity of the situation that we are dealing with. There are prioritization in our management. I tend to use inotropes and vasopressors liberally in these patients because they do need them. Try to avoid the arrhythmic ones, try to avoid large doses if you can, but sometimes they do need that. And I use this even to support my continuation of the somewhat specific pulmonary vasodilators. So that's why generalized vasodilators don't work. You can use selectivity, will be lost with those, but if you give a little bit of selective pulmonary vasodilators and a little bit of vasopressors, I think the overall will be okay. Now, again, this is not something that you do and you go home to reassess next day. This is a very dynamic process that has to be very well monitored. Different response of the patients will behave differently. Lots of times we have to be very aggressive in dealing with peripheral edema through dialysis, and that works much better than Lasix that will have like much more probably complicated consequences. So sometimes we have to do hemofiltration to deal with the volume that we are dealing with. We understand the intravascular depletion, and at the same time we understand the consequences of the volume overload. I think this is just part of the complexity of the management of these patients, and we have to take it case by case, and we have to look at it wisely with the basic understanding of what we are trying to achieve.
Thank you very much, Dr. Hussam Sakija from Riyadh, King Fahd Medical City. As it was mentioned, it's a complex situation. It really a lot of light has been put on by the presentation. So I think we will move to the next presentation with Dr. Khalid.
Assalamu alaikum. Good afternoon. Thank you for staying to late this Friday. So it's my pleasure to introduce Prof. Yuri Serenko from Kiev, Ukraine. He's a professor and head of department, National Scientific Center and the Institute of Cardiology, Kiev, Ukraine. His talk will be an intermixture of cases of pulmonary hypertension. Welcome.
Distinguished chairpersons, dear guests, dear participants, dear colleagues. First of all, I would like to express thanks to the organizing committee to invite me for this excellent meeting, and personally my friend Abdullah Dalan for this hospitality. And it isn't easy to present just clinical case after this comprehensive tutorial lectures before, because we will talk about just one case, and I'll try anyway. I would like to start with this picture. Since invention in 1998, the classification of pulmonary hypertension according to the etiology, the growing numbers of registers and growing numbers of observed cases show us that there are enough patients when, according to their syndromes, can be attributed to the different groups simultaneously. So now we are talking about overlapping and sometimes convergence of the groups. Nothing happened. Okay. Maybe it doesn't mean something. And it's very important in the case when we have to choose prescribe or do not prescribe specific therapy. And according to our current knowledge, the patient phenotype should be close to the group 1 for the prescription of the specific therapy. Here is two current international document which we use for our selection of the diagnostic or treatment procedure. Next year we expected the new European guidelines. Maybe it could be a little bit better, but there is a definition, and I would like to start with the group 5. It's very interesting title of this group: hypertension with unclear and multifactorial mechanisms. And we will talk about patients with myeloproliferative disorders. And in this case, we don't know about real mechanism of development of pulmonary hypertension. And second, we don't know if specific therapy will be effective or not. When we look at the factors for pulmonary hypertension development and contribution in the group 5, the pulmonary vascular lesion and spasm, proliferative vasculopathy or vascular obliteration, should be change our mind to indication for specific therapy. From the other hand, venous component, left heart failure, and severe left heart failure are contraindication for specific therapy. So let's start our patient. 31 years old white man admitted to our hospital with severe symptoms of right heart failure. Except dyspnea at the rest and the minimal physical activities and leg edemas and increased abdominal volume, patient had a severe general weakness, episode of syncope, diarrhea up to 20 times per day, but the stool was a little consistent, and other dry cough, palpitation, heaviness, or feeling pressure in the chest, repeat fatigue, and sweating. Medical history. At the age of 20, patient underwent, it was the first disease at the age of 20, underwent surgery for phlegmon of lower extremity for the first time. Since then, low numbers of platelets and leukocytes have been constantly observed in blood analysis. He had multiple pneumonia up to 4-5 times per year, and 2 years before admission to our clinic, according to the radiography, CT, pulmonologist suspected lung sarcoidosis, and later diagnosis was confirmed in a specialized center in Israel. But after that, they abandoned this diagnosis. In years 2019, hematologists performed free diagnostic bone marrow puncture without clear result. In June 2019, on the background of next pneumonia, appeared edema of the lower extremities, hydropericardium, and ascites. In January 2020, just before 5 months before admission to our clinic, immunologist diagnosis primary deficiency of immune regulation and confirmed it with the genetic tests. Sirolimus was prescribed for first time. The condition improved for short period, and symptoms of the heart failure increased, and patient admitted to our hospital. I would like just to remind you what is diseased. Immunodeficiency due to CD25 deficiency is a rare genetic primary defect with profound susceptibility to viral, fungal, and bacterial infection due to T regulatory cell function malfunction. And those patients also can have autoimmune disease and enteropathy, like in this case. And majority of those patients died before 20. And we have patient 31 years old. Medical examination. Body temperature was normal. Oxygen saturation 90 without oxygen. Peripheral cyanosis, distinguished jugular veins, respiratory rate 24 per minute. Lung auscultation weakened in the lower part, wet rails, crepitation in lower part of the both lung, and sometimes peripheral friction noise. Heart rate 119, blood pressure 100/60. Bandaras of heart are expanded in all the direction. Heart auscultation sound free. Severe pansystolic noise over tricuspid valve, split sound 2 over pulmonary artery. Enlarged abdomen, at least 6 centimeters plus liver, painful on palpation, and palpated enlarged spleen. Lymphedema with trophic changes of the skin. And the patient can't perform 6-minute test, and we indicate less than 100, naturally. Laboratory tests. General blood analysis, nothing interesting except a little decrease of hemoglobin and low numbers of platelets. You can see here 107. In urine, proteinuria 1.3 gram per liter. Biochemistry, low ferrum 7, low albumin 38. Slightly elevated AST and LT, 68 and 70, normal LP, GGT, and slightly elevated bilirubin, 34. Yeah, NT-proBNP, huge elevated, more than 6,000. Procalcitonin is low. There is no signs of infection, and rheumatological test, slight elevated C-reactive protein. ECG, You can see P pulmonale, you can see right bunch block and signs of right ventricular strain. Here is chest X-ray. I'm cardiologist and I refrain from the comments because you know what is a better. But here diffuse fibrotic changes and increased radix of pulmonary. Here is echo. Short axis you can see. I try to show. You can see free liquid. Suppressed, short axis suppressed left ventricular with a severe flattering of the septum and a huge enlarged right ventricle. We tried to perform 4 chambers position, however, it wasn't possible and we did it how we can. And you can see also a lot of liquid, a huge right atrium, huge right ventricle, signs of tricuspid regurgitation and severe severe suppressed right ventricle and small right atrium. Echo parameters from the guidelines that allow us to suspect probability of pulmonary hypertension show following. Now our patient has everything that we need. Peak tricuspid regurgitation velocity at 3.9, decreased right ventricular contractility, TAPS only 14 millimeters, and all other signs for high probability of pulmonary hypertension were present. There is a spirometry that you can see severe. I also refrain from detailed comment. However, you can see severe restriction disorders and also decrease lung permeability. Here is abdomen CT. You can see huge liver and huge spleen that filled abdomen cavity, suppressed and also, I guess, pressured both kidney. Here is a CT of chest without contrast, also diffuse fibrotic changes, enlarged nodules, lymphatic nodules, and here is CT with contrast. Most of all important for us was the absence of pulmonary artery obstructions. You can see free fluid surround heart, and you can see also the same picture at the echo, huge right heart and small left heart suppressed by this. Right heart catheterization. Mean pulmonary artery 58, low cardiac index 1.84, right atrium pressure 20, pulmonary artery wedge pressure 9, and venous saturation 52. And pulmonary vascular resistance almost 14 Wood units. It's clear. First, precapillary pulmonary hypertension. So diagnosis of our patient, pulmonary hypertension due to the lung disease and associated with myeloproliferative disorders. Third and fifth group, syncope, World Health Organization functional class 3, 4 at admission free after stabilization of situation, hydropericardia, myositis, right heart, congestive heart failure, and the main diagnosis, primary immunodeficiency due to CD25 deficiency. And other diseases that at the moment are not so important. So what we have to do with this patient? According to hemodynamic phenotype, this patient belongs to the first group, and we have to start specific therapy. And considering severe condition, we have to start from intravenous prostacyclin. Diuretic, oxygen, and treatment of heart failure symptoms. From the other side, if patient belong to the third or fifth group, therapy of basic disease. For the group 3, specific therapy is contraindicated. For the group 5, therapy is not defined at the moment. There is no any evidence data. So we started with a very, from the current point of view, very dangerous procedure. However, despite of the low numbers of platelets, we started with pericardiocentesis. And we evacuate 800 milliliters of transudate. We expected for improvement. However, situation worsened. And I guess this liquid preserve right ventricle, totally suppress left ventricle. We receive severe hypertension, and we started even for short period infusion of catecholamine, dopamine. Oxygen therapy and intravenously continuous infusion of furosemide, and we started iloprost. In our country, we have licensed only this prostacyclin compound for 5 days, and then when situation stabilized, we followed with inhalation for from 6 to 9 times per day. We tried sildenafil several times, but noted worsening of patient, and we have to withdraw it. We can prescribe endothelin receptor blocker because hepatic disease, severe hepatic disease. So we have to use just only one compound from available. We added eplerenone, sirolimus, and we also added colchicine because there is a huge, not huge, but very nice performed study that showed that colchicine can decrease possible liquid accumulation in the pericardium. After 3 months, we see dramatic clinical improvement, no symptoms without exertion, no peripheral edema. Were evaluated as a functional class 2, reduction of spleen and hepatic, oxygen saturation 94, 6-minute walk test 350, natriuretic peptide, NT-proBNP decreased from, you remember, more than last 2 slides, yeah? Decreased echo, improved, and we decided to refrain from this repeated catheterization at the moment. We have we decided to wait for a little short periods for 6 months, and after that perform right heart catheterization. Unfortunately, patient come to our hospital from the other city, and when he returned back, he got COVID infection, severe pneumonia with respiratory support, and the patient died in other city, and we don't have at the moment pictures of autopsy. Particulars of this case. First of all, long-term survival of patient with immunodeficiency, because as I told, majority of those patients died before 20. And this patient just started all symptoms after 20. Intermixture of etiology of this pulmonary hypertension, lung disease, myeloproliferative disease, and situation when patient with a group syndrome, group 3 and group 5, has severe precapillary pulmonary hypertension that is most attributed to the group 1. And unexpected effectiveness on specific prostacyclin monotherapy in manifest patient with a third group of pulmonary hypertension. And I would like to show the last slide. This is our team. Thanks to my all colleagues for supporting me in this case. Thank you very much for your attention.
Thank you, Prof. For the sake of the time, I think you join us here. We'll leave the question till the end of the session. So we'll come here.
Okay.
So we will move to the third presentation, which will be by Dr. Nawal Kubesi. He is from the Eastern Province, Dhahran, Trained Fahad Military Complex. She is well known to Saph with her interest in pulmonary hypertension, and she has been also handling a good number of patients in that area. She is going to share with us a patient here with a title of Medical History Really Matters. So we are glad to have you here. 20 minutes, Nawal.
I'll try. Thank you for staying till now. Thank you for the introduction. The reason I picked this case was it imposed a diagnostic challenge for me as a pulmonologist. I learned from it, and I'm here not only to share the knowledge I acquired, but to exchange the knowledge with the audience of different subspecialties and experience. And probably it's a topic that we didn't tackle much during this conference. And I named it Medical History Matters because it does, as we're going to see from the story. So it started with a call from our colleagues in internal medicine referring a case of pulmonary embolism. We asked, well, how sure are you? They said, the diagnosis is confirmed. We have a 23-year-old man. So far, nothing is consistent with pulmonary embolism diagnosis. He presented with only 2 days history of chest pain. And when he arrived to emergency area, walking from the parking lot, initially he was a bit hypoxic, a bit tachycardic, but once he got rested, his vitals normalized. They called the radiologist on call, asking for a CTPA, and the report was a major filling defect in the left main pulmonary artery, in the left pulmonary artery. And we are going to come back to this slide later in the story. When we reviewed the patient, indeed, his history was only of 2 days, shortness of breath at rest and chest tightness. He had no medical history whatsoever. He was not on any medications. He did not have any surgeries, according to him at least, and he did not have any symptoms, respiratory or cardiac symptoms, and no symptoms suggestive of any systematic illness, GI, rheumatological, or hematological illness. He was non-smoker. He was not on any drugs. He was recently graduated, searching for a job. And he had no family history of any cardiac or respiratory illnesses. On physical examination, he was hemodynamically stable. His oxygen saturation by the time we saw him, was normal in room air. He was afebrile. His JVP was normal. Precordial examination was normal, normal heart sounds, and he had no chest deformities, and his chest auscultation revealed normal breath sounds. He had no lower limb edema and no signs of DVT. His initial labs run by our colleagues in internal medicine to rule out any acute coronary syndrome with the chest pain showed normal cardiac enzymes. CK, CKMB, troponin all came normal. Even BMP at that time was normal. His CBC, despite showing a slightly raised hematocrit, but was normal. His liver function and his renal function tests were also within normal limits. His chest X-ray, as far as our colleagues in internal medicine were concerned, showed no consolidation. His costophrenic angles were clear, but there was an increased in the cardiothoracic ratio and a bit of a large right hilum. And again, we will come back to this slide, to this chest X-ray later. His lung parenchyma showed mosaic perfusion or mosaic appearance. Otherwise, nothing was of significant abnormality. So their initial impression was unprovoked acute pulmonary embolism, but we also kept chronic thromboembolic pulmonary hypertension in mind given the radiological findings. Either way, he was started on parenteral anticoagulation and shifted to warfarin with a target INR of 2 to 3. His thrombophilia workup, antithrombin 3, protein C, protein S, lupus anticoagulant, ANA, and hemoglobin electrophoresis, everything came back negative. His Doppler ultrasound, lower limbs also was negative. When we did an echo to check for RV strain, his TRV was 4. And by ESC, ERAS criteria, this indicates a high probability of echo of having pulmonary hypertension. Even his RA was dilated. It's not moving, but anyway, his RA was dilated, RV was dilated, and he had no pericardial effusion. Now, within a couple of days of being on anticoagulation, the patient claimed that he was now free of symptoms. He was insisting on discharge, and but we could not ignore the echo findings, and we definitely asked for a VQ, and we kept him on anticoagulation for full 3 months, and we followed him in OPD. Of course, we followed him regularly for his INR, and we were checking on his symptoms on weekly basis almost because of the anticoagulation he was on, and just to monitor his symptoms. In 3 months he came, and echo findings were almost the same, but this time we even paid attention to some changes in the LV. Despite a normal LV size and function, he had hypokinesia of the inferior and inferolateral walls. It was regional wall movement abnormality. Of course, his RA and RV were dilated, and this time we also appreciated some mild systolic impairment, and his estimated RVSP was more than 100 millimeters mercury with no pericardial effusion. So by this time, we had to take him for cath, and on the day of right heart cath, something happened. His parents showed up. We never saw them in any of the follow-ups. They just came because they were concerned he's going for cardiac procedure. We asked him to wait in the lounge, and we took the patient for the cath. So that's what we found. His mean RA pressure was 6. RV pressure was 121/2. RV-EDP was 9. His wedge pressure was 17. I tried to repeat another set, if you can see, on the 4th row, because probably the 3rd one showed some prominent V wave, but still it gave the same or similar reading. His systolic pulmonary artery pressure was 60, diastolic 12, and mean of 29. His saturation of RA was 69% and of PA was 83%. Wedge was 84%. Now, what is the value now? We have a clear evidence of shunt. So what is the value of indirect fix here to calculate the cardiac output? But it was our only mean to measure the cardiac output or to estimate it. Probably it was overestimated. Cardiac output was estimated to be 9, and PVR was probably underestimated to be 1.2. But with that wedge pressure, we diagnosed the patient hemodynamically to have a picture of post-capillary pulmonary hypertension with a clear evidence of possibly a shunt. By this time, the VQ report came back saying the patient has complete absence of left lung perfusion, and this is not typical of CTEPH. His 6-minute walk distance was 361 meters, 54% of his predicted, and he did saturate despite being asymptomatic. He dropped his oxygen saturation during 6-minute walk test from 94% to 80%, and this time his BMP was increasing. So the parents showed up with a report saying that this patient at the age of 2 had a right ventriculectomy, main pulmonary artery was connected to RV and open to the hilum of left pulmonary artery. VSD was closed with Dacron patch, and his pulmonary artery and RVOT all enlarged with bovine pericardium, and he had a BT shunt or systematic to pulmonary shunt. So probably this patient had a tetralogy of Fallot with VSD, pulmonary stenosis, hypertrophy of the RV. And retrospectively, we went back to the imagings. If you see there is left lung hypoplasia, or the left lung volume is clearly smaller than the right one, with an increase or enlargement of the right hilum. And probably the fulminant or filling of the retrosternal space indicating an RV enlargement. The RV in the echo shows thickened wall indicating the chronicity of the condition, and probably an expert eye can even appreciate the membrane that repaired the VSD. And interestingly, when we looked back in the records, we noticed that his D-dimer initially was negative. Now, I wondered why his RV systolic pressure was 120 or 100 estimated in echo, while his systolic pulmonary artery pressure measured was only 60. And some of the explanations I found that this may be due to RV outflow tract obstruction because of pulmonary artery stenosis, or sometimes erroneously there is a measurement of VSD instead of TRV by echo Doppler. But this is not the case in our patient because he had a right heart cath and he definitely had a pulmonary hypertension. Now, where to categorize this patient that has a congenital heart disease, post-defect repair, with a hemodynamic picture of post-capillary pulmonary hypertension? Now, congenital heart diseases fall into one of two groups, either pulmonary arterial hypertension, group 1, and then they are subclassified into A, Eisenmenger syndrome, where there is a right to left shunt, and these patients are usually chronically cyanotic and hypoxic, and they have a multisystematic effect of the disease, and the repair of the defect has no role in this case. B is pulmonary arterial hypertension with persistent systematic to pulmonary shunt that is left to right shunt, and these patients may benefit later from repairing the defect, despite nowadays it's less and less favorable with the presence of target therapy and the complications of the closure of these defects. And usually these patients are not cyanotic. And then class C, pulmonary arterial hypertension with small or coincidental defects. These patients present like idiopathic pulmonary arterial hypertension. Sometimes in the screening, we find out that they have small defect, PFO, or small congenital defect that was not repaired, that has no significant hemodynamic effect, and these patients usually are not candidates for repair. Then D, pulmonary arterial hypertension after defect closure. These are patients who were born with congenital heart disease that was repaired, and they persisted to have pulmonary hypertension, or they developed it later in life. Usually there is no shunt and no cyanosis, and the presentation is similar to idiopathic pulmonary arterial hypertension. And these patients with group 1 have the histopathological changes of the pulmonary artery, probably due to the high flow inducing the endothelial damage of the pulmonary artery. Then group 2, congenital heart diseases that fall into group 2 or post-capillary, such as congenital left heart inflow obstruction, like pulmonary vein obstruction, left atrial obstruction, or LV inflow obstruction, like mitral stenosis and hypoplasia. Left heart outflow obstruction, aortic valve obstruction, whether on supravalvular portion or subvalvular or valvular portion, and LV hypoplasia. Congenital heart diseases can present with pressure overload, such as aortic stenosis, coarctation of aorta, interrupted aortic arch, or pulmonary stenosis, and pulmonary vein stenosis, or volume overload, like cases of left to right shunt, aortic regurgitation, mitral regurgitation, or Epstein anomaly, or both volume and pressure overload, like hypoplastic left heart disease or congenital heart disease with single ventricle physiology, or ischemia, like our patient. I wondered why he had those regional wall movement abnormalities. Now, probable causes, congenital causes, are coronary anomalies and perioperative injury. So we decided to categorize him in group 2, given the post-capillary hemodynamic picture, and given his functional class BMP 6-minute walk test, we risk stratified him as in intermediate risk. So to treat or not to treat. The moment I had this case, I gave a call to one of our colleagues in pediatric cardiology in the hospital. He originally had the surgery, and though unfortunately his records were lost, to discuss what are the therapeutic options. Now, ESC ARS mentioned bosentan as favorable ERA or favorable target therapy for patients with Eisenmenger or complex congenital heart disease. And this was proven by literature to improve New York Heart Association and functional class and 6-minute walk distance test. But because of the post-capillary hemodynamic picture of this patient, we just decided not to put him on target therapy. ERS, ESC recommend classification or class 2B recommendation to put these patients who need on supplementally oxygen. So this patient had ambulatory hypoxemia, therefore he was put on ambulatory oxygen. Anticoagulation was continued, and he was sent for a redo of pulmonary artery dilation in the first referring hospital. Anticoagulation, I opted to continue because these patients with pulmonary atresia or pulmonary stenosis are at risk of still developing thrombosis, so still probably he had really PE. So congenital heart diseases at infancy or early childhood can present in adulthood with persistent or newly developed pulmonary hypertension, and therefore we should always keep in the background of our mind this fact while assessing our patients and trying to dig in the medical history for their childhood and infancy medical history. Thank you so much.
Thank you, Dr. Nawal Rubaisi, for sharing this very interesting case with us. I'm sure there will be some comments and discussion about it. We will leave it at the end. We will proceed with the next speaker.
Yes, so it's my pleasure to introduce Dr. Faiz Al-Ahmadi, consultant pulmonary medicine, pulmonary hypertension, and balloon pulmonary angioplasty from University British Columbia. He's working now in King Faisal Specialist Hospital Research Center, Riyadh. Welcome, Dr.
Introduction. Good afternoon. I know it's the end of the day, but I can assure you that I have really interesting case for you. And simply, I have a patient with only cough. But I need your help to reach a diagnosis. So my patient is a 53-year-old gentleman who was seen first in the clinic as a case of chronic dry cough for a duration of 6 weeks. The patient had suffered from this chronic dry cough for 6 weeks duration that was initially non-progressive and intermittent, no specific diurnal variation, not affected by position, no difference between weekdays and weekends, no aggravating or relieving factors, no hemoptysis, minimal sputum production, and it occurs more during the daytime rather than morning or night. He reported some mild exertional dyspnea after his diagnosis of unstable angina back in April 2021. However, overall, he was able to walk 3 kilometers and take multiple flights of stairs. He got a history of TB exposure. His maid had an active pulmonary TB that was later treated. His BBD was positive. However, he didn't take any prophylaxis. Two AFBs in 2017 and 19 were negative, and 3 of his sons had positive BBD and took prophylaxis. So the history of presenting illness started in 2021. He reported night sweats for 8 months and unintentional weight loss of 4 kgs since his diagnosis of unstable angina. No fever, no syncopal attacks, no lower limb edema. He's not in ACE inhibitors, no symptoms suggestive of GERD, upper respiratory tract infections, or postnasal drip, and other systemic review were unremarkable. So as a past medical history, he had ischemic heart disease, underwent PCI to RCA, April 2021. He had dyslipidemia. He's on aspirin, ticagrelol, omeprazole, atorvastatin, ezetimibe, bisoprolol, and he's not known to have any allergies. So he denied any pets contact at home. He's working as a in housing management, an office job. He's ex-smoker with a 10-pack-year smoking history, and he stopped at the age of 42. No family negative for family history aside from his mother that had chronic lymphocytic leukemia, no history of A to B. The interesting thing that he took a COVID-19 vaccine, AstraZeneca, 2 weeks prior to the beginning of his cough. So during the clinic visit, basically he was vitally stable. His oxygen saturation 95. ENT exam unremarkable. Cardiovascular exam was normal. normal first and second heart sound, GVB is normal, unremarkable respiratory and gastrointestinal exam. So just to summarize, we have a 53-year-old gentleman who presented with chronic dry cough, weight loss, night sweats with no other significant symptoms. His routine and baseline investigation showed normal CBC with differential, normal renal and hepatic profile. His quantiferon was negative. Inflammatory markers were negative. And this is his X-ray, which showed basically clear lungs with, I would say, mild interstitial thickening, however, no pleural effusion. His PFT was reassuring with only mild obstructive pattern, a normal total lung capacity. And this is his first echocardiogram in May 2021, which basically was unremarkable with normal left side, normal ejection fraction, normal RV size and function. IVC looks normal in size and collapsible, normal right and left atrium, no significant valvular disease, and no pericardial effusion. The first CT was back in June 2021. Basically was just showing some faint ground glass obesities with tree bud appearance and mild bronchial wall thickening, goes with infectious process. There were some non-specific bilateral lung nodules. The largest one was 5 millimeter. And the whole picture was going with infectious process. He got 3 acid-fast bacilli done and were negative. Sputum and Legionella culture were negative. And as I mentioned, his inflammatory markers repeatedly were normal. During that time, he had multiple visits to the ER and family medicine clinic. Multiplex PCR respiratory was negative, including pertussis. And during that time, he received multiple courses of antibiotics and steroids, inhalers, treatment for GERD, supportive therapy for cough suppressant, antihistamine, opioids, all with no benefit, unfortunately. So his cough became worse to the point where he cannot finish a sentence due to coughing. So in August, that was the first encounter to the pulmonary clinic. Our colleagues saw him and for worsening persistent cough, which was causing sleep disturbances, affecting his daily activities, and associated with weight loss. And so at that time, they admitted him and to do further workup. So basically, CT paranasal sinuses was unremarkable. We called our colleague, the ENT colleagues, and his exam was unremarkable in terms of otoscopy and anterior rhinoscopy. So during the admission, he was kept on inhalers, monthly cast with no improvement. We noticed that there are some cases reported that ticagrelor actually can cause coughs, so we changed that to Plavix, however, with no benefit. Bronchoscopy was attempted but aborted as the patient was desaturated or desaturating before the starting of the procedure. And this is his autoimmune workup, were negative. Viral panel, including CMV, EBV, and hepatitis, HIV, all were negative. So he got a high D-dimer, around 4, so he underwent a CTPE study. This was in August, and came negative for pulmonary embolism. It showed some patchy ground glass opacities, more evident in the both lower lobes, areas again of septal thickening suggesting of edema. And here in August, they noticed there is a right atrium enlargement with right ventricle enlargement, and it was prominent with the flattening of the interventricular septum. So the patient was started on IV diuretics with the improvement in his shortness of breath, and his oxygenation went to normal. However, the coughs persisted. So the patient was discharged and continued his workup as an outpatient. We did the second echocardiogram in August, and which showed again normal left side, paradoxical septal motion with right ventricular volume load. Right ventricular systolic function was mildly reduced and moderate to severe TR, and at that time his RVSP 77 millimeter mercury. But remember that in his echo in May, just 3 months ago, was completely normal. He underwent a PET scan, which showed a prominent right paratracheal lymph node, likely goes with infectious process, no neoplastic process. Again, he came in September to the clinic, wheelchair dependent, oxygen connected to him, with a significant worsening of his respiratory symptoms. Actually, you can hear his cough from the corridor, actually. Again, we repeated a CT scan in September, and unfortunately, it showed progression in the size of enlargement pulmonary artery. And at that time, he got features suggestive of pulmonary hypertension, interloper septal thickening, focal ground glass opacities. He got, again, this centrolobular ground glass nodules with enlarged mediastinal and hilar lymph nodes. So immediately we got a call from our colleague, the radiologist, and he said, look, this guy has a PVOD. He underwent a right heart cath. You can see his mean right atrial pressure 9, mean pulmonary artery pressure 68, which is borderline 14. FIC cardiac index is 1.8, and cardiac output is 3.2, and PVR of 16.4 Wood unit. So basically precapillary pulmonary hypertension with, borderline wedge pressure. So at the time we were involved, and they referred him the next day to our clinic. So when we saw him, the patient was still persistently coughing. He was functional class 4, oxygen dependent, 24 hour. And when we encountered him and saw him for the first time, we actually questioned the diagnosis of PVD, because his echo in May was completely normal, and in August he started to have these findings in the CT and in the echocardiogram. So the PVOD was questionable given the acute presentation. Having said that, actually we started him on Macitentan with close observation. He got a ProBNP of 3,000, and within 1 week, the ProBNP went down to 300. So he noticed slight improvement in the shortness of breath, however, no improvement in his cough. So just to wrap up, we have this gentleman, a 53-year-old who had persistent dry cough, functional class 4, wheelchair dependent, with a CT evidence of PVOD, and confirmed precapillary pulmonary hypertension by right heart cath. So, to be honest with you, we were puzzled with this case. So, what to do next? So always we have this meeting, and a weekly meeting. So a multidisciplinary team meeting conducted. No clear consensus about the diagnosis. However, we raised the possibility that the patient might have an exposure that led to an accelerated ILD with pulmonary hypertension, with the findings on the CT. So the consensus was to start him on pulse steroid, 500 milligram for 3 days, then tapered. Of course, he was on macitentan as well. So a repeated echocardiogram after the macitentan and the pulse steroid, it showed actually improvement in the right side function with decrease in the RVSP. His RVSP went down from 77 down to 40, almost by half. Interval decrease in the tricuspid valve regurgitation. So again, he came to the clinic with worsening symptoms, with cough, shortness of breath. Prednisone was tapered till off, and on that visit, he developed a new right-sided neck swelling. So CT neck arranged to be followed by ultrasound-guided FNA. The CT neck showed some lymph nodes with a few others in the superior mediastinum, scattered in the deep neck spaces. And again, they raised a possibility of inflammatory / infectious process. And given the appearance of the lung parenchyma, findings again in favor of infectious inflammatory pneumonitis. So he underwent a lymph node biopsy, which showed, unfortunately, metastatic adenocarcinoma consistent with pancreatic origin. CK7, CK19, mucin 1 were positive. So the patient was electively admitted. The cough didn't improve. Now he was functional class 3, oxygen dependent, and we arranged for a CT abdomen, CT cab, chest, abdomen, and pelvis. We didn't find anything by the CT apart from non-specific faint area of enhancement. However, no pancreatic lesions. Again, there was some enhancement in the liver. It showed some tiny hepatic lesions. However, no evidence of abdominopelvic metastatic disease. Persistent mediastinal and pilar lymph nodes and supraclavicular lymphadenopathy. And again, it showed some bilateral multifocal centrolobular pulmonary disease process that actually was against pulmonary nodular metastasis. And, but it showed in the CT multifocal small osseous sclerotic foci compatible with metastatic disease. This was his CT at that time. He complained of back pain, so we did an MRI of the cervical, thoracic, and lumbar, which showed metastatic disease with no evidence of pathological fracture or cord compression. Then finally, we did an MRI abdomen, which showed a focal pancreatic neck mass without pancreatic ductal dilatation or abdominal lymphadenopathy, some hepatic meningioma of his hepatic lesions, and it showed thoracic and lumbar supine metastasis. He got pancytopenia, and unfortunately, we did the bone marrow for him, and it showed infiltration by the malignant cells. And it again, it was a positive for CK19, 7, and mucin 1. So what do you think his final diagnosis? So we diagnosed him with metastatic pancreatic CA with pulmonary tumor thrombotic microangiopathy, PTTM, and lymphatic carcinomatosis. So just I will give you a brief about this rare entity, tumor embolism, tumor pulmonary embolism studied in the autopsy series of patients. Actually, it's usually associated with solid malignancies, and the prevalence between 3 to 26%. It carries very poor prognosis, can be associated with dyspnea, chest pain, fatigue, and cough in 8%. pulmonary hypertension is a common finding. Usually it's associated with GI malignancies, including hepatocellular carcinoma, breast cancer, kidney, lung, stomach, colon, and prostatic cancer. This is really nice case. You can see in the image here, and this actually demonstrates a filling defect within the right pulmonary artery and lobar branch. In image P, you can see at the level of the interventricular septum, it shows the irregular mass passing through the tricuspid valve. But if you can see in this image, this is actually a tumor was extending from the kidney through the IVC, reaching the pulmonary artery, and it's reached through the IVC and the right side of the heart, reaching the branches of the pulmonary artery. So all this actually, it's extended thrombi. So is there any difference between PTE and PTTM? Yes, PTE is a condition that related to PTTM, but it's quite distinct in terms of pathophysiological process. PTE actually can immobilize and metastasize to cause and trigger the local coagulation system and cause mechanical obstruction. So this is why we classify PTE under group 4. And in PTTM, and this is the case in our patient, we have a remodeling of the pulmonary vasculature. And actually, in PTTM, it can occur both with and involve both the pre and post capillary component. So the patient and this condition can straddle several classifications. We can put this patient under group 5, multifactorial mechanism, group 1, as the patient can present with PVOD, and group 4 as a CTEF. So for the diagnosis, you can do CT, a VQ scan. The VQ in the tumor imply patient, the tumors actually typically numerous, symmetric, and peripheral. And the gold standard actually is to do a right heart cath, and during the cath, you aspirate blood from the wedge position and send it for the cytology, and you will diagnose the patient. So they stated here the complex involvement of the pre and post capillary vessels, as well as lymphatic vessels, might make the PTTM unclassifiable in the current classification system. Treatment is by treating the underlying malignancy. No treatment has been found to be of significant benefit, and unfortunately the disease progresses rapidly and is often fatal within a few weeks of presentation. Back to our patient. So he was evaluated by the oncology team, started on palliative chemotherapy. Unfortunately, the patient had progressed clinically and radiologically. He was signed DNR and referred to palliative team, and later on, unfortunately, he passed away. And by this, I conclude. Thank you.
Thank you, Dr. Al-Ahmadi, for this really interesting case, although with a bad end. Please join us here. I know that we are over time somehow, so we will take some questions and comments. Please quick questions so that we can share all the speakers with their response. They were really very interesting cases, all of them, for sure. But I don't think that we will have time to go deep. Please, let us start by our colleague here. Go ahead.
One question for Dr. Nawal. For your case, congenital, any transthoracic short at the level of the aorta?
Dr. Rafai, you don't ask me. You just contribute to the case by adding more information from your perspective. Go ahead.
From the echo-wise, we need to see the transthoracic short axis at the level of the aorta and pulmonary artery, and we use continuous wave Doppler, especially if we see like the right ventricular systolic pressure is very high. So either this is pulmonary hypertension or pulmonary stenosis or double chamber RV or stenosis of the peripheral of the pulmonary artery. The other point we used to do transesophageal echo to exclude occult shunt. And sometimes we have to use contrast saline, agitated contrast saline or contrast to see any small shunt, BFO, or unexplained shunt. That's
I agree, and it possibly would have been better and more helpful if I brought also the Doppler images, echo Doppler. It would have probably showed some residual shunting.
Okay.
Thank you.
Okay, go ahead.
Two questions, one for Dr. Nawal, one for Dr. Faiz. It's very unsettling to have misinformation and lack and obscurity of the history, which could have changed the course from the beginning. But during examination, I mean, wouldn't you isn't there any sternotomy wound that would explain, because maybe it's done to childhood and it kind of really faded, but it's really sad. Withholding this information could have totally changed the course. And Dr. Faiz, my question for you, please. Hindsight is 2020. It's so easy to criticize what should have been done. But in case of sake of argument, in your opinion, could anything have been done in May that could have helped in changing the outcome? That, يعني قدر الله ما شاء فعل, and that's we don't debate that. But in retrospect, is there anything you would think that I hope this was done to alleviate? Thank you.
So no, there were no signs of previous surgery, and I was looking for some chest deformities, probably causing some hypoventilation that may lead to group 3 pulmonary hypertension, but no, there were no murmurs audible, and there was no signs of any surgical scars. I don't know if there are any cardiac surgeons here, but would such surgeries at early childhood, at the age of 2, leave any scars like the ones we see in CABG and X-rays in our adult patients? I don't know.
Just a quick comment. So the patient had a right PT shunt. That means he had desaturation. So he had a severe PS or sub-PS or whatever. So PT shunt could be done lateral thoracotomy. So you could do it without a scar. Now, the second operation, which looked like a very complex one, which is an RVOT, I mean, resection of RVOT and then directing left pulmonary artery to that means that it was a small left pulmonary artery or a stenotic. This is why they directed the surgical node to this way. Then I think the CT scan done, usually it's a classical CT, so it's a right arm angio, and this will be affected the distribution. If you did not time it, you could miss really the small left pulmonary artery. It's not occluded. Now the pressure explanation is very simple, that he had a branch-based stenosis, which is usually the catheter we usually go to with the right heart, you go to the right side, I mean with the RBA, because it's easy to go to right pulmonary artery. The wedge pressures could be taken from there. So most likely his pressure always, I mean his problem will be delineating the left pulmonary artery by a direct angio or looking to his MRI if possible. We face it always. The problem is that once you have a child with a complex congenital heart disease who never follow up, he just come to you you need to just use whatever available to you. So I mean, it's not unusual. We face 45-year-old who had a surgery with her family in US at age of 8 years. She knows nothing. She's a graduate. She's a master degree. So she said, you need to figure it out yourself. So we started with this. So this is the usual in adult congenital. It's not unusual to see these cases. So I think what you did was the right way to do it.
So honestly, Dr. Khaldoon, we looked to that patient from every aspect. So first of all, we approached the cuff as usual from every aspect. We tried to correlate this with the AstraZeneca vaccine, as he received the vaccine 2 weeks prior prior to his cough. Dr. Dal'an called the cardiologist because, if you remember, he underwent PCI with a stent. We looked actually into that. The cardiologist looked into that. Is the stent itself might migrate or causing perforation or causing cough by itself? Again, was not reported and very rare. And remember, his echocardiogram was completely normal in May. And in 3 months' time, he really progressed with positive echocardiogram, positive CT features, and we confirmed the diagnosis with the right heart cath. Then at the end, he got this mass in his neck.
If you allow me, Badr, I think Khaldoon's question was for the CT scan changes, the earlier one. Would it have been better if we have done a CT-guided biopsy, for instance, or anything like this? I remember this patient was originally seen in the general pulmonary clinic, and these small, faint ground glass changes did not really require intervention. And the radiologist interpretation initially of those faint changes were probably an infectious process, and he received multiple antibiotics and so on and so forth. But when he came to our clinic, in the pulmonary hypertension clinic, he was already then having some interstitial lung changes, and he was taken by his primary pulmonologist actually to do bronchoscopy, to have a biopsy done, but he desaturated the procedure was aborted. Also remember that PET scan was negative. Even PET scan was negative. So if there was any a hot spot somewhere in his body, it would have been discovered by PET scan. But it's not unusual to see these rare cases of diffuse malignancies of unknown origin sometime that present already with lymphatic spread. So this has been all taken in consideration, but he was very unfortunate. And by the way, the lymph nodes that were clinically apparent was only happened in the second visit in the pulmonary clinic in August. So even that was very unusual presentation. I think everything was exhausted for that patient, but he had very aggressive malignancy, unfortunately. Thank you.
I think very interesting cases, and we would like to continue the discussions about them, but I think because of our time, we will have to conclude here. I would like to thank all the presenters. They were really very fascinating cases. Thank you in particular to our colleague came all the way from Caib and shared really this case with us. It is really, really important to know such immune deficiencies and so on. So thank you, really. Thank you all. Thank you to
announcement
to all of you for attending, being with us till this time. There is an announcement that we have to hear, and I hope to see you tomorrow, inshallah, in day 3.
Thank you very much, Chairman, for doing a great job and finishing really on time. Thank you very much. The announcement is for our dinner tonight. The whole group will meet at the lobby between 7 to 7:15. Then we will walk down by the gate, by the hotel entrance gate. There are 3 big, huge buses, red colors. So please, some of the people are maybe left from now. If your friends or colleague, invite them to come along. The dinner will be at one of the museums in Jeddah. It will be a surprise, very nice dinner to all. So we are all welcome to our dinner. Okay? Buses will move around 7:30. So please be at the lobby around 7:15. Thank you.